Good morning to everyone trickling in, or good afternoon, good evening, wherever you might be joining us from. We are back on the bottom at East Pacific Rise, down 2.5 kilometers below the surface. As you can see on your screen, we have returned to some of these student experiments where we left different types of food behind to see what the local fauna might or might not be interested in. So we had fat, meat, and liver, I think, on one side, and then got a potato, some other vegetative matter. Maybe, do you remember what the other vegetables were?
Beans.
Oh, potato and beans, right. And then they also left behind an experiment with just beans, different types of beans, to see how those might be impacted. But as you might see from the title of today's dive stream, we are ultimately hoping to collect mesh box experiment number 1 up off of the bottom. So there are 3 of these experiments in place, and this will be the first that we are collecting back. Again, these experiments are a mesh box that is sealed down to the seabed, over top of a venting area, and we are looking for evidence of fauna being carried up through the venting fluid. So it'll be a bit of a process to get these boxes collected, but we'll see how that goes. As folks are trickling in, if perhaps this is the first dive of this expedition that you're joining, we are at East Pacific Rise, so we're about 2,000 kilometers to the west of Costa Rica and 1,000 kilometers south of the Mexican coast. This area is hydrothermally active, so there is venting. Yesterday we saw some black smoke chimneys, but we've been working with more diffuse venting. And we have been deploying experiments as well as taking samples. So today will kind of be a combination. We're going to be collecting back an experiment that's going to involve some sampling of both the fluids contained inside the experiment before we pick it up off the bottom, but also I believe some sampling of the substrate underneath that experiment, of the rock. Here we are at some more of the students' experiments, as well as a sculpture by our artist at sea on board.
No, it's just a chicken. This is the highlight frame grip.
So again, we left 2 sets of these students' experiments down here. We left one at the
And if you look at the
this
art,
venting site, and one at a non-venting site, which we just were at.
he was just in here, Max. Yeah, he was just here.
Max, tell me what to do. How? Yeah, limpets. Limpets colonizing your stuff.
Limpets are good. Like, yeah, that's, I mean, if we pull out a little, we can see those guys dangling over it. So, well, yeah, I mean, that's perfectly framed. That's really, no, it was perfect. Perfect, just a little basically, so we have just to that. Sorry, a little bit back, so we can see the edge of that, just the edge of the sculpture. A little bit back. Yeah.
This we stay a few minutes or no?
Yeah.
Okay.
Then we're good.
And this is a sculpture down here from Max, our artist on the ship right now.
Also, wait, the other
And
question is-
we are planning to recover this, I believe, but just kind of checking it out right now as long as these kids' experiments are here.
A little bit, but it's not really not great.
Well, the camera doesn't move left, so if you zoom in, it's just going to zoom towards the center.
Oh, yeah.
But we can't.
That's good.
I do believe we took a temperature sample here way back when we dropped stuff off, but I can't recall what it would be off the top of my head.
Now I feel bad. Now I feel bad removing this. Everything looks so. Yeah, I mean, it's fully like.
It's also red. These are also masks here.
The whole, I mean, it's really.
They were going over on this because this is the end is here. And
Yeah.
they are moving here. They go over.
I mean, it's doing exactly what we wanted to do, or what I had wanted it to do. But
I will.
are they going to be sad?
So for the experiment, when we do collect that up, if you've been around for its deployment, you're kind of familiar with it, but for anyone who hasn't, it is a mesh box, so a mesh-walled box, but there is then a second wall outside that's acrylic with some holes in it, and we are going to need to puncture through the mesh so that we can sample the fluid inside. And that's just in case there's anything in the water there that has not settled, but also kind of curious just, what the inside's looking like, if anything's being significantly changed by the fact that it's somewhat enclosed. So we're going to break through the mesh, sample the water from the inside. Then we're going to actually pick the mesh box up and secure that. There's also a chain with some sponges underneath the mesh box to act as a substrate for some settlement there. That also has a temperature logger on it, so that'll give us data from the time that the experiment was deployed, or the period of time that the experiment has been deployed. Do some more water sampling, and then also do some rock sampling. And the rocks, we're going to see how that goes. The rocks around here have been kind of finicky. A little tougher than you want, but then once you actually have them in hand, a little more crumbly than you want, it seems like. So we shall see how it goes. But yeah, it'll be interesting when we lift that box up. Everything was stained. at least as per design, everything under that box should have been stained. And so we are looking for anything that has settled out that doesn't have that stain, right? What new things have appeared there since we've put that box over top of the vent there? And that's going to be very interesting, I think.
And then if we're lucky, we can roast it. Thank you, guys.
Oh, we got someone asking. Excuse me. We have someone asking about like whether a crab or something down here would have barotrauma. So a lot of the critters down here are more protected against pressure changes. That's not to say that it wouldn't be bad for them to be brought to the surface and be in an entirely different pressure, because obviously they operate under this pressure for their life. But barotrauma is most evident in fish that have swim bladders, because those are pockets of air within the fish, and so as you bring that fish to the surface, that pocket of air expands. But if you've listened, or if you were around for when we gave a tour of kind of the science going on back in the lab, a lot of the work here involves putting the organisms back under pressure. So if we can't, keep ambient pressure from the bottom and bring it up. But it is all right for these critters to undergo the pressure change for a while, and then once they're up here, they can put them in a pressure housing and pump that back down to the pressure that they're used to. Yeah, if you're a critter that doesn't have like a gas bladder or something, you're a lot less susceptible to the really substantial barotrauma. But that's not to say that you want to be taken from 250 atmospheres to 1 atmosphere.
Where should we go?
So here's another sculpture from Max. He's voicing that it would be fun if one day we just showed up here and it wasn't there. Just had gone somewhere. Lots of eel pouts here. Around here, these are all a type of eel pout, which not actually eels. we do that with a lot of things. We call lots of things that aren't fish. We call things that aren't eels eels. You got like whale sharks, right? Not a whale. We don't have a ton of consistency when it comes to naming these things, but eel pouts are the type of fish here.
Forget when we had to like lower the ISO, get the camera.
That's the lowest ISO. Yeah.
We raised it. What would you remember, right?
So that raises, just makes it pull it out.
Well, then we turn the lights on, and then it will show.
Okay. All?
Yeah. Good shot.
Yeah, got a little there. So this sculpture does have some glowing to it. So we're just trying to get a fun shot of that before we move on. There you can faintly see it in the dark. There it is.
Yeah. just swim across it.
That's cool.
Not bad.
No, yeah.
It's just hovering. Go the other way.
Yeah, just resting on it. It's up against the worms, yeah.
That's like how you. That's kind of how you know there's life. Life just. Do we have a?
That's fun.
Okay.
Mm-hmm.
Yeah. We got the tail at least.
So you have enough footage of everything?
Something drifting across, yeah. That's fun.
Now we can just turn the, can we turn the light on and regular just for one for 30 seconds more. Yeah. The marine snow is really.
Looks like he's in the hand of your
Yeah.
Yeah.
Yeah. Oh my god.
Just hanging out. Yep, so someone notices our temperature's bumping around a little bit. Yeah, we are going to be seeing anywhere from like 1.8 C up through 2 and some change. That's ideally what we'd like to stay at, but it will bump around. It's not static down here, especially as the ROV is moving. Of course, we have thrusters, and those thrusters pull water around, but when we're touched down on the bottom as well, if we're touched down by some of the diffuse venting, that will bump us up. And if we're in some of the venting, it's all right as long as it's this sort of cooler venting and not super warm. So you'll see that temperature now that we're up off the bottom. It's dropping down a little bit there, but as we move around, you'll see it kind of fluctuate through the dive. So anytime you're seeing shimmering water, that's venting of some manner, at least as far as it's coming out of the ground. When we open some of our items, occasionally you'll get some shimmering water coming out, and that's usually going to be a halocline difference, so a difference in salt salinity. We fill a lot of our stuff with some fresh water up here so that it's got fluid in it, it's non-compressible. And then when we open up those containers down in the salt water, you kind of get a cool shimmer up through it.
Yeah, no, of course.
So you can see now that we've come up off the bottom, we're down back towards about 1.8. But even as we fly along, there's been a couple times we've kind of gone through these plumes of shimmer, and it's bumped up to 2 and change again. There you go. Just like that, as we were passing through that little cloud of smoke, I suppose.
Need to go dry those 2 worms. Yeah, in the sun they work. Dry some of them. Great.
All right, so here are the experiments. Again, for anyone joining us, as we're seeing folks still trickling in early in the dive here, we are back down on the bottom of the East Pacific Rise, 2.5 kilometers, and we are looking to collect experiment number 1. So from our view here, that's the FAR experiment. But these boxes here have been sort of sealed down to the bottom and left to hopefully accumulate some fauna that might be coming up through the venting water itself. So that's what these boxes are made to check or observe. And we are going to be collecting 1 of them today. So one of the things that we've been noticing with these boxes is the growth of bacteria on the side.
This is number 3, okay?
Yeah, much more bacteria than we anticipated. I think, what, 3 was placed down
3 days ago.
3 days ago now, okay. So just within 3 days we've got some growth.
Highlights frame grab. It looks like on the top it's not as much. Oh, there's also more food. Can we take another highlight frame grab? Yeah. And another highlight. You can still see the flow coming through, so that's good. Okay, let's look at the bottom. So fast. It's not really changed. It's good.
Question about how we can collect samples, especially this one is asking benthic organisms if we use the like grab samplers. Yeah, we have manipulators. We have a port and starboard manipulator. Those can be used to collect rocks, or we've used them for corals, sponges, worms. We've been collecting some of these large tube worms this expedition as well. And those manipulators can also be used to operate other methods of collection. So we have a suction sampler, which is kind of like a large vacuum cleaner. We can use that to suck up sediment or suck sediment off of rocks, or we can also use that to suck up smaller organisms or organisms that would be not great in the manipulator, like if they're particularly gelatinous or something, we can get them with the suction sampler. We also have push cores sometimes, which are tubes that are closed on one end. They've got a little handle, and we can use the manipulator to actually press those tubes down into the sediment where there is thick sediment, not here obviously. This is pretty much bare rock in a lot of spots. But then when you pull that tube back up out of the sediment, it'll contain within it a little core of sediment. So we have all sorts of different ways of collecting from the bottom, and then we can also use a couple different ways to collect things in the midwater, including the suction sampler again, and Niskin bottles, which are an open cylinder. We have a couple of them mounted on the side of the ROV here, and those cylinders can be slapped shut by the pushing of a little trigger. When they do that, they sort of capture whatever water is inside them at that time.
Sorry?
If they sit on the
Inside they are happy.
no, outside.
Oh,
if they
outside
sit
also,
on the-
yeah.
if they sit on the biofilm, the big one, this is like sitting in food.
Yeah. Okay, you can also make a highlight frame up here.
This is way better, no? This is way better.
18. Well, okay, so the box that covers it
Yeah.
is 18 inches square. So it's just a bit smaller than that.
Okay, oops, sorry. We can make a highlight frame up from that box too. Overview. And then we can pick up the frame and we take it with us to the box number 3.
Yeah. Yeah.
And that's also outside. Yeah. Yeah, better than 18.
So there, we were trying to figure out the dimensions of the box here. Well, you were busy over there. What do you know what the dimension of this box is?
50 by 50.
50 by 50. Okay, perfect. 50 by
50.
50 centimeters is what we were looking at here.
We're just gonna hold on to it.
Yeah, we just bring it to experiment number 1. And at the end of all the things that we do today, we put the frame on that we find it back tomorrow easier. That's all.
And you might have noticed
So we can just place
on the
place a
box when we
summary.
were looking close, down at the bottom, there's kind of this little transparent buffer there where it's not quite sitting on the bottom. That's actually on a silicon seal that's on the box itself, and it's pretty squishy. We've seen that. We actually had one where there was some damage to the seal, but then when we pushed it down onto the bottom, it sort of smushed itself back together. So again, we want to seal these boxes to the seafloor itself.
We want to look at the other side of box 2 and 3. Not really, good. No. Yeah. Yeah. So we can go to number 3. Number 1. Sorry.
Seems like we've got some new folks tuning in, so that's exciting. If you've never been on for any of these dives, this is live footage. This is actually happening right now. And although the vehicle is down 2.5 kilometers, this is the remotely operated vehicle named Sebastian. We're actually up on a surface vehicle, the research vessel Falcor 2, and that's where Sebastian is being controlled from. So we have a very long umbilical tether that connects Falcor 2 all the way down to the seafloor and Sebastian. And the pilots here on Falcor 2 send commands down to Sebastian. Sebastian sends data and video back up to us, and then we can send that even further out into the wide web, where we love for folks to jump on and explore with us.
We're looking at it from the side of the angle. You mean like yesterday? Yeah, that's fine. Yeah, as long as she's seeing two sides of it, and I try to give her like a corner so you can see two sides.
So since we've got some folks kind of asking about the ROV, let me show us around the ROV just a little bit. This is the main camera view that you're seeing there. That's 99% of the time that's what we're going to have up for you. And this is also experiment 1 that we're settling down in front of. This will be what we're collecting today. We also have the situation camera. This is more for the pilots to kind of see what's immediately in front of and below the front of Sebastian. But then we also have a couple other little cameras around to either side. So there you can see the outline of that science camera. And this is another one up above looking down. So again, you can see the science camera, and off to the right side you can see a rectangular frame that's being held by one of the manipulators. We have this one as well with a better look at the science camera. And also behind the science camera there on the right, you see some clear tubes. Those are the collection containers. Those are the other end of the suction sampler. So when we use the suction sampler to suck up stuff, it goes into those cylinders back there, and the pilots can rotate those cylinders around to select which one we are actively collecting into. There's another camera that we rarely show, but we always have eyes on, and this is the camera pointing directly aft of the ROV, so right behind us. The pilots use that for just kind of keeping situational awareness of what might be around the ROV, but every once in a while something cool will show up there as well. And then we have some smaller cameras. Let me bring those up for you. So we have 3 of our 4 on right now. We're just not using the 4th. Oh, Jill's gonna turn on the 4th for us there. There we go. So we have, I'll just kind of walk through them. We have what we sometimes will refer to as the bullet cam. This is actually where that umbilical connects into or attaches to Sebastian. And so obviously an important point for us to keep an eye on. We have, this is just kind of a camera we can move around. I want to say in Octopus Odyssey, the previous expedition, we had this mounted kind of down by the side so that we could see the heat flow probe that we were pressing down into the sediment. But in this expedition, we've had this mounted where we can see a sample bag. So this is a bag, it's deflated right now, but this will ultimately hold 55 liters of water. We'll be using that today. We have another camera that shows the slurp sampler or the suction sampler containers up close. So if we're trying to collect something small, we can kind of watch this screen and see when it actually shows up in the suction or from the suction sampler. And then this was the one we just turned on, and this is a camera on the starboard manipulator. So when that manipulator is doing things, the pilots can kind of use this to maybe better scope out where something is. We've actually used this to look under things a couple times as well, which is fun. But this is science camera, and this is the primary view we use.
other side. Yeah. Good. So, I want to settle away all the questions. First thing is that we put away the frame just out in our way. And afterwards, we'll take what's the name of it? Finito knife, the knife, like the
So we're primarily seeing, I guess, 4 main organisms at these sites, but there are many other organisms. So we see lots of worms, first of all, and even just here we have 2 main types of worms in our view right now. We have the large tube worms. These are Riftia tube worms. They're the ones that kind of have the red gills sticking out. They look something like a lipstick. But there's also smaller tube worms, and those are the little white doodles that are all over the rock. Then we've had the eel pouts, the long eel-like fish. Those are cruising around. And we have mussels. These are kind of like those golden brown bivalves. There's some amongst the larger tube worms there. Then we've got a couple different types of crustacean. So over on the right side, you can actually see like this little cloud right above our landing skid there. You can see a little cloud of things cruising around. Those are tiny little amphipods, and we see their swarms around the site pretty often. Then we have some larger crustaceans. So over on the box right now itself, we have a crab. And down on the seabed, in between the box and the segment or the clump of large Riftia tube worms, there's another little crustacean there, and that's a squat lobster. And you will frequently see those kind of shooting up and into the water column. A lot of folks see them and they think maybe they're like a little squid or an octopus or something, because their legs sort of dangle behind them and their body is quite round. We have seen some octopus around here though as well. But those are probably the most common things that you're going to see.
But at that point, I think we're safe. We're not going to be pushing the box. Everything will be frozen and steady.
So it's just important to be steady with the rope.
Yeah, I have one concern. When these arms freeze, they sometimes drop or raise a little bit, and so I'm afraid we'll hold with one arm, get it steady and puncture. Either that arm has to stay live, which is not a good idea because any little movement in this. So, but when I freeze, it's going to want to move, and when I lift this, I'm afraid of it picking up. So I'm just a little bit worried about it as well.
What if you like stabbed it and just did a script? So you're still using the weights to keep it down. So when you freeze it, because it's going through the plastic walls.
It's possible.
So the pilots and the scientists are currently discussing how best to approach the sampling and collection of this experiment.
The big thing. Probably put more like, if it's possible, we function that more on the lower side.
Yeah, so
So when it comes up, it's not
my other
lifting.
thought too is like before I actually puncture, I can do a test freeze and see which way it's going to go. If it goes down, then yes, I would put it on the bottom. That way it wouldn't be able to go down. If it wants to go up, maybe I could correct for it a little bit before it goes. But we'll just take it slow and work through it. I was just expressing my concern with it.
Yes.
Another question about
This is what it says.
how the organisms here might respond to the light. Some do, many don't. There are many organisms down here that don't have eyes, or even those that have eyes, we're not really sure how well they see with them. We were talking just the other day about a shrimp that it clearly had eyes, but I guess it's still unsure or uncertain about whether or not it really uses those as an adult. It's possible that as a larvae, when it's floating up in the water column, it might be in a shallower area where the light could be useful. But where we are now, there's no sunlight reaching these depths. That's not to say there's no light at all. Bioluminescence, right? Organisms making light is a big thing in the deep sea. That's the way that many organisms here can either find a mate in the dark, or they can attract food or they can even prevent themselves from becoming food, all through strategic use of bioluminescence, of being able to actually glow or produce glowing. So some animals do respond to the lights every once in a while. Sometimes the fish seem to be a little bewildered by it. We haven't seen too much in the way of that here. None of the critters we've been by have had particularly large eyes. Sometimes you'll encounter deep sea fish that have very large eyes, or deep sea organisms that have very, very large eyes. And those, again, would be very sensitive to the faint light of bioluminescence, but then when the ROV shows up with its lights, that's a lot more than the bioluminescence it might be used to, so it can probably bewilder them for a little bit, but we usually are moving on.
Land like in the middle of it, or I can move it if you would like. I'm moving it left. It is, but you can still ram in, so I think you'd be okay.
We have not come across any brine pools at this site, no. For folks curious what that is, it's an area where there's fluid that is much more saline, a lot of dissolved salts and minerals in it, but
It is too far.
because of that, it's denser than the water around it, and so if there's a depression, it can kind of settle into it, and it almost makes like a lake or a pond, or, as the name implies, a pool on the bottom of the ocean. They're quite cool, but here we've mostly just been looking at venting, where, again, water that has a lot of dissolved material in it is coming up from the bottom, and that is heated water, so it is rising up and not pooling like that.
I'll work on this.
I'd love to see a brine pool, though.
Can I move your color or your 4K? Sure.
All right, so folks that have been along on some of these earlier dives, this might look a little familiar. We are getting the probe ready that pumps into this bag, but the probe is actually going to look a little bit different today. You might notice the tip has another attachment on it. It's kind of angled now. So in essence, that is a hypodermic knife, or a large needle, a hypodermic punch, I suppose. And so this is intended to punch down through the mesh of the box so that we can suck the water out from inside without lifting the box up just yet.
If you know how with this, when we're going to put this in, we use the other arm to kind of just hold the top. Would that same method be possible? Just hold the top,
Yeah,
suction sample?
Just then, like this way, because you have to get inside.
Or do you want to have
Question in the chat of
No, it doesn't matter, like
what
how
the
it's,
structures, the little white worm-like structures are all over the bottom. Those are, in fact, worms. Yeah, we've got a couple different types of tube worms here. We have little tiny tube worms, which are the kind of white doodles all over the ground, and then we have much larger tube worms, the Riftia tube worms, and those are these large white stalks on the ground. So two different types here, but yeah, they are not just worm-like, they are in fact worms. Lots of worms.
And then we want to basically hose.
Another question of whether or not we've encountered any debris down here from humans. Sometimes we will find, you know, cans, soda cans, or I think we found like a boot before and that sort of thing. On this particular cruise, we have certainly encountered things left behind by humans, but most of them intentional. I would say I can't think of off the top of my head anything that was like, whoa, someone dropped that overboard. But we are finding a lot of ballast weights, and so you can find little blocks of metal around. The other day we found a couple bags sitting on the bottom. These are weights that other submersibles have used to get down here, and then they drop them off before they return to the surface, and they kind of corrode and dissolve away in the water over time. But yeah, we found those. Yesterday we visited some black smokers, and there was a temperature logger at one of them, and then there are also little scientific markers around, sort of noting other experimental sites. So we have encountered many things down here from other people, but not so much in the way of just trash or something like that yet. Not uncommon though, not terribly uncommon, which is a sad thing to say, but you will occasionally just come across some sort of trash that went overboard. Currently, the pilot's just working to get this loop out of the hose there.
I really would feel better if we pulled that out. Okay, go ahead and push in now, and I'll follow you back. I just want to keep that kink out of it.
So we're coming up on about an hour here. So for anyone who's joined us since then, you're curious maybe what we are watching, what's going on. I see a question about what the experiments, the contents are going to maybe tell us. So a little overview here. We are 2.5 kilometers down below the surface of the Pacific Ocean at part of the East Pacific Rise. So it's a hydrothermally active area. Hot fluid is venting up from the seafloor, and there are organisms associated with those venting sites. So, for example, these large tube worms, the Riftia tube worms with the red gills sticking out from their white tubes, those are pretty well associated with venting sites. But scientists are curious, how do they get from an established clump at a older vent to a new vent that doesn't have the animal, but how do they show up there? And so one idea, of course, is that they could drift through the water column and just randomly happen to land at vents, and those that get there are lucky, and those that don't aren't lucky, I suppose, are unlucky. But also these vents, right, there's water coming up from below the ground, but that water originally has to go down below the ground from above it. So there are areas known as recharge sites or recharge areas, and that is where water is actually drawn down into the ground At some point it is heated by the thermal energy below the surface of the Earth, and then it comes back up, and where it leaves the ground again, that is a vent. And so if you had the little fertilized zygotes of the tube worms, or little larval tube worms that had not yet settled out, but if they somehow got drawn into those recharge sites, survived the process of traveling through the ground and being heated, right? They weren't too heated, and then they were expelled out where that water leaves the bottom again. That means they would be expelled directly into a venting site, which could be a prime location for them to then settle and develop into an adult tube worm. So, to test this hypothesis, or to look for evidence in support of this hypothesis, these experimental boxes have been placed down on the seafloor. They are sealed to the seafloor. So we placed down some silicone caulking, and then there are also little silicone seals on the bottom of these boxes. And the boxes are mesh walled, so the venting fluid isn't like building up pressure inside the box, but the mesh would stop anything larger, any fauna from coming out of that box. So the boxes were placed, they were sealed down to the bottom. There's some other little instruments inside the boxes that can help collect some of the animals that might show up. And after the boxes were placed, a dye was pumped into the boxes. And this dye was meant to stain anything that was at that site at the time the box was placed. But that means that if anything shows up afterwards, if anything comes up in that venting fluid, it won't have that stain. So what we're looking for today is we're going to collect some of the fluid from inside the box first. Then we're going to lift the box off and put that on the front porch of the ROV here. That box has a lot of bacteria growing on it that scientists would love to see more of and learn more about. Then we are going to collect a little bit more water around it, and we're also going to try and collect some of the rock, both rock on the inside as well as the outside, so we can compare and contrast those and look for evidence of new organisms that aren't stained. So that's the gist of these experiments, and this is the first one we placed, experiment 1 of 3. From the foreground to the background here, we have 1, 2, and 3 experiments that have been placed. We're going to pick up number 1 today. And then, time permitting, we're also going to go around and maybe collect some other worms and mussels in the area.
Yeah. We are still rocky. Okay, where is she want to go?
She was talking about going like straddling the bridge, which would make us good front to back, side to side.
And this is all being done right now. This is footage is coming to you live. So the remotely operated vehicle Sebastian is wired all the way up to a surface vehicle, the research vessel Falcor 2, where we are watching this footage, and we have pilots piloting the ROV. There are scientists on board, obviously, who are able to look at samples when they come up, and right now they're actually conferring with the pilots to figure out the best approach to this experiment. And we can also then push that footage out to the web. So the general public, we love for folks to take an interest in science and deep sea science, and so an engagement there is fantastic. And it's also a useful tool to be able to push this out online, because sometimes there's scientists that want to take a look at stuff, but they can't be on the ship. And so it's great to be able to expand our reach that way as well. So at these communities, the primary source of energy will be the chemosynthesis. That's what the tube worms, for example, kind of indirectly are feeding off of. So the tube worms and several of the organisms down here, the mussels as well, they have bacteria that they have relationships with. And so the tube worms in particular are fun because when they start out, they have a digestive tract. When they're little larval tube worms, they have a digestive tract. But they will lose that digestive tract. They will not be able to feed themselves. But what will happen before they do that, hopefully, if everything works right for them, is they will become infected with a symbiote, a bacteria that will live in them for the rest of their life. Quite a lot of it, actually. And that bacteria is able to use the chemicals coming up in this hydrothermal venting fluid. So those bacteria are chemosynthetic, or they're able to chemosynthesize, so take the chemicals that are in the water and convert those into energy that they can use, but an excess of that energy will then go to the worm and keep the worm alive as well. So where these worms are growing, where the riftia worms are on the bottom, those are areas where venting is happening. So kind of an interesting indicator, and you can see the worms sort of follow these cracks along the bottom, and some cracks don't really have worms, but the cracks that do, more venting there perhaps, and that provides a food source for that environment there. Detritus is certainly another food source for the deep sea, but one of the things you'll see around here is not a lot of collected sediment. There is sediment, but something like an abyssal plain where it's very deep sediment, lots of detritus settling out there. You'll find lots and lots of holothurians or sea cucumbers, which will feed on that detritus. We've seen a couple holothurians around here, but the chemosynthetic environment is what we are interested in with this cruise.
I think this is what we're going to have to deal with.
Yeah, the little doodly tube worms all over, the tiny little noodle ones, those are a different type from the large riftia. Yeah, different tube worms. And you do see a lot of stuff drifting around in the water around here. Some of those are little critters, little amphipods that are swimming around. We'll also see little bits of like bacterial flocculate that's can get kicked up either by the ROV or break free from a vent and get pushed up into the water, and that flows around, and that can be a food source for many things. Many of the crab around here, for example, will eat the bacteria that's growing on the ground or near the vents, or bacteria that might fall out of the sky that got pushed up into the water column by venting. So, interesting environment.
I feel like it's shaking. Doesn't it look like I'm moving it? Well, you're not. So.
A question about how long these experiments have been here. So these were placed, the most recent one was just 3 days ago. That was box number 3. Box number 2 was placed 5 days ago, and box number 1, this one was 7, I think then, so we're like a week out. So they've been down here just about a week. But even the one that's only been down for a day has a pretty incredible, or excuse me, not 1 day, but 3 days, has pretty incredible bacterial growth on it. So we're ready to collect these because the bacteria is really grown on them.
Yeah, I'm just concerned that I'm ruin the experiment somehow. Like it's already kind of moving it. Are you guys comfortable for me to go ahead and poke this in? It's probably not going to get worse.
Or you can also do if you're poking, then it actually can get loose again. Because once it's in, I think it should be
Okay.
hard. That's also, the silicon is like really wobbly, so you see some movement, it's still on the silicon.
Right.
So you really have like at least a centimeter that, it wobbles above as soon as you touch it, it starts to, to move, but not on the silicon.
Yeah.
Just because the whole thing is like
It's got some flexibility to it.
yeah.
All right, so the pilots are again trying to figure out the best way to approach this, working with the scientists. So this probe in our left manipulator has a sharper skewed tip, and we're going to puncture through the mesh with that tip and start sucking out the fluid from the inside of the experiment.
Okay. Freeze, see it comes up. Okay, so that actually did work. It didn't drop at all. I'm going to do another test. Okay, it did that time.
Sabine, we have 10 liters water in the suck that we have to get out.
There's 2 minutes, you said.
Yeah, but before, now you need two hands. Now you need two hands. Now you must put first out 2 minutes before you penetrate.
So basically you need to unfreeze this here, but you can also, it's just still water, you can also pump it in and then.
Yeah, that is going to be a problem.
Okay.
10 liters out of it.
We need to be like we are initially, where we're on a corner, so as we come from the side, we all have clearance. We have to be on a corner. We cannot be on a straight onto it.
Okay.
So
One thing with there's water in the bag. There's actually 10.
There are ways of measuring CO2 obviously in the water, but it's just not something we're measuring right now. Frequently as well, like if a cruise is happening where scientists are interested in X, Y, or Z, that'll come up in the planning for the cruise, and either we'll have a device that we can fit onto the ROV or use another instrument for, or we can integrate something that the scientists themselves bring. So for example, this bag that we're going to pump the water into back here, this isn't ours. The scientists brought this. We provided with them, we provided to them some of the measurements for the ROV and that sort of thing, and they would work with us to figure out how to integrate this gear. So. And the other way we can look at water, if there's a lot we want to look at, it doesn't have to be in situ. It doesn't have to be right there. We can collect a water sample like we're going to do here, and we can bring that water sample back up to the surface and run a much more intense analysis on it. There are ways to do it in situ, but it doesn't have to be done necessarily.
Yeah, it's there's a few difficulties. Cannot be straight onto a face. There's a couple reasons for it, but the main thing is the arm is in a position that blocks this arm from turning the valves on if you do that. So I need to have like more of it coming from the side so I'm clear. And then every time we've tried to get on a corner, we're not landing.
So
on
the
ramp.
scientists on board, this is one of the things, is for each cruise of Schmidt Ocean Institute, it's a different science team on board. While we do have folks on board that can operate our scientific instruments that we have and will, and for example, mapping, right? We will typically map whenever we're underway, and our marine techs on board can work with that information. But the cruises are for scientists that come on, new science parties each time. And they might have funding from a variety of sources, but Schmidt Ocean Institute itself is a nonprofit. And so we actually have a form over at our website that we'll put up when we are planning out New Year's. And scientists can say, hey, this is the project we're interested in, this is the site we'd like to do it, and we see you're going to be in the region this year, and they can apply for that. And then if selected, they basically gain access to the facilities and capabilities of the ship. What Schmidt Ocean Institute asks in return is that their findings are made public. So all of the findings from this research will be made public, and you can actually find that over at schmidtocean.org. If you look for the tab up at the top for publications, we have publications from past cruises on there. As that data is published out, we'll share that. While you're there, you can also find the cruise pages, both for this cruise, the Underworld of Hydrothermal Vents, but also past and some future cruises. We have kind of the cruises through the rest of the year on the site. I'll post the link for the current cruise. I'll post that into the chats on both Facebook and YouTube. And if you are watching live on either Facebook or YouTube, feel free to type any questions you might have in the chat. And we do try to get to those. Right now, the scientists and the pilots are quite focused on the experiments at this time, but we try to get those answers when we can. So again, this is the end of the wand that will collect water into the bag at the back of the ROV. We need to punch this wand through the mesh of this mesh box and collect the water from inside.
I'm pretty curious what it's going to look like when we move that box. This is going to be obvious. Like, I wonder if you'll even be able to see a difference in the box level from the stain or not. All that little shells. Jump on one of the arms, Carl, or are you good to hop it back and forth?
Which one you want?
Alright, so we do have 2 manipulators, and currently we're going to have 3 pilots here. We're going to have our ROV pilot Alex over at the controls of the ROV as well as the camera. And then we are going to have Zach and Chill on the manipulators themselves.
Because you're going to have to immediately turn this thing on anyway, right?
Just remember, I'm going to need clearance to get in there, so you're going to have to see.
We're okay to go for this? Go for the stand?
If you're gonna need slack, I'm gonna have to do it pretty close, just in case.
I'm sorry, there's two classes. What was that?
So if you're gonna need slack, just let me know, like, before you get close, so I can push up.
I think
But
the
this is
holes.
also on a reel, right? So if I pull it, it's
It will, but I would rather not. I'd rather push out.
Yeah, let's go ahead and push it out. Sorry, what was that, John?
No, that's a. Looks like.
Try that, or he's still gonna have to get all the way over to the switches.
Probably good. We'll just walk to the hills here for now. How far in do I want to go to the yellow tape?
Yeah,
The middle
past
ones.
the yellow tape. You want it in as far as you can, so like just before the yellow, just the gains point here. That's 3.
Oh, that's extra.
We got it.
Yeah.
All right, so before I do this, can you look around for Carl and make sure that this position will be pretty good for him to reach the switches?
It looks okay.
If it worst comes to worst,
Do a test run.
worst comes to worst, I'm pretty sure I could let this go once it's stabbed in.
Yeah.
Someone noticing the cooler that's on the front porch. That is indeed a Coleman cooler. Scientists had that, and We've actually been using that as a bio box the past couple dives. It has kept the tube worms nice and cold on the way back up, even though we're traveling through some very warm water near the surface. It keeps everything down about 7 or 8 degrees C.
There we go.
My best imitation of a mosquito. Does that make the buzzing sound?
It looks like. That shadow's gonna be helpful.
Mm-hmm. So the pilot's moving to puncture.
Yeah, pretend you're picking your nose. Really short fingernails. Yeah.
Let's try. Push it down a little bit, nice and easy.
There it is. Keep going.
Lower down.
He's just gonna go slow.
Yeah, I just gotta go real nice and easy, so don't cause any shenanigans here.
Mm.
Freeze and unfreeze real quick. Jump a little bit, that's okay. Down frozen,
All right.
and I am locked.
Nice. Good job, buddy. You still got it.
All right, so the box does have a goopy little silicone seal at the bottom, so even though it'll wobble around a bit, it's looking pretty good. And now we have to turn the bag back on.
Just good, that's good. And start.
You get back in here?
All right, now we wait.
It's a very good decision to do it from the top. Very good.
It makes it a lot
Yeah,
easier.
yeah, yeah.
Other arm to hold it down for sure.
All right, if you're just joining us, we have just punched through the wall of one of the mesh box experiments, experiment number 1, and we're now collecting water from inside the experiment into the bag that you can see on the inset there.
So this is set number 8.
Yeah, for folks watching from home, this is a remotely operated vehicle. So as difficult as it is to begin with, we are controlling it from 2.5 kilometers away. But although that may make it seem much more difficult. It's not too bad. The pilots have little to no latency. It's not perceptible, the latency, and so when they are controlling these manipulators, they have little controllers in front of them, and what they do with that little controller, it's like a miniature version of the manipulator. The manipulator itself will then move as well. So there's not too much of a challenge introduced by being remote like this. But yeah, it is controlled from a surface vessel that's 2.5 kilometers above.
Consider what they were trying to do, and we had no problem.
There's so many different directions that can go. It's got to be the same. It's the bars. It doesn't.
Oh, yeah. Yeah. Yeah, this could have been impossible to have. Make a little shape. Maybe I'll just walk with a little shape.
It's inflating, for sure.
Okay, so. When you see this, right?
Yeah.
That's our indicator. They said when you see the smile.
Yeah. Yep, so the bag is inflating. It is slow, but yeah, that bag will hold something like 55 liters of water. So nice.
No, it's just.
It's going. You can see that's what it was. That's it now. So
So yeah,
Otherwise, there's a lot of water in
stream.
the.
see right sitting in the back.
Yeah.
Is that line?
yeah.
That's a smiley face. So when the smiley face appears,
Yeah.
it's working.
All right. So if you didn't hear that, let me put up the bag full screen here. They've drawn a little face right on the fold of the bag. And so as the bag is expanding, we're getting a nice little friendly smiley face to inform us that it's working.
Did you leave any open containers to get rid of before you came on ship?
Yeah, the umbilical tether, although the ROV is down, you can see in the corner there, we're down 2,514 meters. The amount of tether that we have paid out right now is 2,572 meters, because you don't want it taut, because then if the ship is moving over swells or anything like that, it would tug at the ROV, so we don't want that. But then ultimately there's just shy of 6 kilometers of umbilical tether, and so we're sending the signal down those 6. The data and camera is getting beamed back up those 6, so everything's a 12-kilometer loop, but it's fiber optic. So it is, for all intents and purposes, yeah, the bag is getting collected to be brought back up, and exactly we are looking for evidence of faunas that might, or fauna that might be coming up in there, or anything really, bacteria, but also just a general water sample of what's in there. And we wanted to get this before we lift the box off the bottom, because once we lift the box, right, then everything can just flow out. So we're going to do this sample first, then we're going to get the box. It sounds like they're going to suction sample some of the bacteria off the box, but the box itself is going to come up as well, and we're hopeful that a lot of that bacteria will stick well enough to the mesh that it makes it to the surface. But just in case it all gets kind of washed off, sometimes the surge up near the surface turns us into a little bit of a washing machine. They'll suction sample some of that mesh bacteria. We'll get the box stowed away onto the porch of the ROV, and then we're going to go about collecting the sponges from underneath this box that we put there, as well as take some rock samples there. While we're waiting here, let's take a look and see what we've got out and about. Take a look up at the surface real quick. So, a couple dives, we've had some inclement weather, and it has messed with our internet connection to y'all, and it looks like there might be some dark clouds on the horizon there. Actually, I'll kick over to look in front of us, so might be a little bit of nasty weather coming. We'll keep an eye on that and hope that our internet remains intact. Other than that, a time allotting, depending on how the time allowing at the end of this, depending on how the collection goes, we will go around and collect some other samples from the area. And on the way back up, we will hopefully stream the Ascent again. We're enjoying bringing those to you. So stay tuned for that. During yesterday's Ascent, we encountered some fun stuff. We had a little midwater octopus that was very exciting, but up near the surface at it was just like 11 meters down or something, we had a white-tip shark, oceanic white-tip, which was very cool. So that was a good ascent. That was a very nice ascent yesterday. So thanks to everyone who tuned in for that. But we're just waiting for this bag to fill up. Yes, we also had a deep staria jellyfish on the way up as well. Cool siphonophores, little curtain siphonophore, and midwater amphipod. So you never quite know what you're going to encounter on the ascents on midwater. So we do enjoy streaming those to y'all. The way that works, right, is we, if the point of the expedition is not to look at the midwater, if scientists need their samples up on board, then we just want to come up. So the winch is always running, and it's running at a set speed. The ROV can slightly outpace that winch, or, well, it can outpace on the way up. And as I mentioned, we don't have exactly the amount of umbilical paid out that we are from the ship. So if we're, for example, right now, right, we're 2,514 meters below the surface of the ocean, but we are paid out 2,572 meters of umbilical. And so the way that we do these midwater ascent shots, if we find something cool and we want to look at it, is if we have a budget of extra umbilical, which sometimes you'll hear the pilots refer to as delta. And that delta is basically the extra umbilical. And they don't want that to get too low, but if they're at like 100 delta and we happen to find something really cool, they can sort of hover there as the winch continues to take up tether, and then once they get down, 30 delta or lower, then they have to leave and continue the ascent. So sometimes we find something kind of cool, it's neat, and we stop and we look at it and we're like, all right, we're out of delta, and we pick back up and keep moving, and then we'll find something super awesome, but we've already spent that budget for the time being, and we just got to keep going. So if you ever see us just blow past something that looked really, really cool, we probably just didn't have the delta for it at the time. But it doesn't slow down the recovery, ultimately, because the winch is still just going at the speed it's set to recover us at. All right, here's a fun question. How would any organisms get into the boxes apart from things like bacteria? Because they look pretty sealed, and as you saw, there's a real fine mesh on there that we had to punch through. So that is what this experiment is looking at. This box is placed down on top of venting fluid. So this is hydrothermally heated fluid, or hydrothermal fluid, so geothermically heated fluid that's coming up from below the ground here. That water originally entered the ground somewhere else at an area known as a recharge site, which is an intake basically into the system. So seawater is drawn down below the seafloor. Eventually it gets down into a geologically hot area, and it heats up and it gets pressed back up through the surface at a hydrothermal vent. And so scientists are interested, how do the animals that live at these vents colonize new vents, right? If you have worms, if you have a bunch of tube worms at an older vent, and then a new vent pops up kind of down the lane, how do the worms ultimately colonize that new vent with their population? And so the larval worms, they do drift through the water, and so it's possible that it's just purely chance. Maybe they have enough offspring that some of those offspring make it to the vent, and that's possible, for sure. But something we're interested in investigating with this expedition is, could those larvae be drawn into recharge areas? Could they be getting sucked down into the seabed, making their way through the seabed, making their way through the heating process, and then actually getting expelled out where that water is leaving the seafloor again? And that means that they would basically just get delivered right to a venting site. Because if they're getting expelled from the bottom like that, then wherever they're expelled, that is a venting site, right? There's hot water coming out there. So what the box is designed to do is sit over top of one of these little venting sites and prevent anything that might get transported to that location through the venting water from leaving the area. When we put the box down, we originally stained everything. So we put the box down first, we put another box on top of it, and we pumped the whole system full of a staining dye so that everything would get the stain on it. The same sort of dye you might use to stain like a microscope slide, if you're familiar with that. And the idea is that when we recover this box and we recover this water sample and we look in there, if there's any organisms that don't have that stain applied to them, those should be organisms then that arrived after the experiment was placed. And that would be very exciting. That would really be something to find. So we're pretty excited about it. And the anticipation, I think, is high to see this box recovered. So a question of what does this mean for historical activity, right? So again, this would tie into how do these vents actually become colonized. This area here had a large lava event in 2005 to 2006. So it's been about 18 years. And so there are areas where it's just bare rock, and then there's areas where there's these vents, and some of the vents are quite well colonized. And so understanding the processes by which these vents are colonized kind of gives us an idea then of the progression. There are species that, like you might be familiar with like forest progression, right? Which if there's a forest and then there's a fire and it clears it out, there are plants that are going to show up first. Kind of make that area a little bit different of an environment, and then eventually you get these different progressions of organisms that come in and take over. Same sort of thing at vents, right? There are some organisms that precede others, and so understanding how the organisms get to these vents would be just fascinating, and it tells a lot about the development of those vent communities. Yeah, that's one of the other things someone notes, that if these critters are surviving that heating, right? I mean, that many of them are already defined as extremophiles, and that would be surely evidence of that. But yeah, some of this water we've measured around here, right? The ambient temperature in the area right now, the ROV is measuring about 1.9 C, and we've seen it up to about 2 or so. But some of the vents where we've placed these boxes were up over 20 degrees C at the surface, at the seabed, right? So down inside even hotter. But then you have things like the smoker chimneys that we visited briefly yesterday, and those get much hotter, much hotter. So depending on where you go, yeah, you could get quite cooked if you were riding the flows down there. And if you're interested in learning more about this, I have pinned a comment in the chats, both on YouTube and Facebook, that links on over to the expedition page, the cruise page for this expedition. And this is the Underworld of hydrothermal vents. And so if you go over to Twitter, you can also check out the hashtag #VentUnderworld. And there's lots of information that's been posted there as well from the scientists involved with this expedition. So those are two great sources, either the hashtag #VentUnderworld on Twitter or the cruise page over at schmidtocean.org, where there's actually some blog posts that are being posted up by the scientists as well. And then on our YouTube channel, there are going to be weekly update videos, the first of which is already out, and that one goes kind of into the principles behind this experiment. So all sorts of resources out there for folks that are interested in delving a bit more deeply. You can see that that bag is quite a bit more filled now. Got our little smiley face informing us of that for sure, but otherwise just the bag. We're about 13 minutes in. Oh no, you're coming the other way around. 13 minutes remaining. What did we start at? What are we deciding to pump for?
35.
For 35 total minutes, sorry. So about 13 minutes remaining on this pumping.
And see what's inside.
Yeah, I'm curious about what it's going to look like under there. I know experiment 3, there was like a crab that was just hanging out there the whole time, so there's going to be like a little pink crab now. So And this site, experiment number 1, we cleared away the tube worms that were on this site, I want to say.
But here this is a site where we had very small tubers.
These were smaller tube worms here. Yeah, okay. Yeah. So it'll be interesting to see what we get. Give me a little different view. Just kind of toss the camera around a few places. So there you can see how we're holding that that wand out for the pumping here. And again, on the mesh itself, you can actually see, if I switch back on over to the camera a bit closer, you can see bacteria that's been growing on there. Quite a lot in some cases. It's kind of uneven across the surface. Some areas have more than other areas, and there's some areas where the plastic and the little discs cut out of the plastic, basically those are filled with plugs of bacteria now. We're looking to be on the bottom about the same time as yesterday, but again that can kind of flux here or there, but we're looking at about 6 more hours or so of bottom time. But we've ended a couple dives earlier than that. This expedition just, if we wrapped up our stuff and we had some samples that we wanted to get to the surface, we decided to do so. So we'll see what happens. But again, we're planning to stream the Ascent, assuming everything cooperates with us. Coming up on about, well, we're a little shy of it, but about 2 hours of bottom time and stream time so far. So if you've just joined us in the past hour or so, and you're kind of curious what's going on, we have returned to experiment number 1, 2.5 kilometers down on the East Pacific Rise. We're in the process of taking a sample of water from the inside of that experiment. We're filling up this bag on the back. But Sebastian, the remotely operated vehicle, owned and operated by Schmidt Ocean Institute, has kind of turned into a large mechanical mosquito of a sense, and that probe there is actually drawing water from inside the experiment. once we're done with that, we will be lifting the box at some point and seeing what we see underneath, stowing that box away on the porch, collecting samples, including rock samples, and then, time allowing, we'll move on from there.
That's
I think we got about 10 minutes left on this particular sample.
sorry.
I'm sure we'll have to. Somebody's using that, though. That's a waxy color.
Again, one of the things that I always enjoy with this sort of scientific work is the combination of custom parts and the adapting of off-the-shelf parts to meet the needed purposes here. So the boxes, for example, the mesh boxes were custom made, but the black silicone caulking at the bottom that we used for sort of sealing up some of the cracks, like that was just silicone caulking that you could get for sealing a crack at the bottom of a pool, for example. And the scientists tested several different types to figure out what might work better than the other. As someone noted earlier, we also currently have a cooler on the front porch, like you might take for a day at the beach or out to a ball game or something. It's actually proving quite useful, and as it's intended to do, it keeps things pretty cool inside. So that's been getting used for transporting critters back up to the surface, tube worms in particular. When we were collecting the tube worms up, it was keeping them about 7 or 8 degrees C on the way up, even though the water we're going through near the surface is much warmer than that. So sometimes you just find something that works and make it work.
So not only is it a smiley face,
Yeah.
it's a winky smile.
A winky smiley face, a little sly, because it knew that it was going to work. Let me put the wink. There we go. A smirk, maybe. Yeah.
Winking, maybe sort of side eye. I'm not really sure.
Coming up on the last 20 seconds or so of pumping here. So,
Turn the pump off.
oh, no, 3 minutes, not 30 seconds. That's what I get for trying to spy a phone across the room. You'll probably hear the alarm. I am making up times, Chris. That's exactly what's happening.
Got 11 seconds more.
11 more seconds of suction. All right, so we just had some pilots swap over.
So you have 3 minutes you can look at this.
What's that?
3 minutes.
3 minutes. That's pretty good.
Yeah.
Good for recreation.
So when folks aren't in mission control, yeah, I mean everyone's kind of busy different times on the ship. There's a question of what we do for recreation on board the ship. So obviously we have a lot of work depending on what's going on. So when the ROV's in the water, folks are busy with that. But then when it gets back up to the surface, a lot more science activity going on in the labs at that point, because then they have samples. But outside of that, we do have like a den. We've got a couple little lounge spaces. There's a theater on board with a big screen that folks can put up stuff on. There's screens all over the ship, as people have seen if they've seen any of the tour footage of the ship. In the mess, for example, there's some large screens there that when the ROV's in the water, we'll have the ROV footage up on that so people can sort of stay abreast of the situation. There's also a gym on board for folks that would be missing their exercise from home. Rowing machine, bench, weights, stationary bikes, treadmills. Yeah.
Indoor and outdoor.
Mm-hmm. Yeah, we have two lounges that are kind of covered outdoor lounges. Some of the exercise gear's been moved out there. There were some folks doing yoga at one point. So there's timer. Have a selection of board games on board as well. Alright, so you may have heard the timer go there. Chris is going to turn off the pump here. You can see the bag back there. Pretty full. So we actually have a couple controls over here that you can see. We of course have the two controls for this bag for pumping in, pumping the water in. But we also have a couple triggers in between there. Look more like little, I don't know, piano keys or something. And those are for triggering the Niskin bottles, which again are a way for us to collect water samples.
Yep. Just touch it, buddy.
Mm-hmm. Okay, very good. Good. So next step, we push take the needle out of the how should we call that thingy?
I'm good with needle.
Yeah.
It's like needle. You don't want to get a shot with it. No, you get blade out pretty quick. All right.
And this we don't need anymore today, so this can be secured.
And we'll just throw it in the milk crate.
Mm-hmm.
There we go. There's one more maybe we can combine that between the coaching between the two. They're gonna have a flat section. It's like lay out the gap between the two. We're not putting the milk. Well, that's just gonna be sticking out. I'm going over these. These great ones. There, you try it. Coming up. Nice and easy.
So now we'll remove the probe.
Cushion bacteria. Right.
I'll go back to.
Maybe we can take it along, or it's just.
All right. Zoom out on the set if you can. What's after this, suction sampler?
Yes. Mm-hmm. And I'm not sure what is.
There you go, sure. Yeah, so, oh yeah.
All right, so up next will be some suction sampling to get the bacteria
So it would be
on the
good
box.
if the tip is not destroyed.
It's gonna go through,
Yeah.
it might go through all of it.
Mm-hmm.
Well, I could lift it up, you could try it with a diagonal. Just have to kind of see what it does. Try and put it in an angle like this, you know what I mean?
Mhm, genau.
Probably throw an angle on that one. And that's kind of goes over the top. That'll be. When you let that one in, kind of like that.
Yeah, just throw it in, that's good. And then that handle will lay into that box.
Don't stop it going all the way through. That's what I'm thinking.
Trails. Mark. We're gonna go here, we'll just see what happens. If it's not good, we'll just.
We may have to just go over that other handle. Just sit on top of the handle, if you get underneath that one. That'd be great.
Right.
There you go.
Edge. Yeah.
Almost there. Beautiful. Just get it every day up. It's up there.
Think that's going anywhere?
Yep.
If it does, we'll deal with it.
I was kidding.
Okay.
Sorry, I'm gonna coach in.
Throws lunch.
Very good. So the next step is that we lift the mesh box up. Then we want to suction sample the inside and also a bit of the mesh.
Okay.
So I'm not sure if it's better to just put it
Turn
on the
side.
sea floor,
Yeah.
like turn it on the side and then go in, or
Yeah.
probably just keep it hold on one arm.
Thing is, it'll start spinning.
Yeah.
It's probably easier just to flip it on its side.
Yeah.
And then
But I think you have to hold it somewhere, otherwise it will.
Yeah,
Yeah.
hold it. But I think it was like this on its side. And then
And during the whole operation, it would be good because there are also, these experiments inside, the
Yeah.
sponges, so
Can
that
we move
this,
it out off the road?
we move it afterwards, but they shouldn't be moved whilst all this operation. And if you think it's difficult, like to do the suction sampling without disturbing the sponges, we could also recover them first. But I think it's fine.
If we take that off and look at it and see what, take this off and see what's inside.
Yeah,
and then
yeah,
take it off.
then we can decide. Yeah. Mhm. So just gently switch it like a... And we take a highlight video.
All right, so getting prepared to remove the box from the seafloor.
Removing box.
That's it, like, if you want to take it off there, and grab the hand and hold it in the air.
Then, yeah.
That's probably better.
Yeah.
As long as you do it away from
Yeah.
the thing.
We're bringing the gift box. See what we got inside.
It's like a Christmas gift.
Yeah, exactly.
No?
Okay, lift it up again. I want to see the silicone. That's what
So much.
Oh, okay.
All these bacteria.
Okay, Chris. So you have to do the rope and I'll have to get the handle. Okay?
Alright.
That's definitely it's not good.
Yeah, the answer to how fuzzy is it going to be? Very, very fuzzy.
Yeah. These are different types of bacteria, not the ones we see on the rocks.
An exit is not.
Something else, yeah?
Yeah.
Wow.
Like confetti in the Christmas cookie.
Yeah.
Oh. Okay.
That's crazy. Whoa.
You turn the blower like that.
Well, that's enough. All the underwater pillow fight, but
That's like
when the prunes
Ah,
are
okay,
when
yeah.
they were surfed in 91 after the eruption, there was like a lot of these smoke blowers, like that. And that's this type of material actually that wasn't checked before the substrate. So it's produced by bacteria. You see normally, and in our case in the metals. Which is an extension of the substrate.
You say you 100 meters higher. Water, okay. That somebody does it because this is it at the end.
Yeah. I write it down.
Frozen.
Exactly the time. If you look at the data, so exactly this type of shape that they form.
Okay.
Mm-hmm. Very good.
I'm going to exit there. Just held.
And probably keep it in this position that the bacteria cannot escape. So get the suction sample, like the hose first before you turn the box.
Good idea.
Yes. Must be exit.
You can see the sponges at the bottom of the experimental site there as well. Appear to have some stuff growing on them.
I think it's gonna the cover of the handle to do that.
All right.
And we will use the jar number 4.
So now grabbing the suction sampler. Mm-hmm. Thank you.
Watch out well before he gets any strength.
Hey, watch out now. And we just go up in there and maybe just go up in there.
Sorry?
Just go up inside of it.
Yes, and then I think you can turn it that you see what you're doing. Otherwise, you're totally blind. So basically what we would like to have is the material that's now inside the mesh box, like all the white stuff.
Yeah.
And I guess it's also a bit attached to the mesh, but just, you know, go in and then we try to scratch a bit the mesh from the inside. And if it's destroyed, it doesn't matter at this moment.
Okay. That's beyond real low resolution, so I'll just rotate it a tiny bit.
So probably you can just rotate like this, not fully, but like that we can see a bit inside.
There's a bunch right there.
Yes, and if you can suck in this would be great. And then probably go inside a bit more, just inside the box, that you can, you know, stick the...
You go out a little bit, Russ. Yeah.
So further in as far as you can, basically. Yes, very good.
Oh,
That's perfect.
nice.
Very good.
That's great.
I'm just gonna exit it and let you
Yeah,
suck
this
up.
is the biofilm.
Yogurt.
Now it's coming. Yeah, now it goes. Yeah, it's just a bit of time that takes it through.
Man, that's crazy. Look at
We
this.
take two of those samples.
Mm-hmm.
Well,
It's going to reach, oh, this is number
it's
4.
like cardinal
Hopefully
fruit.
some of it will be
Yeah.
Hopefully this will be retained in the...
It has a 32 micrometer mesh on, like
Yeah.
the suction
That's
sample.
okay, I'm sure there's something in it.
We just continue
Just
sucking.
correct one for us and one for you, so.
Okay, probably we can switch now to number 5, I would say. What? Number 5? What?
I mean, this is a lot. Okay,
Yeah.
let's do
Two.
5.
Mhm.
Number 5,
Mhm.
nice and easy.
Turn that down to 40.
You-
We need to push it back up. Alright.
That's better, by the way, it works great. Okay, coming on.
Mhm.
Number 5.
You can go all the way in.
What I'll do is I'll puncture again.
So this is no samples, I mean,
So the first one was sample 86, suction temperature 4, and now it's 87.
Which is the last thing I
It
have.
is still a little to do there.
And that's bacterial from inside the mesh box.
it's going to 5.
0.5 is 86, 86.
So for sample and samples, looking at 5.
If bacteria would be always that quick, I would more like to work with them because they're visible. I would like to work with invisible stuff.
Yeah.
But this is
Chris, good old,
Yeah, a lot of it is sulfur.
doesn't matter. But yeah.
I think we just, I can still see stuff coming in, so we just continue.
Yeah.
Just
Bit,
shake
yeah.
it a little bit.
You got like a plug in there, yeah.
Yeah.
Super cool.
Yeah, there's more coming into the
It's like a
container.
better version of the Akku shot that we
Logging.
It's light.
Yeah.
in the past we
Wait,
did
Phantom.
we're also trying to that wasn't always in this
Still coming through? There's
president working with Archibacter, the pump system with the sulfur. We were also trying to put it on a bend and then use it. But it wasn't a success for us.
This is the side effect of something. So it's the
It's a
side
perfect
effect.
it's a perfect Akku trap. Version 2.
Alright. Then we'll do the whole one in it now.
I can.
Okay, I think that looks pretty empty now. Probably can turn it up a little bit, that we can see in.
Can you see, with your camera?
Good call also to
So
look from
you
the
look
side.
at the bottom right of our quads up there.
I mean, a lot of it also will be in the water bag
Yeah.
Yeah, I can put this up there for a moment.
So,
because that's
No, no,
that whole thing was
Yeah.
full of it.
Yeah.
So like the blue bag will have a lot of that stuff in there.
But
still on.
also,
Yeah, I think we are good.
we're good.
We are good. So we can stop the suction,
Yeah, so,
and we can store the suction thingy away.
okay, I'm gonna index it onto 8. Right there.
Is the community really complex, or is it like few bacteria?
Yeah, so I mean, it evolves. So I think the first organism usually to colonize and form this type of stuff is Archaeopteryx. And but then over time, other organisms also come in as kind of succession.
Yeah.
But they're kind of the early colonizers. They're very opportunistic, fast growing, and they like high sulfide flow conditions and no oxygen.
Okay, cool. So the bacteria can be an indicator of the composition of what's inside of the.
Yeah, we know that this
Yeah.
is 7 days, so it's primary succession. Yeah.
Yes.
We don't know,
Okay,
of course,
yeah.
how it changed from
Okay.
the beginning.
I can bring it around if you want,
But,
yeah.
A lot of this stuff is very similar. I know when
Bring it around,
you
we touch.
papers, but it's very similar.
So they're discussing with the environmental succession. Bacteria like this are very, very early in.
Very good. Then we can put the mesh box somewhere, sit over there.
It's okay to sit on the side like that.
Yes, we could also like put it already back in, but I think we have more room to collect, so I think it's better
No,
for now
it's-
just to store it away.
Yeah, I can see that.
Yeah,
Then we store because
at the very end.
It's easier to leave it this way.
Yeah, then the basket is like, more empty and it's easier to work around.
So if you're just joining and you're curious what is going on, we have just
Okay.
lifted up the first
Now I would like
box,
to zoom-
the first mesh box, experiment number 1, and it was filled with bacteria, which when you say that, it sounds bad. And we were kind of joking, it was like a moldy Tupperware or something like that in the fridge. But in this case, very exciting. The scientists are quite excited about the amount of growth inside this box over such a short period of time. So, very exciting. But now that we've done that, we're going to be collecting some more samples from around. We got to pick up these sponges that clearly have some growth on them as well. That bluish block there, that is actually meant for and has been successfully used to recruit the Riftia tube worms. They will settle into that. So.
Yes, it's super. It's perfect. It's really nice.
But if you are joining us, welcome, and thanks for coming along and exploring with us. If you have any questions, feel free to type those in the chat, but there's a lot for us still to learn, and we're very excited to get started with the samples as they make their way to the surface.
Just that we description sealed. I don't know, sealed. Yeah, how do we call it? Silicon and basalt. Yeah.
border of frame, border of mesh looks
this looks very good, no?
Yeah, it
And
looks
that-
very
that
good.
thing looks that where you can see that it did not go out.
Yeah,
because otherwise it
you
would
would
be
see-
dispersing,
yes.
but it did not.
So you see exactly the borders of the.
Yeah, and this
so that's
is-
very good.
And actually it's in all areas.
Yeah.
See,
So you can see here.
if it's not seen, then you would
So
not
this
notice.
limpet is outside. You can see it very clear. Like it's really super good. And also here it's fine. You can see some, I don't know, somewhere there. Oops, sorry. We'll probably make another highlight shrink wrap, just to have a few pictures of the. Yeah. Ah, it's black because here the glue was on. Yeah. Okay, good.
But it's still alive, huh?
Like this one, let's zoom it.
So these sponges were placed immediately before the box went down on top. These offer a nice porous substrate for recruitment of any organisms or anything that might be floating in the water to be able to settle out into the nooks and crannies of these sponges. Likewise, that little acrylic block there made of multiple sheets with some tubes drilled in them, that's been successfully used in the past for recruiting tube worms.
Yeah, yeah, yeah. You can also see it's colored. Yeah.
Really attractive clothes that they might not be good staying off because they can follow this good carriers.
Yeah, but
For the others, we can see what's
yeah.
actual. This is some cutouts. And here you can see color. There's something here in kinky.
Yeah, here. This one. Yeah. Okay, good. So we make probably one last highlight shrink wrap. Okay. So next is that we recover the sponges
Yeah.
and either in quiver number 3 or 4.
Okay.
So we first open the quiver. And here it's also important that this is like done gently with that most of the stuff basically stays on on the sponges and on the baby trap.
See if I can bring it out a little further. That's it. I'll put your lights on again.
Bring some pictures for
Is the one question? Will this hose be in the way when we put in the chain with the sponges?
Yeah, probably. This.
Okay, yeah, that's fine.
You
Ah,
can
that's-
push it back. You can probably push it back to number 7. Just by doing that. Yeah, another one. There you go.
Very good. Ja, wenn ich dann auf so. Okay.
Safety. So which quiver you're putting it?
Number 3.
Number 3. Okay. Choose the video, say randomly, find the video, then make it work.
See ya.
Can't even be interested. Not me. See your arm again.
Yeah, so to figure out how to get close and down without dragging it across anything.
Okay. We can do it.
Yeah.
Much better than not getting that.
Yes.
Down.
Yes.
Would it be easier with the other arm?
It's the distance is the problem. Yes. Yeah, also concussion.
Hoppala. Are you fine?
Yes, okay. Man down.
Pilots are just working to get this in there nicely.
Down.
Get a shot.
Cover at it, the other arm, and then
See what you do.
Try to move it gentle, gentle, gentle.
So again, these sponges just provide another substrate for organisms to settle out of the water into. So there are the sponges. There's the rope at the top. Obviously, it looks like some stuff's grown on that as well. The chains are primarily just weight. There's a
Can I grab the chains, or do we?
Sorry?
Is it okay if I grab the chain, or is it?
It's okay to grab the chain, but I think it's, I don't know, can we still then get it into the quiver?
We try. We'll figure it out.
There's
I've
a
got
temperature logger on there as well. So that would have logged the temperature for the amount of time, or for the duration of the experiment. And then finally at the bottom, there's a tube worm recruitment device down there as well.
So we should just
So the tube worm recruitment device I was talking about is at the bottom there. It's that little block. So that's sort of stacked layers, and then there are little pinholes drilled into each layer across the wide plane. And this has been used successfully in the past to recruit larval Riftia tube worms. So there's a camera angle showing that tube worm device.
Put it right where the one after it is, you know what I'm saying? The one I'm trying to go in. A little bit more in.
You're right next to the...
Alright.
If you look on this one, you've still got about 30 degrees on your floor.
It's, when you see it and you see it, it's just, you just, you bring it back a little bit more. Yeah, good now. Trying to get that in there, like the height of it,
the chain, drop that, and
the height of it. You just couldn't get it to be like that.
over the starboard.
Yeah, and then you can't reach the condition.
Can it go in the
Robert?
No.
Don't go over the tail.
I mean, I could grab it again with,
Grab it lower with the starboard one.
yeah, grab it lower,
No.
but like on the chain, flop over a little bit. But I can get it out.
I think we might have to. I can do it with starboard if you want. You want to get
Yeah, if you want.
teamwork makes the dream work. Right?
So, alright, bring that back out. You wanna try that?
Sorry?
We want to grab like lower down on it.
That way you can kind
No.
of get it in.
The problem is if like the sponge is then like falling down, then
Yeah.
basically all the animals that now like
I can do it real slow, but I just don't think it's gonna reach with this.
Well, no, it won't, that's.
Yeah.
But the other possibility is to just open the blue cooler and put it
But
there.
Monica, but sorry, wait, I'm not sure if this is good because we will get a lot, this will be open a lot, the box, and it will a lot probably also be washed out.
Before.
Where do you want to grab it?
So I'm not gonna do it, I'm just
I
gonna
would
show
say
you.
you have to go
Okay.
below that first sponge.
Yeah, like,
To
well,
the
I
eyes.
wouldn't go, I would go above the first sponge, like right below my jaw. That's that much difference, because I had it at the very top of the red.
Yeah, so I mean, that's like,
I think I would grab it right here because that is the max. Well, I'm not gonna touch the sponge. And then just try it, and if that doesn't work, then we'll assess again.
Is
Take
it
this-
possible that you move both arms, you know, that it's not like that the sponges are not falling?
Yeah, or he could just set it
Yeah.
down real gentle like too, because it's
No, but-
okay if it's doubled, right?
But if you
No, it
like,
doesn't fit if it's double.
no, it doesn't fit. But if you take it now here, at the same time, if you keep this and not let it fall, because if you take it here and let this loose, it whop, it falls this and it falls down.
see if you go in here, that sponge is held up by that rubber, okay? All that distance is gone, and then you should be able to drop in the box. That's what I'm thinking.
I mean, all we can do is try it, you know?
I'll say,
It seems for sure that this port
I
arm's not
think,
gonna cut it,
yeah,
so.
Yeah.
I think if you're gonna go above that top sponge, that would give you enough as well, 'cause
Okay.
that's this far.
Okay, but then go like just below, like at the first sponge,
I was gonna say, just go,
like
You say
Just
in
go.
that
I
right
think we're
here?
right in there.
like
I'll
right.
say go
Yeah.
right there.
I don't wanna cut that tire up though, because I got the tires, remember?
But I think here is good.
Here is good.
Grab with that.
So just leave it right in here. Can you look up a little bit with the sit tubes? Second opinion.
That's it. Should be good to,
It's gonna be like under.
No, but fine, just having a bit of
that should bite really nice. Should drop.
For me, this is not relevant on this.
Alright, when I let go, I'm gonna push it a little bit towards you. Alright.
Okay, if you wanna come off it real, just come off real slow so let them just come down and bash you, you know?
But that should be fine.
Yeah. keep it low.
Yeah, very good.
Alright. Come back on that route. Sure, yeah.
Isn't there a real slow though? 'Cause you're losing
Yeah,
it.
very slow. Yeah.
Isn't there a temperature monitor in this?
Yes.
Yes.
Yeah.
And that's inside of that plastic?
It's the
Yes.
red, it's the red bag.
It's gonna be kind of a test run, see how close I can get it, I might actually have.
And slow, move slow.
Oh, yeah, that's
Yeah.
slow.
Not really. Give me that.
Can that reel come out anymore?
Mouse.
It's maxed out. Can I roll it up on my wrist a little bit? Like turn my wrist like this?
Yes,
Yeah,
slowly.
this is okay.
Just
Yeah.
to make it a little more high, you see what I mean?
Mm-hmm.
Yeah, this is okay. If I press it from in, then it works.
So then, right? See the shadows there, it's like.
We should be able to get it now.
Clear, almost clear. There you go.
Do this real slow.
Come back towards us a little bit.
Mm-hmm.
What's that thing there that looks like a bag of marbles?
That's temperature.
Temperature sensor. You just see in the center, push
Way down.
way and
Real.
I think what we should do for the next two is grab a couple of these from the barn, I'll just bolt them in the basket.
Alright, I got freeze real quick. Adjust.
If we put it on one.
All right, so you can see they're making progress getting the sponges stowed away, the length of it, and we can't use the front coral quivers there. It's making it a little difficult. The pilots figured out an approach.
Alright, at some point here pretty soon I'm gonna have to drop it, that's okay?
Yeah.
Okay. If it doesn't all go in, I'll just boop it in?
Yep.
Alright, so I'm gonna release now. Gentle as I can.
Perfect.
Well done, yeah.
Thank you.
And now we want the lid on there, right?
Mm-hmm.
Got our camera menu, there we go. I don't think the soft jaws are the yellow any different from the red. They're just different printed.
Different material.
Yeah,
Different quality material.
different quality. Okay. Wow.
No line material.
I mean, the other one would have held up to yesterday's activities.
I think you said twice as good. This is twice as good as the rest. Very scientific measurement.
Uh-huh.
Let's roll. This rope is getting messy.
Yes, it's a good taper.
Does this rope have to go in there, right?
Yeah.
Find it all dry.
Floats.
Oh, you're.
No, I can't.
No.
Maybe I'll. You know, for the next one, I wonder if we could put it in with the rope first. So the rope's on the bottom.
Grab onto the plastic.
No, on the plastic you can, on the
Everything
chain.
Yeah,
squeeze
chain,
out,
yeah.
you just jam it in there.
No, this is not going to be sealed.
No, this is not working.
Okay. So we need
Well, you
rope.
can cut it open.
Can we cut the rope?
You can cut it, tie it up.
That's okay?
The tie wrap.
Yeah, yeah.
Yeah, okay. Perfect. That'll be no problem then.
And it may actually complicate motion up.
What's that, Chris?
When I would put them in that one, I would put it.
What's that?
Yeah. Trying to see, set it up. And have it up the other side on the rubber. There you go. Down.
Open up a little bit. If I can turn and see my blade.
No other options. Left that a little bit.
I've got the rope, so it's the rope lock.
Yeah, say you cut it pretty much for
So I might have to come up with an angle.
try this. Get between them.
Set.
The Olympics here, man. See what they said?
Check out your outside the box. On your start.
I'm okay, or I need to do something?
Easy.
I can just snap it.
Is getting a little bacteria. There you go.
that's the new patch of the oceanish.
Yeah, it's not too close.
There you go. Oh, middle. You're in the middle of
Yeah, it's not cotton.
you're in the middle of the other plastic.
That's what we want.
Well, no, you're not. I don't think you're.
No, it's
Plastic at all.
between the blade and the rubber. See?
Yeah, I think that the blade is
It's not
in between. Yeah, it's between them.
And it's a better cut near the tips of the jaw.
That's definitely.
100%.
Try to go alligator death roll.
Just remember to get bacteria on them.
You may want to jam it again from the top leg.
Freeze again.
So, the top leg. Top leg.
Freeze.
Just try not to get the sponge out. Oh, this tyrant.
If there's some really strong blue rope, use that next time, it'll still seal
That's possible.
with the cap.
Put both just in there.
Excuse me? I
I'm
don't.
just saying, when rigging, you can use their blue rope, you're using it again. That way it seals in the cap, it doesn't matter about try it.
I'll try this now. That's the part this is all about. It's the pressure will come through.
Yeah.
It'll just cut through one side against the other side.
To give it more of a foundation.
It's sharper on the top.
Sharper at the top.
Yeah, when you know, with the real jaws, look at it.
Johnson, we were both of them kept together. Yeah.
Yeah, as long as I hope,
We might try that if we
Can you try just to squeeze it in and probably it's holding by one of the sponges? That this would already help?
right. Pushing it down and I see this one hold.
Probably the sponge would hold it.
No, the rope floats.
Yeah, but if it's like squeezed a bit?
That's what I was trying to do at first.
Well,
Yeah.
when we would put it down, I would squeeze it on top of the tie wrap and then like tie it on there. It wasn't water.
Okay.
Alright, I think we have to try this, stick the hand in there, but kind of thing. Alright, so the rope is usually
No.
part of it that we don't have our sharp, we don't have our sharp jaw right here right now, so it's a little harder to cut stuff than normal, you know? Yeah, for sure. Alright, see if I can do here. I'm not trying to be rude, but I don't think this tire my friend. Might even be a little cross with it.
It's axle across. You know that.
Yeah.
Is that like a camera?
It's not actually a car. Yeah.
Just got it on rubber, but maybe I can do it with that. I mean, how firm that rubber is.
Right. Maybe. Maybe.
No, it's exactly enough to.
No. Shit.
Yeah, get the
If
jaw
you
in
can
there.
get it all in there, yeah, you'd be alright. There you go. There you go.
Oh,
Alright.
hey.
More now.
Little rocket surgery here.
That was impressive.
I actually lost 3
Yeah.
Soft, 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 soft. There's a sponge inside the river, please.
Yeah, I don't know if that gets too much water. I want you to go all the way to the edge.
Yes.
Okay.
Okay.
I just remember this sponge.
Nice to the sponge. I don't want to mess up sponge well.
Yeah, I don't think it needs any water.
Ah, nice. Good.
Boy, and we'll just have to see the compass.
Phew.
Don't forget to rotate around seven.
Alright, can you walk it over to me?
Let me turn to try and not let it float away here. Okay, it's underneath this. No, you got a good piece underneath the joint. That's good. They can just send the FRC to get it, I'm sure. I grabbed that. Oh, I got it.
Yeah.
But what do I do with it now, though?
It will float away. Yeah, it's already gone.
We got a plastic thing. Can I put it in the milk crate?
Yeah, that looks like that.
Yeah. Can we please close the coral quiver number 3?
Yeah, hold on, we just grab this then and we can do that. I'll have.
Froze. See, you got it? Okay, open it.
Back to the edge. Alright, if you want to go, I'll hold this and then.
Okay, the let's down now. Zoom in.
Very good. Thank you.
Thank you. A little intense.
You think it's easier to use like the front ones, 1 and 2, or does it make any
It would be-
difference?
1 and 2 would have been a lot
Yeah.
easier. But
Because these are like at the moment not like one is leaking, the other one is like destroyed, so it would be good to replace them for the next time.
Yeah.
Yeah.
I would suggest for the next one, we cut that kind of like the rope first and then.
Yeah, but then
if
you
we can.
move it a lot, you know. The more you move the sponges, the more chances that you usually the animals like they hold fast.
Throw in the good blades. Yeah. I'll throw in a brand new set of blades.
so
Mhm,
you-
yeah,
sharp,
yeah,
sharp,
mhm,
sharp,
yeah,
sharp.
mhm,
1
yeah,
and 2 would be a lot easier.
mhm, yeah, mhm,
Probably,
Yeah, okay.
the problem you're gonna have is that like,
Yeah.
you had up for that.
yeah,
I mean, even if we swap 7 and 8 out with the forage.
Well, I have some
Have you got a couple of
in there,
spinners?
I'll test them.
Yeah.
Okay, good. So now we take a picture, like we go to the spot and make a frame grab when.
Let's go.
Sorry.
Yes. You're in charge.
Now I'm allowed to touch again.
Yes, you're right, that's it.
Very good.
Yeah, it's very easy to see.
Yeah. Okay, so we make a highlight frame grab.
Yep.
Okay, and next is that we take a water sample. This is connected to the temperature probe,
Oh, yeah.
and it's operated from your side. It's like with this
XVX.
C-link. And let's first check where we take it.
Right.
So we want basically to. And Monica, please look with me. I suggest that we try to stick it in
Shim
here
right yeah.
to the water sample. the water bag.
Yeah, as you can see here, there's this
Yeah,
crack here.
this here,
You
yeah.
just go here.
Good. Then we agree. That's good.
You see that?
No.
You see what that was? Yeah. basically.
So like here and there's a crack.
Black hole.
I mean, you can,
Yeah, that's the one.
wenn es heiß wird, dann kann man auch stoppen und nehmen. Also du siehst ja die Temperatur. Das ist gleichzeitig Temperaturmessung. Also
Okay. This will be this will be sample number 89.
The water
sample is not
Right.
Wheels coming out.
Und kann magst du essen gehen oder soll ich essen gehen?
Sorry?
Dass einer von uns anfängt?
This is going to section sample
Yes.
this. It's the same, right? Oh, the, okay. That's good.
yeah, you can go, huh? I continue.
Michael, it's 15 minutes. It has to come in.
15.
15. Okay, good. Yeah.
You can do afterwards not the temperature so much because it will be taken together. You know, we can, since you open this now because it will take a
Ja, passt. Geh mir aus.
15 Minuten.
You're taking this section sample or?
No,
It's,
it's a special new pump with a little bag. We call it a little bag. Chemistry.
with water then. I will put the sample with water. Because it was such a sample.
Yeah, no,
it's
nothing of this is other.
No.
Right, is water.
Location around water, use that from surrounding the sample.
Yeah, new bag for chemistry. Also water.
Yeah. The
In
water is
little bag water, in little bag subsurface water. Water, surface water in little bag. The silicone crack.
oh, sure.
Crack. Chemistry should like
Okay, I'm gonna follow you.
for chemistry.
Okay.
Okay. We just check for hot water, basically.
Okay.
The temp is
Maybe a time for you. Very good. Go for a bang.
Whoops. So, right,
I can track that.
just be probe a little. What is the temp probe? Is the,
7, 8,
where is the
9,
temperature?
it's up top there. 9.3,
Where?
10.
Up there.
HD probe there.
13.
15,
Is it-
16, 17.
Is it keeping the highest then? Or do you have a button for this?
John.
Ah, here, thank you.
19.6.
Okay. Let's just wait a little bit.
little high.
Where's the temperature measured? On this point or on this
On the
point?
little, so the one
On the
that's-
little, on the
no,
smaller
no,
one?
on the longer one.
On the longer one?
Yeah.
Okay. Alright, stay. Maybe it goes up here. Here, it was, okay, yeah, this is good stay, just it goes up now here. Wait. Because it was already,
Yeah, maybe a little bit closer.
yeah,
We're kind of sitting outside the vent a bit.
right, more in this back direction.
Yeah. Might be.
Let's see,
It's going up.
9,
It's going up.
11, it should go to 20, basically we had 20 before.
It's moving.
It's looking good there.
Is this,
Something keeps on.
this is 15,
Under 17.
but now it goes up. If you can hold here, that would be good. Here it was already 19 something earlier, but it was not recorded. Now it goes.
Is it?
You're moving. It's very difficult to see where you are. Okay.
So I actually, that's a good spot.
Mm-hmm.
Rolling.
But hold.
Yeah.
Hold here.
That's good.
What's great? I think that the orbital, yeah.
Huh?
Like the
The
whole
whole
stuff.
sub's moving. It keeps creeping to the floor whenever it's frozen.
Well,
Oh.
it's solid. I mean, I've got as many times as I can on it.
It's not solid. I mean, it's not your fault. I'm saying, you know what I mean?
With the arm.
So what happens then when we sample the water for 15 minutes? Is it then also
What we can do
moving?
is if we get another arm down and stored, at least down, maybe that'll help with the balance.
Ah.
Just
Ah.
2 seconds there, I'll get this weight out.
Okay.
from the-
Yeah.
Maybe it'll help stabilize that a wee bit.
Mm-hmm.
Okay, that's I'm back in its home. How's that feeling? Yeah, one more down that. That's good. That's good.
Mm-hmm.
No, we're down.
That's it. It's fixed.
Yeah.
We're 51% done.
Yeah, we sample it here. We sample here. I write down maximum 19, and here where we sample is like 16. And
You happy? Can we
it's
go?
got, yeah, I switch on
Yeah,
the
okay.
15 minutes now.
Okay. Maybe the 2.
Yeah,
Or
just-
it's sitting there.
That's steady paddles.
You can see us with threads.
Okay.
Yeah. So it was like, it was there, and then we just kept doing this.
See it.
You can make an temp. Actually,
See it's something at all.
this sample is, you can log in this sample. What do you have observation something? Like I wrote down maximum 19, but we sample at 16, maximum 0.3. If it shifts up a little bit up and down.
Okay, sample at max 16 point.
Yeah, but not, yeah, 16.3 it was. but I measured this the highest A19, but in,
Okay, this is 89.
this is 89 and you log it in,
Yep.
and then make a temp observation or something and just write down basically the same thing with the temperature. Maximum 19, sample at 16.3 maximum.
So
Okay.
we speculate that the water going in is warmer than the water that's being measured.
Possibly.
The water
That
is-
could be,
It was
yeah.
at 19, and then when we pushed the sub down, it went down a little bit and dropped to 15. So I think that just above, where the smaller tube with the shorter
It's
tube
okay.
is,
Yeah,
it's like probably closer to 20 degrees.
yeah, it's okay. I write it down and I mean,
Few degrees.
But
so we have 15 minutes for this, and the water sample is the number
90, no? Or no?
89, no?
89,
89,
yes.
water chemistry. Okay. I have this switch on. Yes. And okay.
let's go back on. Okay.
Oh, yeah, okay.
The sponge and river free, the sponge free. Okay,
Yeah.
thank you.
After a long fight, we got
88, that will be the best because 88, 8 is my lucky number.
it. Is it?
Ah, yeah.
Oh, it's lottery time.
Ah, yeah. I'm very superstitious.
Really?
Yeah.
I didn't know this.
Yeah, I need a opposite to science. Like 4-leaf clovers, numbers, I don't know.
No umbrellas inside, no shoes on the table.
I have here this 4-leaf clover and the backside here. I mean, you don't see it, it's inside because I found one before I left.
Alright,
I was looking for
we'll
it.
keep that.
It's in here, so
Yes, don't lose the phone.
then my son gave me this. He bought it extra for me. Also in gold, the 4-leaf.
Oh, nice.
For
Nice,
luck.
Fair enough.
All kinds of luck. You can never have enough.
No, you can't.
See?
Especially when you're doing the lottery.
Okay. I'm gonna write this. Sponge. DP trap. Temp log. What else do we have? 89. the temperature still recorded or?
Yes, it is. It's just that it's not on the screen.
It's not
I've
on the
still got it up
screen.
here, though, if you want to see it.
Yeah, I would like to
Jeans
see
jumping
it,
around.
just to see a little bit. 14, yeah. This is the number 89. Okay. HT probe, high temperature probe. Okay.
Okay. Who wants to eat first? Chris is good.
Doesn't get breakfast, so he gets hungry this time of day.
Fill your boots.
Want to go right, Russ?
Not really. Well, it's better than most settings, but you know.
All right. Fill your boots.
Yeah, it is 16, 17.
Yeah, it bounces around.
It bounces around between, I don't know, 13 and something like this.
Yeah.
14. It's okay. It's good. Mm, 19.7. Okay, then we are good. I write down
The temperature.
19. Also,
Ah, yes. Good humidity.
Like a window screen then.
yeah.
What is your temperature there on the gray rock?
We have 5 more minutes for this water sample for the chemistry, and then
Good crap, but
Is this stained?
Looks like,
what?
there is in the middle of this window, there's a stain. This is the rock or?
This is a stained animal, but it's embedded now in the mucus, also in this biofilm.
Yeah.
So let me see if I find this.
Right. So just take the little bag. between that side, the complete side, like or something like that. And yeah, try out and form out so you can put somewhere. Alright.
Could you look for a special, can you look for this here in this type for this highlight frame wrap? 2 more minutes.
Okay.
And then we have the rock.
It's
Yeah, it is A. Also, yeah, it has several cracks.
Is this that silicone here stuff?
This is the main crack here that has to go through here. Then we glued also. I just try to find my bearing because we're sitting a little bit, but I'm just trying to print out the picture from before, so we look at the crack. But I think the main crack we removed. The main crack goes here to here, and I'm sure we removed here.
And the piece of rock needs to be what was inside the box.
Yeah, but it does not have to. Also, we have to start from somewhere breaking and just try to
Hey, Monica,
grab.
I know it's too late now. I know it's too late now. But I have an idea if y'all ever do this again. What if we went and grabbed a rock that was loose
Yeah.
and put it in the area and
Yep.
then did your experiment, and that way you know as soon as you pull it out, you got it right there, you can
I was thinking about this before already, but you cannot disturb the flow because then it changes. This is the thing. Also, there might be something now on the sponges and the baby trap. They are on top of it. So we have several things. We have the water, but I think we can. Let me see. Yeah, we can switch off the pump.
And it's off.
Can you lock this then also? Pump off.
Yes. Yeah, you
And
That's it, right? Is that-
yeah, you can.
yeah, the pump.
We
What is
can-
the, is that?
No, you need that one. That's okay. That's really not too
It
serious.
is really, okay, I didn't know if we had that.
So you just shut it off, right?
Yeah,
I did.
and then we can store this.
Sure.
And
This is the one.
the frame is on it now.
Yeah.
Just remember that, oh, we're not gonna holster it? Yeah. we do. Do you remember that,
This yes, print is good enough because
Portfolio?
the next one is better than
Super
maybe the
good.
next one or so. But
Yeah.
I will look at this here. Oh, this is a gone.
It's on the, see that?
Okay.
track the sign camera, so I can't see on this.
In?
Yeah, the camera showing anything or anything.
I can push out more. Start to lose sight of it, but it's better for your arm. This is there. I'm just gonna walk in with you a little bit. Does that help? I almost feel like you need to see it a
This
little
we have
more.
already. Yeah. Just
Okay. That'll do, right? Yep. You're going there.
He's up a little bit. Grip, so not slide over the other side.
Sure.
Leave him grabbing it now.
There it goes.
Okay.
I think we scan the area really good first, and I take a overview and we print it, and then I try to mark where the rocks are. So
Well, yeah, you can see.
okay,
See good.
can you take a frame?
Yeah.
Frame grab here and then print this.
Can print?
Mm-hmm.
Yeah.
Did you take?
Yeah, I took.
Okay.
Both bars. We have them both.
I'm gonna look.
You see that spot right? On the left, there's a pretty decent gap right here to start. So you gotta just
Stick it in, I'll just start doing this.
Kind of clear. It goes here, and there's a little
Sure.
rock. Should be like a corner here.
You see that loose spot right there? That might be a good ledge to get under.
Here, back from here. I don't care about those were outside. I know this you can move away a little bit or so
Sure.
because it's just a blob. We wanted you to seal something, so maybe this when you take this, then there's really a crack, and you
Yeah.
go like this.
Exactly, yep. And the hope would be to get this piece here,
Just
yeah.
be started very soon. Yeah, but here the thing is just this here was the place exactly that should be the least touched, but there's also cracks here. We sealed here also here.
Yeah,
This is a crack also.
sure. So see this whole plate right here?
Yeah,
I think if we start here and we can get this loose, we can start working it this direction.
Yeah.
But I think that's the starting point, the best, because right
Okay.
here we have nothing really to
we have
get
to
into.
get in. Yeah, and this was a crack because we sealed it,
Yeah,
and
but eventually this
Also-
whole thing will tip. I don't know how we would
Yeah.
break that. That seems almost impossible, but we'll try.
There might be another one here too, on that bottom right corner like that.
yeah.
If you're looking for cracks.
But at least this plate.
There's also basically here.
Yeah.
You see this here. I don't know. That goes I think like here or joins here. You know.
Sure.
I don't know really with this here because the crack goes here, so the ending this plate
Yeah.
must be here.
I think we'll learn more once we're starting to pry the rocks up. We'll see what's
Yeah.
coming loose.
Yeah.
Okay. Ready to go get the bars? You ready to go grab the bars? Or do you need to look more?
No, I don't need to look more. I get a picture, but I mean, I think you saw the same as I saw.
So the bars are both somewhere else?
Yeah, they're.
I mean, I think it. I don't know if you need the big one. Yeah,
We're gonna grab both. Just
both.
so we
Yeah,
have
the
them.
little one is inside the. We recovered it, I think.
Yeah.
Oh, so we got the one now?
The little one is inside and the big one we can grab first, so we just have both of them. Ich hab da, du kriegst noch was zum Ausdrucken, das ist das, aber es ist noch anders, das war das alte, aber man sieht es eigentlich gut. Du hast praktisch, im Prinzip gibt es da so eine Plate und da oben Haararme und die gehen auf vorher, aber sie holen es jetzt nicht. Also sie holen sie was schon mal und dann.
All right, if you're just joining us, this is live footage from ROV Sebastian, remotely operated vehicle that's been deployed at the bottom of the Pacific Ocean by Schmidt Ocean Institute. This audio is coming to you from Mission Control Room on research vessel Falcor 2. Quick glance at the Pacific Ocean there for you. We are on the East Pacific Rise. It's part of the Underworld of Hydrothermal Vents expedition. And the scientists are tackling some interesting questions about whether or not organisms can travel through the hydrothermal vents to repopulate areas. So what we're doing today is we're checking out some of the experiments that were previously deployed. You see some of those white boxes in the background. And we have uncovered one of those mesh boxes, and they're going to do some sampling in what remains underneath there. Right now the team is maneuvering the sub to grab a couple pry bars. And currently in the control room we have Dr. Monica Bright, the principal investigator of this expedition. We got Chill piloting, Zach is running the manipulators, and I'm Tyler, one of our marine techs for this expedition, on the other microphone. And it looks like Dr. Sabina Gollner has just swapped in with Dr. Monica Bright. And feel free to send questions in the chat. If we have time and space, we'll do our best to answer those.
Set it down, or? Okay. Let me grab that up more.
Just getting it out of the way so you'll land on it. Just grab that one as soon as we land. What if I drop this and do some damage?
Das Wichtige geht, glaube ich, da durch. Genau.
Und deswegen.
Und da sind auch die kleinen Typs gesessen.
Da
Der-
waren sie, ja.
Genau.
Weil das ist jetzt,
Das ist zugewachsen. Ja.
genau, aber wie sie es ist.
Aber, okay. Und vorher den Cooler öffnen, gell? Monika? Ups. Und vorher den Cooler öffnen, oder? Müssen wir vorher den Cooler öffnen? Der ist vorher, gell? Ja. Genau.
Holen Sie das einmal.
Genau, das haben wir schon.
Sie holen das, dann machen wir den auf.
Den haben wir schon. Ja.
Ah, okay. Ich habe das da eingestellt, aber Sie haben das am besten. Ich bin ruhig.
Almost dropped it. Come straight on this guy probably.
So Sabina,
Actually, Zach.
while the team is getting set,
that
Sorry?
crap
hold on one second, sorry.
Might have you kind of in this segment. Should we move that box before we do this? Because you'll have this in your jaw. What I'm getting at is that mesh box going to be a little bit in our way.
Move it. Is it a problem to move the box?
No.
Problem with that?
So while they're getting set up, but when I bumped into you earlier, Sabina, you said you're pretty excited today. Do you mind telling us what's exciting about this for you today while they're getting ready here?
Yeah, there are again a lot of steps that need to go right that we actually can get what is in the box. So the first critical thing was to basically punch this mega needle into the mesh box. without actually moving it. So we had quite some discussions in the beginning, like what would be the best way to do that, because when you freeze the arm, it usually goes up a bit. So this would move the box. So we decided in the end to do it like from the top, and this was an excellent decision because we got like really a nice sample from the water. So that's like super good. And then when we moved the box up, we could see like all this incredible amount of bacterial mats that would like escape. But we also like had the box in the right position, also made a good move to like keep it in this position, because also then we could suck out a lot of the bacteria that were still trapped in the box. So there were a couple of things that really went well already. And now it's the next critical step is to kind of, to get the rocks out. So many different steps. Until now we are going really good. I'm really happy about that.
Excellent. And it was like a blizzard of bacteria
Yes.
when you guys pulled
Yeah,
that box
yeah.
up.
That's that ledge.
Maybe I could even go a little more this way, you know, like.
So that's where we're going to
Yeah.
try to pry under right there. Yeah, that's better. I like that. Sorry.
Oh,
So it's been a day of successful maneuvers down here on the seafloor.
But don't jinx it.
yeah, but it doesn't.
No
This box,
jinxing.
it doesn't matter. It's fine. All good.
You know what that box size now? It's not its job.
And this is the first time these experiments have been deployed on the seafloor, so we're celebrating the victories on this.
And what is also very nice that we actually can see when the box was lifted that part of the silicone is still sticking to it. And we can also see it sealed very good because you can basically see the imprint of the box because of all the bacterial growth. So it worked really good. I'm really happy about that.
Very good.
Yes, the evidence of that silicone on the seafloor still is showing how well it did throughout the whole experiment.
All right, Minecraft. Let's go. There it goes.
Just set that right there and we'll get it later.
All right, so we were talking about doing over here, right? That's what we're thinking.
If you get that chunk lifted up, I might reach out with the other arm, grab the big bar and try to help you maybe.
Can you brighten it up a little bit?
Sure.
This might be kind of a slow process of being on.
Yes. Yeah.
We want to take a good sample for you and also not kill our arms.
Mhm.
So we're just going to go nice and easy.
Yep.
Not do our lead around too much or anything.
I can give you more downs.
That's a really nice starting point there. I wonder if I let go and grab it sideways so that I can just rotate my wrist instead of pushing down, and that would knock you around so much. You know, what do you think of that? Or should we just try it first?
You might be able to jam it right there and get your regrab.
That's what I was
Yeah,
gonna
go
try.
for it.
All right, I'm gonna let go. Oh well, I can still get the side grab.
Oh, sorry.
All right.
Far. Do you need help you or you got to grab you up?
I think so. I might have to rotate it a little bit, that's all. But yeah, let me get right here. All right, there we go. There we go. He's sneaking in. Just kind of exploratory, poking. Like a shark looking for dinner. What?
I didn't look for that first one. Around the code.
How big does this rock sample need to be?
Yeah, ideally, like we want to have like everything that comes from this crack. Also here has been a crack, but I think we just have to see how it goes, to be honest.
Am I whacking you around too much, Carl? Or
No, I'm zoomed in, so it's not as bad as it looks.
I think I saw it move a little there.
I saw that as well. So what happens if you pull the arm back?
I see stuff moving. See it?
Yeah.
Mm-hmm.
The whole crack's moving.
Mm-hmm.
Like, so stuff flying out.
All right, kind of.
See this?
Mm-hmm. Like
This
here, that
triangle?
thing is now moving.
Yeah, we might get that piece.
Let it clear a minute.
Did you see that, Zach?
What?
So there's a triangle right here, and that's what you have loose.
That's right, Liz? Okay.
So you might be able to get that guy.
So I'm going to keep working it down to get deeper in, and then after I get deep enough, I'm going to try and flip it to the left and
Mhm.
pick the rock up
Yeah.
to the right.
Mhm.
If you guys agree with that, and jam
Yes.
it under so it can't fall back in. Where's this going to go if we get it loose in the cooler?
Into the cooler.
Okay. Can you look like down a little bit and yeah, just kind of center up that.
Didn't know if you wanted to see your jaw at all.
No,
or-
no, I do, but just like a little more of the crowbar, you know?
Okay.
Yeah, something like that. Yeah. I'm about to drop this thing, but that's all right. I'll re-grab it if I do. I'm gonna freeze. I'm gonna re-grab. So it might fall, that's okay if it falls. It's okay. Get back in there.
That's a good grab.
That should be beefy.
Unless you want me to spin it. So you got
That kind of thing, you know? Maybe. I can definitely get you some small pieces. I don't know if you want that.
Very
but
good. Yeah.
We got some small
That's-
pieces
mhm,
already.
yeah.
Liz. There's
There's
another one.
another one.
Kind of push that
Do we
up
want
there.
to take the small pieces first, that they're not further destroyed, whilst trying to get more of the rock?
It would
What do you
also make
think?
it easier for me if we
Yeah.
do.
So probably can pick up these three pieces and already put them in the cooler.
Sure.
Okay. Wonder. Because those are best. You're gonna try it with that? Because
Unless you
I'm
want
worried
to.
that it's gonna crush.
Yeah, you think the rubber would be better? Go for it. But the only thing is, I don't know that I can hold the cooler up there
That's
for you.
that's my concern too. But you might be able to just like tip it up, and then I'll be able to get underneath it.
Let's just see what happens.
Okay.
What could possibly go wrong, Carl?
Remember what happened last time we did that?
No. Short-term memory loss.
You blacked it out. Oh, I got you.
All right. Press that cooler up and I'll chuck her in.
Is that a new sample?
This is sample number 90.
What is going to be the location?
Sorry?
The location?
This is experiment 1
But outdoor?
in the cooler.
So every drop will belong to the sample.
Sorry?
Every drop will
Yes.
be
Pilot's dropping that small piece of rock in the big blue cooler, which has performed admirably well thus far.
It's totally not my fault. It's just luck.
You're gonna be lucky if you get it. I also really.
I have to flip that guy out of there with the pry bar. I'll try one more.
There's one in the back that you can take, I think.
Got yeah, let me flip them out of there. All right, where's my instrument? There they are.
We've seen the manipulator use being likened to deep sea claw game. And this is about as close as it gets to that analogy.
If I get it out of here, if you can even get up the rubber ones, then cookie rock. So that one's out.
Right there.
Won't get. All right, and is there any more to do that to in there? Or is that pretty much it?
I think that's it.
That's it? All right, let's just go back down. Okay.
What if you set it on the gelatin so that you have a nice deep grab for your next attempt to pick it up?
But probably you want to pick the rocks first and put it in the cooler.
Oh, that is
Oh, God.
Carl throwing an additional layer of challenge in there for Zach.
How about now, sucker?
Zach is undeterred.
He deserved it.
It's true. I don't know you're talking about, but it's true. Oops.
And a slam dunk of the rock.
Man. Other one is right under that.
It is.
And there's Zach giving himself a challenge.
Or if you just want to continue on with your breaking. This
Things weird. Oh. How would you
Oh.
like
Oh.
that
Oh.
rock sample?
That's the
That's-
silicone.
That's only silicone?
Yes.
I'll just move that to the side then. I'm guessing you don't want the squishy rock sample?
This will now float in your. Okay, that's good. Because otherwise it's in your way when you. So, this was the one piece, right? Just that we remember.
Yeah, we'll get it.
Just that we don't forget that it's
Sure.
there. That's all.
The hope is that we get you a
And,
nice
yes.
piece.
We experienced some slight turbulence, Carl. Normal part of the flight.
Yeah, that whole little piece is gonna be perfect for us. I mean, when you're going to the left, it's doing a lot of good.
I think I actually might want to come in different, but I'll work with this a little more, see what it does. I almost feel like I need to come up with a flat blade straight at it.
Yeah, I think that would be beneficial.
Oh,
I didn't do it.
God. Oh, God. Okay.
Okay.
Thanks today.
So Sabina, why do you want a large piece of the rock as
Because
opposed
then
to
it's
small?
easier to screen it than for all the animals. And we can also better see where the organisms are coming from. Also with the small rocks we can also do, but this would be much more,
There we go.
Alright, I'm going to try and re-grab it to where I can put the flat spade under this thing.
Okay. You want me to, we're getting awful close and you're cramped up. You want me to give you some space here? feel like I need to slide right and back for you.
I won't even drop, I don't even want to even notice hold it.
Got the pilots
Too much, too much.
Far's good.
repositioning the sub.
Far in the middle. So what do we do now? Oops. Gotta set this thing down, yeah, I'm trying. Maybe you can rotate it for me. Would you be able to rotate it for me?
Yeah, I'm not gonna go into it, but
3, 2 Stunden haben wir schon.
I just really need to
so
see.
the thing you just did was amazing.
Gute 2
Opened
Stunden
it,
Zeit.
exactly. Fell right into it.
I just mostly needed
Mhm,
to
dann
be,
drüber zu saugen oder was auch immer.
with
Genau.
this, where I can put this right under that hook, you know what I mean? So like you would turn it clockwise like 45.
Okay.
If I think a good idea, so we don't
Yeah.
have to say anything.
No.
I'm gonna open a little bit.
Ich meine, der Blob, der da neben die Feder sitzt, ne, wenn du den entfernst, da gibt es den Blob, den Großblob, das da, das wegzaubst, dann musst du mehr nutzen, oder wenn du es zugmachst. Und
A little bit clock. I think that's better.
Take that.
Did we notice, does that happen like more if we have more lights on? Like, is that because we have too many lights on kind of thing?
So I just turned the inner cameras on, and it's kind of got more frequent. So let me turn those back off and see if it stops. So also that piece that you're wiggling is down here.
I want to come in like really at an angle, you know what I mean? Like, not like I have been like this kind of angle, if I can get under there.
That is the piece above it, just so you know. I mean, that may work too, but I'm just saying that piece you have loose is at the bottom of the string.
It's not this one I'm on right now?
No, so if you see this, it's right here. It's this whole piece here.
Oh yeah, I'm on the corner.
You're on the corner of the piece above it. I'm going to turn the outer cavities off as well.
Okay.
Oh, you know what? I might be wrong.
I guess I gotta go a little more of an angle to get in there.
Oh, is it this piece?
Was? Ja. Das hier.
Yeah, that's what I thought too. Yeah, you're a little high, Zach. It's below there.
Can you look down a little bit then?
Sure.
So I'm trying to go down here.
It's, yeah, this triangle piece.
Ja, da müssen wir uns noch ein bisschen rein, kann man da in das.
Yeah.
Wie es gemacht. Ja, genau, das passt ja. Nein, passt nicht dabei. Ja, genau. Na, die Kirchen.
Und bis wann sollen wir probieren? Wir sagen,
Also um 16.30 Uhr müssen wir, also sagen wir 16.15 Uhr. 15.15 Uhr,
eine Stunde haben zum Sammeln.
15 Uhr, 15, spätestens 15.30 Uhr weg, oder zwischen 15 Uhr und 15.30 Uhr. Nur um die Kurven untersuchen, der Marker ist klar. Aber da bin ich dann wieder dran.
Okay, passt.
Weil die Info 30 ist jetzt das Land.
Okay,
Dann schreiben wir das jetzt.
If I get enough of this broken, I might be able to get a jaw here.
Yeah. This kind of feels like when you're trying to help your kid take his first tooth out.
Well, I don't know.
Loosening it. Teeth.
So my wife just said, can you back up a little bit, and just pull back, and I'll stay in it.
I can try. Let's move backing up. Maybe if you lift this up while we're doing that, like basically push down. Yeah.
I made a little headway on it, though.
Okay. Let me just go ahead and reposition. Why is it?
Am I really, I think I took some ground, I was trying to.
It's like if you don't let it do its little floaty ball thing and then lock it, it floats up on it. Don't understand. I've been wondering too, because we're opening this crack. I wonder if we could get it in there and start breaking this chunk out from the other angle as well. Then again, ooh, you do have it going over there. Maybe if we keep chipping the rocks away, that's probably not a good thing. Minimize the damage area, kind of. That's not enough for section 2.
See where this, goes.
I would keep trying to work down.
Does it go here?
Yep. You're on the upper left section of it. Try to flip back at the same time. Yeah, see it move? Your color. Now you can see where it's at.
Trying to do this a minute, and then get harder.
Okay, me pulling back's just disturbing the side.
Clear up from that. That's a lot of snow, white.
Yeah. Yeah.
I think I'm gonna just poke it with a finger real quick and see. I don't know that that's the way yet, but it'd be a lot more easier than this if that'll
We'll
work.
grab in between the small piece so we don't lose it. It's still,
Let's go ahead and grab that small piece while you have an open jaw. Sure.
okay, yeah.
Quite a number of small pieces. Now you're talking about this larger small piece right here?
This one.
So I can increase the brightness here.
Is all this white stuff bacteria floating around, Sabina?
Sorry?
Is all the white things floating
Yes.
around, is that bacteria?
Yeah. And also when you try to like loosen the rock, you can see how much bacteria also in the rock, like in the cracks. So it's like kind of when you push in, you release them basically.
Yeah, there's a lot of it.
Ooh, that whole thing just moved in my jaw. We're about to get a big piece for you, I think.
That's all silicone.
Yeah, I got the piece too. Does it matter if the silicone goes in
No, no, it doesn't matter. It's fine.
there? Is that,
It's good.
it's okay?
Easy. Oh. What? Oh, sorry. You're good. Don't worry about it.
Okay.
Yeah. Ooh, fast. I was afraid of the camera for a minute.
Really changed my underwear. That's cool. You're scaring me. All right. We might have to back up again. I'll give it one poke there, see where we're at. Yeah, let's just back up. If you would. Just about 2 foot straight back. Perfect. Perfect. I'm trying not to knock you around too much. See, when you place fluids coming out of it. Drawing on it. This is the piece right here, right?
Yeah, that is the upper left section of it, so it's a little closer to us from that position.
You might go right back to the pry bar, but I just want to try it.
Dude, you might have something there. Oh, yes.
I better not knock you around too.
Yeah, 50 more downs.
And go 50% if you haven't already.
That's
Oh, ooh. Excuse me, Lord. It's 50. All right.
what?
Oh, the cruise. That got me the other day. I'm just gonna come up out of the way from that. Should be clear. Froze. No worries, man. We're gonna get this thing.
Yeah, I think you had it. Just need to get more down for. Okay. Okay.
So you're giving some more down thrust chill to counteract the arm moving this up?
Yeah, so it's moving sideways. I'm trying to use the friction of the bottom of the RV to stop that. But we can only do that for a limited time as well because we have temperature issues with the motor when we do that. So it's a balancing act.
I have to push down quite hard to get the jock in there where I need it. So I'm lifting the ROV up and shifting it around as I try and do this. So it's kind of like a delicate ballet would be how Carl would describe it.
That is his favorite term, actually. The pilot's walking you through some of the troubleshooting they're doing to try to get this large piece of rock lifted off.
I wish it was something we could Excalibur in there so I could hold on.
You know, I'm trying to Excalibur my arm in there. Once it goes so bad, look at it. I'm just gonna come.
So instead of pushing right, what if you use the yaw to like pry it, sort of, so we're not pushing this up sideways? You know? So like a lever instead of shoving right?
That's kind of what I'm doing, but
Okay.
I can do it more.
Could you put in the bar in? Let's try that it's holding and then push with the arm on the bar.
Yeah, I'm just trying this real quick because it's a lot easier to maneuver the jaws instead of the bar.
Yep.
So if I can do the arm, that's great. But if I can't, then I will. I'm just coming straight up right now, just seeing if I can just peel it up. But it's not really wanting to. But I'm pulling very hard on it right now. You see, boink. So that was quite a lot of force up. So if nothing else, this is giving us even more leverage we can stick our bar in.
Industrial Jason, I was getting you anything when you question that?
It gives you a little bit, like,
Cool. Time for repositioning right now.
Yeah, okay. Coming out. All right, we'll go over the bar again. On the froze.
Pilot's repositioning the sub so they can continue their delicate ballet of trying to get their rock pride up.
Oh, I flew too close to the sun. Got it. I'll mess around this time, really. Froze.
Okay, I got it.
Got it.
Just giving you a better gravity there. So I'll need to do it.
Okay. I'm gonna let go.
Okay.
And
Do I
I
want to
will
rotate this?
sure.
If you want
Anything I can do.
something like that, right? Or you want it more down?
That's good right there, I think. Yeah, they do. Go. It. Crazy, it's a little bit of a purplish. Oh, that's probably from the dye. Huh?
Mm-hmm. Yeah.
I think they're working right in that little zone right there.
This was very good because you see that the dye actually went in, which was very good.
Here we go. I'm gonna probably push around a little bit, Carl. Go down. Turning, turning, in, turning, turning. Back. Oh, you can see what looks like worms under there. Alright. Ooh,
Nice.
got a lot of movement now.
We almost got her. That's an excellent spot. I'm gonna freeze there. Oh, you got it, okay.
One more, and then we can
Can you
grab
take a
one.
highlight frame or something?
I wonder if I could get underneath it and help you maintain you got, but it's staying on its own.
There we go.
Boom.
Okay, slow.
All right, we got it.
Okay,
So,
it's loose. Yeah.
I'm gonna drop this so that the rock falls on top of it, so that it still kind of keeps it from going back in there, you know?
Yeah.
Just come straight out and see what I can grab.
Looks like a little cape.
There's loads of bacteria coming out of there too.
Can you make a highlight frame?
Ooh.
Maybe I should have brought my hands.
So the scientists pick this site because there
Need the
is
second
hydrothermal
to
vent
help because it's
flow
so heavy.
here, and you can definitely see that the shimmering water
Yeah.
around
No,
this rock
it's
the
not
pilots are
sure.
prying off.
He's got it.
Mhm.
I just gotta try not to crush it too much, you know?
Mhm.
That's okay, that's okay.
And
Come
take
back
the
to
second.
this.
Mhm.
That's a nice big chunk
Mhm.
right there.
Very good.
Probably a baseball size. You want a picture of that or anything, or you good? But you can see the red on the side
Yes.
going down like in a
Can
pattern.
zoom in. Oh, nice. And medium sharp and highlight frame grab salver. That's really cool. That's super good.
Okay,
You have it, Sarah? Yes. Thank you.
you want me to put it in there?
Yes.
Okay. I'll get the rest of this for you. It does kind of look like there's a bit of a cave under there.
Yeah, I think so. Very interesting to look afterwards what we.
Try and get this
Yeah.
smaller piece and then the big one in
Mhm.
the bottom. Oh, just bacteria just blowing out.
Yeah, yeah.
Yeah.
Very good. Make another highlight frame grab salver. You can see here. So let's
You got lucky too that this is just small enough to fit in my job.
yes,
If it was any bigger,
it wouldn't be possible. Yeah.
that would have been a problem. It's quite a cool cross section.
Could we zoom in again and make a picture?
Yeah, I won't drop it yet.
Mm-hmm. Okay. And highlight frame grab. You have it, Sarah? Yeah. Okay.
You want to see the other side?
No, but it came from this side. It's fine. I mean, it's.
Just flip it.
But this is like the side that was not in the flow.
I got it.
So probably we can also make a picture to have a control.
I'm dropping
Yeah.
it.
Mm-hmm. Okay. Yep. You can drop it. Thank you.
So that colorful side of the rock.
It's really nice to see also that the staining worked.
Oh yeah, it went deep. in
Yeah,
there,
it went
huh?
deep in. Yeah. Really good.
It's a good cooler.
So there's a color that is the side that's facing the vent flow.
You can see the worms in there, huh?
Yeah, these were the ones that were sitting there. You can even see them.
Don't lose our bar in that cave.
I mean, I hope I don't, but pretty sure they would like to see That's a chunker right there.
Oh,
I'm holding my breath. And slowly down, probably it's not crushing all the other stuff, if it's possible. Can we take another highlight frame gap salver?
Here is a little
Oh, I also don't know what. It's like, yeah, probably another. Can you? Yeah. That's again. Yeah. Okay. Can you stop it? And Salva, can you make a highlight frame grab? So this would be a very nice spot to look then what is potentially like new. Like then under the microscope, because what we're looking for is like baby tubes that have like a size of like a millimeter. That's what we would look for.
See a lot of stuff that's not.
But you can, yeah, but the so likely the tube itself is not stained because it needs like biological activity to get the stain. So basically we need to look inside to see if the worm is stained. So that's all good. That's fine. We've expected
I'll leave
that.
that to y'all on deck. We're gonna put this one super deep in there.
So Sabine is saying that this would be a great place to look for new millimeter-sized baby tube worms.
Thank you.
Did you release something from the back?
No. Oh, okay. Can we look towards the back?
It's deflating.
It's deflating probably.
Is it on EXP or on?
It's on EXP, yes.
So could
probably.
it still be on there?
No, it's like probably releasing. So we check if it's on, if the head moved a bit.
Okay.
So we push it more towards the off. Yes, it moved. You can see it here. So if you can push this back.
Yeah. You want me to mess with your lights or anything, or you happy there, Carl?
That's nice, so it helps with the
lower,
material.
lowers, will turn off, and then
Lowers off? Sure.
we'll actually say that we messed with camera settings. Usually it's dark.
So the team is checking on this
Let me wreck you.
sampling pump that we've shown a few times throughout this expedition. Just making sure that the bag's not getting water into its own. It's a good opportunity to refresh what's happening so far on this expedition. So right now, we are looking at
Yeah, that was a good catch.
footage from the bottom of the Pacific Ocean. We're on the East Pacific Rise. I think we're about 2,000 kilometers from Costa Rica, about 1,000 kilometers south of Mexico. And this is the Underworld of Hydrothermal Vents expedition. So this is live footage from the ROV Sebastian. And we are in the control room here on RV Falcor 2. And in this expedition, the science team is examining whether organisms, specifically tube worms, will travel through the hydrothermal vents to repopulate areas rather than going through the water column. So they're taking that seafloor subway to the next spot. And so the team has deployed 3 experiments. In the background of the shot, you can see one of the mesh boxes has been overturned, and you can see 2 of the other mesh boxes.
Is that all bacteria in there?
Yes. We also make a highlight video for like 30 minutes.
Yeah. You want us to move the bar out of your picture?
No, it's fine with the scale.
So the scientists have dyed everything in those boxes, and they're identifying what's coming through that's not dyed. So today, the ROV pilots have pried off some rock here. The science is taken back to sample, and that's what we're looking at now. We're looking into the hydrothermal vent fluid.
think we can stop the video. Let me have a look what's going on there. Is it okay to move close to the camera?
Really did get to see the underworld.
That is the underworld.
Yeah.
Hydrothermal vents right there, yeah.
It's the underworld. That's where Carl's gonna go when he dies.
It's not true. Going to a much more terrible place.
Be curious to see what the temperature is.
Okay.
Thanks.
Any more ideas, just let us know.
I was thinking about that.
You know what I'm wondering is that because we popped this off, is that gonna reduce the hot flow in the other cracks around it?
Yes.
Like the orange?
Yes. That's also the thing that is concerning about deep sea mining at inactive vents, because you can reactivate them, but of course the total energy stays the same, so you kind of draw the energy off other vents.
So I wonder if we come back like in a year, is there gonna be one giant tube going around right here while the water's coming out then?
It would be nice to observe. Yeah. Make a higher frame up here. You can also see here again the eye.
This is where we're gonna need the big bar. We're gonna get
Yeah.
that top piece that went on that, we're gonna
Yeah.
stick that big bar in the bacteria suit.
Oh, I thought we might get more of this. I don't
So
know.
it would be cool if, I don't know, to try if we can break this here to get like this side of the
Yes.
rock also.
That'd be a big bar job.
Yeah.
I'm gonna say it's possible, but I don't know.
Yeah.
I think you just get things.
Can we again a highlight frame grab?
Dude,
I'm just kidding. What kind of crazy ideas is he trying to churn up?
They don't want to talk to the mic.
where's Monica? I figure she'd be all about this part.
That's just very
Rest.
She fell asleep right at the peak of the whole. When she wakes up, just imagine her. You gotta tell her we couldn't get any rocks.
Yeah. Sorry.
That's what you get for sleep.
Yeah, our night will be long tonight. Because you have to look at all this stuff. Oh wow.
Whoa.
Stirring it up.
Ew. If you just like drop that there, Carl, whoa.
Then-
I mean, whap it, you think? I mean, not hard, but you know what I mean?
That's so crazy.
Wow.
So whap. I'll probably work straight on it down kind of thing.
Sick. What? You didn't have no idea.
About what?
Can we try to push it in?
About anything. No idea about anything.
That's probably for the better.
Yeah.
Wow. Oh.
Here we are.
Oh my god. Oh my god. Look at that.
Okay, take a highlight
Ridiculous.
frame grab.
I don't think we're gonna get that.
No.
How big that piece is.
Yeah, you'll get it.
No, we're not.
Just put, just whatever you put your mind to that.
Oh god.
Look down.
Unwrap, unwrap. Reverse wrap. Too much wrap. Overwrap. Overwrap.
Did you expect Sabina for it to be, for it to look like this, kind of hollow underneath those rocks?
Yes, no, I wouldn't expect it to be like so. Oh.
Behold.
That was a power move right there.
Oh my
Okay, we make a highlight.
We're gonna call this Chill Leavers the Seafloor.
Okay, wait a bit. Jesus.
Holy crap, alright.
Okay, I want to see what's on there a bit.
Sorry?
On the, so we've just flipped it, so it's interesting. I want to, can I use the camera?
Yep. Oh yeah, we go crazy. We're out of control. That's awesome. Anyway, go ahead.
So look at that, there's a tube worm, a little tube here. Can you see this here?
Oh, look at that. That's the underside, yeah.
That's the, okay, so we take a highlight frame grab salver. And please also write a biological observation that there are tube worms on the lower side of the basalt. And we make a few pictures here. Yes? Good pictures we need.
What is this material that looks like it's dripping?
Which one?
This is obviously upside down.
Yes, this is
These
upside
would
down.
be like,
I don't know, like it's, I think it's from the volcanic eruption, then like the lava like is slow and then probably like it's melting and it's like also getting solid very quickly.
that's lava.
So, yes.
Okay.
So all the basalt is like all lava, it like whoop, and that's also why it's hollow. So it basically rises up and then it sinks down again. But it gets in contact with the cold seawater, so it also like constant, immediately also like you know, freezes, like gets hard. And then I guess it's yeah. This is how I imagine it starts dripping. But I also don't know if the, you know, if these geological structures are like, I don't know, described somewhere. But that's so cool. Okay, so. Sorry? This red thing here? Yeah. Can we go a bit closer with the ROV to get a bit closer with our camera? But first, sorry. Salva, take a few nice highlight frame grabs. It's very important. Okay?
So if you can't tell, there's quite a bit of excitement in the control room right now. Flip. So the red circles we put up a second ago.
Okay, so take a highlight frame grab. No, thank you. We have one, we have a picture, yes. Then we have number 2. There's monitor up. I also have never seen like a tube worm growing. So this, it's Oasis. If you need more time, Salva, let me know if I'm too fast. Yeah. Okay. And another one. And then I zoom out a bit.
And Sabina, you're saying that you don't believe anyone.
That's it. It's like
down there, like in the hole, right?
yes.
This is super cool.
So you're saying, Sabina, since
And trying to figure out what is colored, what is not colored. There. It's not possible that they go from the
so you can tell the clear excitement
over there.
in the
No, I
control
don't.
room right
I
now.
have it out.
Not through the water column.
Okay, I explain you. So
Well,
a small triangle is in the box in the cooler. Now upside down, the whole piece. See here, and it's Oasis here. So there's no. It's Shia. Now we need a lot.
How did you do this? I have
Thanks,
not,
Carl.
I should not
And
clean
thanks,
the
Dave.
room.
Is that a worm inside the tube?
Whew.
Boah.
There's no way around it. It's
There's
like
no way
that's
around
like
This is like when you come from above because you have to
Yeah.
crawl inside. Pain.
Do we have slopes, right? No, I mean
The side also.
the cave.
yeah. The cave we care later about. Underneath.
I saw what you did. It's like actually, and this was, I was correct. I think this, like our attack. Okay.
Okay, good.
Possible. So
obvious question: how does it feel, Monica?
the shock is still real.
Yeah.
this
For
rock,
sure.
you
That's
got this rock up.
That's
And
amazing.
try to
Yeah.
We can
reveal
look at the size of it and calculate
kind of.
the
I was
and it
too,
was like this thing was formed and it was dripping down,
Down.
no else, and you have holes in between potentially.
Oh,
What do we say?
You?
Yeah.
Okay.
Allowed.
And it was with the mega chisel thing.
And what was
Okay,
baby's
good. Then
tube confirmation of their idea. But in my limited experimentation of is valid or not, this was, as she termed it, maybe the only chance. The first time they've pulled the mesh box. So there are 2 more test sites to check out intact and flipping it over so we could have a look at it. Moment here in the control room.
To you, whatever's best for you. I'm not arguing.
Can the hose be blocked?
No, because the pump is super strong. It does whatever it does. That's why it ripped off
What about
almost the
turning-
bag.
I mean, turning it on and off again, like you know, that maybe
Yeah.
So we are now at
What do you say that?
getting out, but put it basically on off, maybe on off.
yeah, that's what I mean.
The top on off again.
Yeah.
Because and the
But not too long because we
Yeah.
don't want the.
Yeah.
Should I look for you?
No, it doesn't matter.
man you can no can have it.
You want to go all the way to the on position or just a little bit?
Yeah, I think a little bit more,
Wanted
like
to
yeah,
go
yeah,
that position
like
right
that.
there,
I think it's
pump's
good.
already
Yeah,
on.
yeah.
Okay, that's good. Let's go back
The
on.
thing is just that we don't know.
Problem too is the position where it turns on and off is drifting towards the
Oh, really?
Yeah, 'cause it's like magnetic ordinance. So it could be that it just won't turn on.
Okay, that is all the way.
Okay, I think that's all we can do here.
Yeah, we just Salva, can you
Watch this a bit.
watch this a bit? Because
Okay, but anyway, we have the proof. I don't want it back anymore.
okay,
Okay, good.
Okay.
So we have the overview. Now we
go to the details of
go
the
to the
ones
details.
we find really clearly.
Yeah.
No?
So we probably want to start.
Yeah, this is
With
for
this
sure
one.
also one, but
We can take, so probably move it a bit to the right, then we have more tubes in. Like now we can see it's like
This
this
one.
one, we have 1, 2, 3, 4.
this one, there's one here,
Mhm, yeah, and
there's
then-
one
yeah.
here, there's one here.
5.
Yeah, this take this
That's-
one and then
that's good.
just move.
Mhm.
Highlight.
Is this all the way zoomed in?
Yes, that's all the
Yeah.
way. And we already also moved closer.
Yeah. So I love the rock.
It's
actually a
it's
lot.
super cool, huh?
This is super cool.
Yeah, okay. So is the plan to recover this rock?
Yes.
Okay.
big.
It's very big, so we have to see how, but somehow but first we take pictures before it's.
together or something.
Then here's a nice one also.
This plan?
Yeah.
Yes.
And you don't know. I mean,
Okay, so another highlight, frame grab. It's so cool. You know, just with this picture we have, that's all we need.
amazing.
Yeah.
Probably yes.
Yes, that's all that you need. Yeah, I am yeah.
Why didn't you just turn the rock in the first place?
Because it was very difficult.
Just kidding, of course.
Because
Okay.
it needs a little bit of craziness. I think.
Little bit, just a tiny little bit. This we already had, but you can take
Yeah.
another one.
Now the very right was a live one.
Yeah. Okay.
That's on the top right corner.
This is so cool. This
Mhm.
one.
This one, yeah. We take a highlight frame grab.
Yeah.
And then we have a look here. What's this here? Hoppala.
This is
This one,
no,
right? This one?
this one we have.
No, but like the red stuff. Here, this one.
Red thing, yeah.
No, this is.
This one.
If you go down a little bit.
That's probably a live one. Could this be? Can we get closer even with the rough a little bit? Can we
One
try?
thing I can do real quick.
So either
Crack
this is-
out. I put it in when we were doing the valve.
Uh-huh.
So see how that looks. If you want me to move some more, I can take a look at it.
Okay. So let's find it first again. Where it was.
Yeah, you don't know. I mean, if you can look at this
Where is
side,
it now?
there might be small ones.
Yeah,
you
yeah,
know?
yeah. And where was I? Let's get out again. Somewhere on the edge here.
The indent at the bottom, it's out of the image.
The best would be you just clean it and we put it together when it's a picture.
Ah, like as a,
possible, you know,
yeah.
because
Like this. Huh? Where? Where's, here,
yeah,
there.
but, I mean, it is also, but I mean, it doesn't matter. Just stay.
But this is alive, yeah. That's.
Yeah, but just stay and screen
Then
it
we
through,
can,
you know.
yeah. But that's also like, okay, highlight frame grab, but that's a live one. This is like, yeah. No,
This one is dead.
this one is dead, but this one is alive.
And this is also not Daphnia, this is Riftia,
This is,
and this
this
is
was,
also Riftia.
and here is Oasisia.
And this
It
is
was
also,
his,
this is Oasisia, the other one we don't see.
Yeah.
Amazing.
Okay, people should.
Yeah, go all over the place.
Oh, okay.
He has a theory.
Where are you seeing? How do you know?
Here is
So
a
the red
live
circle.
tube worm, and that's like the
It
upside
popped up on
down of
there.
the rock. So we are now looking at the
identifying-
underworld. We are now
Looking at the underworld and let's see. Oh, this is the underworld. This is the underworld of vents.
This is even better.
This is like even better.
So
Die Plenumsklick. Die Plenumsklick. Was? Die Plenumsklick.
Genau, so, ja.
So I'm gonna guess, you guys possibly want this?
Yes.
So one thought that I have, and I don't know for sure, but we have the mesh box. I wonder if we could slide it into the mesh box. When we bring it up, it would be inside the mesh
Ah,
box.
that's probably a good idea. Very good.
Yeah, you like that idea?
Yes, I like it.
Try
I
it.
mean,
don't
it's
know how it's gonna
outside,
fit.
but
Uh,
I have the color.
But we
no,
don't
I mean,
get it in any box anyway.
Is this so big?
Oh, is it so big? Let's have a look.
Even
Is
if it doesn't fit, if it just, we can secure it in there and get it closer to the cooler,
Yeah.
then maybe we can slide it in the cooler if you'd rather be in there, but
Yeah, rather the cooler, but I mean,
we can try that.
But that's too big for the cooler, huh?
Yeah, I don't
I don't
know.
know.
I don't know. I won't
I
know
think
until I
it's
get
It
it
too
in
could
close.
also be that it just breaks.
This breaks. I don't know if this is breaking, to be
Yes,
honest.
because very often when you start to break, then actually it breaks. I mean, it breaks easy.
I think
If
we
you don't
could
want
use
it to
the
break,
cool-
it breaks.
I don't know.
I think we could use the mesh box kind of like a scoop and just kind of get it, hold it, and maybe get it close to the cooler. And I honestly think it will fit in the cooler, but
Honestly,
You
too,
think?
would it be possible to bring the mesh box up tomorrow and we could use
Yeah, yeah,
this?
yeah,
We
yeah,
can-
yeah. The
we
mesh
can
box we don't need
also-
anymore.
We
Like
could
this is.
also just leave it, you know, and think about it how to recover it the next day.
Oh, yeah.
Yeah.
And we just go on, we take pictures and everything, and then we move it aside. And then we think about it really what would be the best way and stuff.
Yeah.
I have an idea too, before we leave here, if y'all want, I think we could carefully take the starboard arm and use the net cam with the lights and look under in the underworld.
Ah, that's also
Yeah,
very
Yeah, that would be
Yeah,
super
that's
cool.
It's
also
not
very
high
good
def
idea.
or anything, but.
Yeah,
Yeah, that would be
but,
super cool.
And then just take, I think you have not done all the pictures.
No. I'm like still like
But I
all
mean,
over the
if it
place
fits
because
in
I'm
the
too,
firebox, then
I'm
there's no
too, I don't
reason.
know, excited.
So the team's been working out some plans for getting that big slab of rock in the sub and back up to the surface. And I've been trying to, when the scientists get excited, capture their excitement on the audio.
All the small.
This we don't know, but
Yeah.
it's stained.
Yeah.
This shows us that
This
the
thing.
training work.
Yeah.
This is also darker, but here,
Yeah.
this is for sure the color. And
Yeah.
if you go a little bit down here, can you?
So scientists
This-
are
this
continuing
is
to
what's
examine
sticking out.
this rock
And when this is a little bit gush, this is not gush, but it could easily
and
be,
identifying
if you can look
where the
careful,
dye penetrated
This is the typical rocks I always found.
Yeah.
This
and
goes
what
down here,
Yeah.
there's here,
worms
there's
are living here.
Everywhere, small
Small
ones.
ones, yeah,
everywhere.
everywhere, yeah.
And are they staying?
They're dead. So this color is shows you that they grew, but they died.
Yeah.
With the highlight frame.
So a lot of excitement at this juncture in the dive. I'll try to recap again. So the idea the science team has been working with is that tuberm larvae will travel through the hydrothermal vents. And so in flipping over this big slab of rock, the underneath which is in the hydrothermal vents, they found tube worms. And the idea is that the only way they could have gotten there is if they traveled through the vents. So this is confirmation from their idea, which has been several years in the making for Dr. Monica Bright and the team. And so the team has been working out how to get this big slab of rock back up. And they're working through a variety of ideas. But as Dr. Bright said one moment a few minutes ago, that as we were looking at the underside of this rock, this is the underworld of hydrothermal vents. So we have exposed that now. And you can look at it, live footage here from the seafloor from ROV Sebastian. And we are 2.5 kilometers beneath the surface of the ocean. And we are looking at the underworld of hydrothermal vents at this moment. And they found what they're looking for. That is the main takeaway from this description. All the scientists' hard work and their ideas, they paid off. It's a great moment here in Mission Control to see the culmination of this work. There's still plenty of work to do, but to see the confirmation of their ideas. So while the rock slab is in its reasonably unmarred state down here, they're taking an extra close look at everything they can, getting plenty of video, plenty of photographs, frame grabs, annotations. There's a lot of people in the control room right now, and you can probably hear them in the background. So as word of this got out on the ship, the scientists started piling in, and everyone is sharing in the excitement right now. So as we mentioned before, this moment was possible through the hard work of the science team and the skills of the pilots and their patience in slowly prying that rock up and then working to get this big chunk flipped over. And as we've said many times so far, nothing is as easy as it looks on the deep sea. So kudos to everybody. And thank you for being a part of this moment. So right now we've got Dr. Stefan Sievert, Dr. Sabine Engelner, Dr. Monica Bright, got a pilot Carl Hill, co-pilot Alex Paris is here, and I am Tyler Smith, a marine technician on this cruise. And we are happy to share these moments with you.
Yeah.
And so if you, whatever parts we lose or... Do you have a second one, Salva? Yes? Wait a second then. Okay, now we can totally zoom in again in this area where you were before,
Okay.
because I think this is good. Like this here.
Mhm.
So this is 10 centimeters, so you, yeah.
It's all waiting. Should we make a highlight video, or what should we do? Probably make a highlight
The
picture.
video is on.
Yeah,
With
it's
all
always
the
on.
video,
Yeah,
Just
yeah.
take a picture now.
Salva, do you take pictures? Highlight. But now the scale is off again, yeah?
Yeah, but you can scale it later
Yeah.
because you have it smaller.
But this
then
we
it's-
already
now
took
you
pictures.
have it larger, so you
Yeah.
took pictures of
Yeah,
this
yeah,
already.
yeah. You can take another one, doesn't matter.
Yeah.
Here.
So you mean
Highlight.
those big ones are stained?
No, but this I don't know, but I don't look inside. But this for sure. This is for sure the stain that went actually in the cracks. It really went inside also,
Yeah.
which is cool.
Yeah.
And sometimes you need to, when some animals are so red that you have to look,
Look carefully.
under the microscope, under the microscope to see is it stained or is it just red naturally.
Another highlight frame grip. And another highlight frame grip. Are the limpets? Can we? No, don't get them sharp. Okay?
The limpets, I think.
They are stained. They're also the. Yeah.
Actually, yeah, they
Yeah,
are pink.
They are pink. Yeah. It's getting better and better.
If you want, I can try and focus on that a little bit.
It's just funny.
Yeah, coming. Yeah.
So obviously
There's already on the starboard side.
Forget about this.
It's good. So you can also take a highlight frame grab. Okay. You can also see here. Stain.
Yeah, I think this.
But this was like. So what should we do? Should we go on top and?
Go on top and let's just. Okay.
Should we start
And the
from the
team
very
is
top?
continuing to
Remember
inspect
where we have been?
this rock.
Oh, that's the max.
What's that?
That's in the background. It's a polynoid in the background.
Also.
But like not on the rock. It's like in
Yeah,
the, you know.
but this
on
we
the-
did already, no?
Yeah, it's a question like because I was so excited. I don't know like how well I did. So probably should just concentrate now. Concentrate. Okay. We just go on from now and then we go like this. Yeah,
Grid
good.
pattern.
Grid pattern. Okay, so you have to.
You always say when you're done, but make it
And
maybe
these
short,
scientists are
because it's all they want.
alternating between being as serious as they can to continue documenting what they're seeing
No.
with those brief bursts of excitement.
That's all.
Okay, so we can start with our little transect.
Yes.
Just say one or so.
Okay, so highlight one. You have it, Salva? Can you say something when you have it?
Have it.
You make a little bit of overlap. We can stitch them together. Okay, highlight frame.
And this is only my second expedition with SOI, but the first one was the Octopus Odyssey, and it was very exciting when we sat down and found the octopus, and this is also very exciting. So it's been a privilege to be here for these moments of scientific discovery.
Have it.
this.
Okay, highlight frame grab.
Have you?
Okay, highlight frame grab.
Have you?
Highlight frame grab.
Have you?
Als het staan niet af. Highlight frame grab.
And just to reiterate, these are the baby tube worms you're saying, wrapped around?
Have you?
These are actually relatively large tube worms.
Okay.
Though they are part of them are dead already, but at least they can
Highlight
bear
frame
them.
grab.
I'm sure to the size that they are adult because they go adult very early. Because they're very young, so very small, sorry.
Have you?
And whether there are smaller ones, we only can see them under the microscope when we retrieve it.
Can you
but
get it a bit sharper?
the fact that they live there,
Here on this area?
closed
You have to look at
for cracks from the surface.
Yeah, yeah, but I
means
want
that
to.
it must come
No, no,
from
we have
above.
like a. It's easier to stitch
Yeah.
together afterwards.
That's it. So Dr. Bright explaining further that the worms that are wrapped
Highlight frame
around
grab.
what look like stalactites on the bottom of this rock aren't the babies necessarily, but they are evidence that the tube worms
Have you?
have grown there in this spot that's only accessible if they traveled through the hydrothermal vents. So when they look for the babies, they will be small. So
Highlight
she
frame
mentioned that
grab.
they'll have to get this sample up to the surface and they can examine it further with a microscope so they can see the
Have you?
full spectrum of tube worm life. But
Highlight
visual
frame grab.
confirmation 2.5 kilometers below the surface of the Pacific Ocean
Have you?
that they were on the mark with their ideas.
Highlight frame grab.
Have you?
Highlight frame grab.
Have you?
And go a bit up. Yeah.
And so
Was he saying this one?
scientists are continuing to document this rock.
Says you want to share.
Highlight frame grab.
Because this is as pristine perhaps as we'll see it. There's always a chance in transporting samples or the ascent to the surface. They won't get as crisp
Highlight
of a
frame grab.
view of things as we are right now. And you may have heard some chatter earlier about how to get this large rock sample onto the sub and back up to the surface. They can examine it further, so we will see what they devise for that.
Have you?
Highlight frame grab.
Have you?
And I think that's it. Now we look in the cave. Let's look in the cave.
What are you gonna do now?
The bag, should we check again if it's on? It's,
I don't know what
yeah.
it's stable.
It's stable, okay.
This is a only
So probably it would fit into the cooler, we think. Yeah?
I definitely think
Yeah,
it'll
yeah.
fit in the cooler. That flat edge,
Mhm.
if you get it low,
Mhm.
it will be like tipped up inside there.
Mhm.
But I am weird t-shirt.
Okay.
Let's look at this.
Let's first look at
cave.
the cave.
So the underworld. And as much as we can.
Scientists notice we want to look in this underworld cave. And to reference some of the comments we're getting,
This is so shimmering, but
yeah, it's important to remember that this work is
isn't
done by
destiny.
people, and to get these very human moments
What's that?
in the midst of
The
a
block, yeah, the rest.
rigorous and driven expedition is awesome.
Here, what is this? No, you don't, but I just don't know what it is, that's why I'm
I
asking.
mean, I don't. It's a part of a tube that goes down.
Ah, yeah.
It's just a tube from a large tube, or larger ones here.
Yeah,
goes
yeah, here and then up, yeah.
Somewhere it came up here.
Pwah. That's so
And
cool.
this-
And if anyone is interested in
Take
Like
a
looking
highlights.
at the roots and
going back to previous dive, a few days ago we did take the camera around the ship and talk to some scientists. Let me track down what day that was, because sometimes out here at sea the days blur together a little bit. But if you're interested in seeing a little bit more of Falcor 2 and seeing some of the scientists and their work, I will determine what dive that was, and you can check back on the live stream of that dive, which is now recorded on our YouTube channel, and see what we've been up to here, see some faces, and see some of the work that's been going on here on Falcor 2.
Yeah,
covering the.
but this should be sucked into a bag or something.
Mm.
We should, I don't know,
Yeah.
because we have none right now anymore, no?
Right.
Like a water bag, for example, or
Right.
I don't know.
Yeah, we should maybe.
Ah, but it's-
Could be another.
This is the only option we have.
Could be, maybe we could use the smart bag, but that's.
Yeah, like we do tomorrow.
Oh, I see what you're saying, yeah. I thought you were
This-
asking which one. No. I think we can stick the other.
I don't know what to do with this now.
No,
I think it's stable.
I'm not
Sarah
sure.
says it's stable.
Yeah.
So
You know, we don't have to put a frame on to find it back.
Tyler, is there, are you able to get that T4 camera larger on any of the screens?
I will make it our main screen.
Okay.
I'll see it. It'll be on whatever it is streaming out to YouTube.
Okay, and do you have, I think
Can
it's
you
turned the light on.
scale?
Yeah, it's on.
This 345.
you see the scale?
What do you mean, the 10
I just
centimeters?
would like to see, this is about how long this is.
Yeah, I think about like 50 by 60 by 40, something like that.
Like this.
I think like that.
But,
There's your larger image there.
Uh-huh.
yeah.
So we are going to give you an unprecedented view
So you can
of
see
the underworld
chisel.
of hydrothermal vents here. So Alex, our co-pilot, is deftly maneuvering the starboard manipulator into that exposed section of vent that we uncovered. And we are that manipulator. It's not high def, but this is a view that apparently no one has seen before.
Keep doing that. Okay.
We're going to take photos from that camera. Are there any of any good quality?
This is exactly what I want.
Mm-hmm. Yeah. But the other side is even better, you know?
Okay.
Yeah, no, yeah,
That-
one,
yeah,
that's
that is,
it, but
yeah.
this is,
Yeah.
just look if there's the other side to this
Yeah.
also.
Yeah, that's,
Okay.
unfortunately, the hole's not
yeah,
quite big enough for
yeah,
us to get
yeah.
under it.
Yeah.
This
Yeah.
is okay. It's good.
All these filters is also
Everything.
Yeah.
Okay.
Okay, good. Okay.
I think they're happy.
What are we gonna do now? I would
Um,
shift it.
I think I would try to get the rock in the cooler, probably.
So there's a narrower section of the rock.
Mm-hmm.
I think if we got that mesh box, put it on the side
Yeah.
facing that narrower section and try to slide it into there.
Yeah.
And then it may take 2 arms. It's big enough that I think we could work together, get it close to the
Yeah.
box, maybe even tip it a little bit so we have it, and try to get it in. It's going to be a very slow process, but I think we can do it.
Yeah, should we do
Yeah.
it? Yeah, good.
Yeah.
We do it.
Awesome.
Yeah.
I'm excited about doing
Try
that.
this.
That's the kind of stuff I like to do.
Should we make one picture before that we know?
Wait.
Wait a sec, 1 second.
Carl,
Yeah, that may be a good idea, so we don't lose it. I'm sorry?
One
Can
picture
we
first.
just
One
leave
like-
this
Sure, go ahead.
far away with,
Whoop.
with this, yeah.
Okay, probably
Would you
also
like
in the
this
back.
out of the picture, maybe
Yeah.
better?
Or like even with the other two experiments, huh?
The other experiments.
Yeah,
But if
That's
this, if you can
Can-
slide the
Yeah, let me
a little bit
reposition, I'll
in,
get you
Sorry.
a much better picture.
because this is the
Mhm.
And after that view of the underworld of
What
Yeah.
do you
hydrothermal vents,
take a picture without this in it.
Ah, yeah,
we're
like
going to
that,
work
yeah.
on getting that
Go like so that there's
large
no,
slab
No stuff in there.
well, no stuff.
onto
Yeah,
the
but like
sub
yeah.
as best
And
as
even
we
with the
can.
frame, everything is on this
Click,
picture.
click, click, salva.
Okay, salva. We just have to stabilize a bit.
I think it's
Otherwise, I think it's not so super sharp, I guess.
so, I don't know.
Okay, highlight frame grab.
Highlight frame grab. They have all
Can
the
we
tools
get
in
more
there.
light?
Bunch.
Can we get more light?
Mesh box, other mesh box, which large blue worms with stuff underneath.
Yeah, just ask them to
Yeah.
get more light, if it's possible.
Yeah.
Yes, very nice. Okay, then we take another highlight frame grab. And I don't know, does it matter when we're moving and taking a picture? Is it sharp then or not? You can also see it. It's sticking here. It's like, oh,
So here
what a scenery! Yeah. Exactly the position. Yeah.
Hard working and have it over.
This tells the entire story.
Yes, yeah. It takes like the frame, the boxes, how it's like it's
Yeah.
it's perfect.
Yeah.
Okay, another highlight frame grab.
Just in case.
Don't up me.
As the team just noted, this image does tell quite a lot of the story of these experiments. So if you are new to the stream right now, this is a wonderful image to look at. So there are 3 experiments you can see happening right now. Sort of in the middle of the screen you'll see a frame, and then we see 3 boxes. So a few days ago, the team used that metal frame now at the top of your screen to mark out a site they wanted to run an experiment. I came back the next day, and using some adhesive caulking and that box that is now off screen on the left, a mesh box, they set it down over the site precisely, and they were covering up an area of hydrothermal fluid flow, like you can see on the screen right now. And so they pumped dye into that box to determine if anything appeared in that box that was not dyed, it would have traveled through the vents to get there. So the mesh on that box prevents organisms from getting in through the water column. So they came back today, they did some sampling on the box, they pulled the box off, continued to do some sampling, pried up a little bit of the seafloor to get some rock samples, and uncovered what you see on your screen right now. So they were able to flip over a large chunk of rock, and as you can tell beneath the rock there's a little bit of open vent. So they've been called the cave or the underworld. That is the underworld of hydrothermal vents. We're looking into it right now. So in flipping over that chunk of rock, they examined it closely and found that there were in fact 2 worms that had lived on that underside of that rock. So the idea the team had been working with is that 2 worms can travel through the hydrothermal flow to repopulate other areas. And so by discovering 2 worms on the underside of the rock that is in the hydrothermal vent, to them that confirms that their idea was correct, that these organisms are traveling through the vents, because there is no other way for the tube worms to get on the underside of that rock. They're not going to swim against the flow of the vent fluid coming out of the seafloor. So if you're just tuning in, you missed a bit of the celebrations, but it's a moment in expedition where the team has confirmed their ideas. There's a lot of excitement in the control room. And so now they're going to work out how to get that large slab of rock onto the sub, which will be its own feat. So again, these are live images from 2.5 kilometers below the surface of the Pacific Ocean. We're on the East Pacific Rise, and this is the Underworld of Hydrothermal Vents expedition. And you are looking right now at the underworld of a hydrothermal vent. We are peering into it.
Für mich ist das ziemlich eindeutig.
Ja, total. Das ist mir besser wie das aus meinen Buchen.
Ja, das auch nicht normal, also, aber
Das ist das. Also das ist das. Ja.
ich habe so viele Bilder von dem. Also noch zuerst schon.
Ja.
Und das sind ganz feine Cracks, was da oben geht.
Das sehen wir oder was auch funktioniert.
Just the right edges.
Yeah, I know, but
Yeah, kind of thinking
wir müssen uns jetzt nicht
the
überlegen.
open face here, right arm kind of scooping into it.
Genau.
At some point we're gonna hold it. I don't know how we're gonna do that.
Weil man
We'll
nicht
just work on it.
gebunden ist ein Rätsel.
Ja.
The other
Also
thing
jetzt
is,
on-off ist ja.
eventually we may be able to push out to support. And then we can use two hands.
Yeah.
I don't know.
That's just good.
We'll just work on it.
So,
Da muss man weiter brechen. Also, wenn man das leicht kriegt, dann ist
Ja, weiterbrechen, weil bringt nichts.
aber wenn irgendwas liegt, wenn wir wegrutschen, dann nehmen wir es natürlich.
Ja. Aber es liegen schon 4 Storner drinnen, gell?
Ja, das passt schon.
Kleinere. Und da sieht man total schön, dass das gestehen hat. Haben auch Fotos gemacht.
Gut getestet. Und gut biegt.
Gut biegt.
Ich habe gut gestemmt und gut biegt. The Queen of Glue. Sag mal. The Queen of Dye.
Der Rockbreaker.
Ja. Du flippst die sauber.
Ja, Karl fliegt
Karl.
drüber.
The King of Rocks. Ja gut, so groß this is it's not that. Also I don't know, when it lays next to it, the box is 42, 45
45
centimeters.
times 35. The outer is 50-50.
Sollen wir schauen, wie groß meine Box ist? Die Abmessungen.
Hast du das?
Ja, sicher.
Innen auch? Die Innenabmessungen?
Nein, die innen nicht, aber die außen. How big is the cooler? Ah, I can look because I bought it here.
Yeah, probably just set it down. We hit regrab.
Yeah, okay. With just the
Just
bottom
squeeze it down a little
out.
more, and then you may have to go for an easy let go. But
right, let's see if I can just.
I think you can go down a little more from this point.
The van is losing the battery.
Ja.
Was machen wir?
Corners down. Let it go.
That's the
Settle.
Da kann man gegen Vorwärts, dafür ist es irgendwie.
Ich glaube, dass das verrutscht ist.
Ja, aber
Dafür muss er
wie
raus.
das mit wieder richtig rutschen. Das ist
I
das,
did
was
not
funktioniert.
expect that to happen.
We have another
You've got the
sequence.
back. You've got the back. Go ahead and try to tip it backwards.
Okay, the box is, the outside dimensions is 92, 45, 46.
Doesn't it say the inside dimensions?
No,
A little bit. Try to
it's
support.
this is 50 here.
They say 3 centimeters less.
But it's important to know how much beer fits inside.
Yeah, they say the leaders, but
Oh, yes.
the leaders you can put.
Yeah, okay.
But then, yeah,
So I'm hoping that when we start pulling this, eventually it'll rotate a little bit, so we're working where we can see, but
You know that the inside of the box is like about high like that because of the weights.
Yeah.
it'll have to be lifted somehow to
yeah,
get in.
yeah.
Yeah. Hoping, so you see my ram here? If I put the ram out, I'm thinking eventually we will corner the box in this corner
Ah,
and support
yeah,
it, and I'll
then,
have two hands to get
We'll
under and lift it.
Okay.
It's gonna be hard.
Yeah, yeah.
The other, I mean, I'm not sure if it fits in the box because the outside dimension of the box are 92, 45, 46.
Out of the cooler, you mean?
The cooler outside.
Okay, do you wanna not do this then?
Yeah. I'm not sure if we can close it.
Yeah, the problem is if, you're right, if it doesn't fit, I don't know, do we
I
get
think
it back out?
the more important then we have it as a whole because we're sure it would break. Do you think it would break if we just
Just
lift
pick it
it
up.
on this, not really where the hole is, but if I array a little bit and just let it fall a little bit?
Or not, I wouldn't let it fall.
Or
We could try
Or
to pick it up from two sides and put it on that spot on the porch if you're not concerned about getting the mesh box today.
No, mesh box, we don't care.
Yeah, so we can try. I mean, but if it falls and breaks, like,
But
We'll
if
try
it
our
goes
best.
need
to
to
the
cut-
water,
we
if
need
it
to-
travels to the water,
I think that, yeah.
I mean, fact is also, I need to put my microscope in a different way because I cannot put it on the table, so I cannot put that rock on the table and the microscope, then I need something lower where I can put the microscope and the rock because I can move my microscope, but I cannot, I'm not,
Ideally, if it breaks in two,
that would be ideal. But I think
When you
even
grab-
putting it here is tough because
Yeah.
that hook
the,
is like this high, so we would have to get it very high and then on.
yeah, and my
So-
point is,
Could you grab it here and like
Background
try to,
conversation
like,
you're
I
hearing
don't know.
is the pilots working with the scientists
See how
can you
stable it
try
is.
to shift it away from the
to
hole?
figure out how to get this rock sample up to the surface.
Go to the box.
So in an ideal world, we could get the whole thing up there,
We need to do it before.
but it'd be a challenge. So they've discussed kind of sweeping it into that mesh box that's right in the screen there. We're trying to get it in the cooler. But the size of the sample they pried off might be a bit of a hindrance. So yet again, another example of the problem solving that's necessary to run these operations and the dynamic nature of doing deep sea dives. Things could change. You might uncover something you didn't realize. You might need to find out a way to bring a huge rock up to the surface. That's key to your research.
We'll set this aside at the same time. Let's just go ahead and move this here out of our way. Go ahead and set it down right there. Just go there. Oh, perfect. Okay. There.
You just look to push away from the hole and let it see the sturdy.
I wouldn't even lift it away from the hole yet because though you use the hole to get your jaw underneath it. But she's just kind of wanting to grab and see how it acts. If it's going to start crumbling, then I think we're going to stop.
Right.
But if we're able to pick it up, it's more of a test than anything else. See how it goes. Still a little unstable.
Maybe we should still, in case,
Okay.
open the box.
Mm.
You know what I mean?
Yeah, I can do that.
Because if it's closed and then you have it, then you cannot open anymore.
I'll take the starboard arm and I'll work the box. I'll let you.
So we got pilots handing off manipulator controls here.
Yeah. Ja.
Jill's gonna work the starboard. Alex will work
Also
the port.
das hätte ich mir nicht gedacht. Also was so rausging im Raum, das
So
war
they're going to get that cooler open.
Ich war echt worried, dass das die leuchtet. Aber das zum Glück
And do a little test run to see if it's possible to pick that rock up, or if it breaks, or if it crumbles.
Box just yet. But if we need to push fully out to open, and if I push fully out, I think I'll interfere with him getting this. So we may just take it one second.
One step at a time.
So the team working through the process here. Gonna take a stab at gripping that rock first. Hold off on opening the cooler because they'd have to porch out, which may hinder things.
Yeah, just go full open, see if you get your bottom down under it. You can, when you lift it, you'll fix your angle. I might need to help you by supporting the other end of the rock so it doesn't just slide. Okay, I'll work on that.
I just hold right up to me there. Pull away.
And that's so completely moving now. Alright. Okay, I'll stop there. Yeah, so it's there. It will eventually support it.
So they're using the starboard manipulator to brace the rock.
I got another idea. So the issue is that the rock is at a different angle than the arm can get.
I see.
So we're not flat.
Oh, yeah.
You might actually get a little bit more. Yep. Okay, so if I take the big bar and he's able to just pick up this side, slide the bar underneath it, so then you'll have a better shot of it. We're gonna go for that. try and get that bar without.
So using the manipulators is all about finding the right angle for things. So right now the port manip can't quite get low enough to match the angle of that rock. So I believe the pilots are going to try to get the big pry bar wedged under there. Maybe. Do something to help correct the angle.
This is a good news. This is a super story. Yeah. Also,
Because we are not posting, because Schmidt Ocean Institute should be first one. So.
very good. This work is so verrückt, oder? Mhm. It's a little bit more.
Ich hoffe nicht so gehen, wenn der bricht.
Yeah.
Okay.
Probably. Okay. If you smash it down, it probably breaks here. If you, I think it's not fitting in the box. Like it's worn, just I don't know what.
Well, this is not bad. There's another piece that broke off below.
But what is like this? Is like. Well, can we try to put it in the box as a whole?
We can try, yeah. Let's just try it. If you can try it, then we try it.
I think now you have it more easy to break.
But I think also we have it easy to break.
Like ideal would be like 2 pieces, but that's like
But if it's 4 pieces, I don't care either, because it's more important to get it up cold or to get it up close than to get it up dry. Because if it's in the box and we cannot close it.
I think we should have broke it before we grab it then, because if we have it, I don't know what, I don't have any control really of how it breaks.
Yeah. If you pull the other arm in the center,
Are you worried that I'll damage a lot of the area that you want?
no, it's also not good. Is it fitting in the box?
with the fingers.
So, Paul's right there, so he's got it.
He's
right?
got
so
it.
There is 10 centimeters. So let's do another quick
10.
idea of how long this is. So it's 10, 20, 35. This direction?
What? Let's see. This is 10. So basically this is 10,
Yeah, 30, 35.
20, 30, 35, more 40.
Yeah, the other direction doesn't matter. It's like it's not just
also-
But this here,
It's also going to fit in the box.
Box like
and
this.
tip
Yeah,
over, so
yeah,
it's got
yeah,
a little
yeah,
extra.
yeah,
I'm
yeah.
not saying it will fit.
No, but probably we can try.
let's try.
Yeah, we just try. It's like yeah. Bit.
Piece.
Picture here, but
Okay, let's try it.
The damage so much of what they
No, we tried. We put it in.
So the team's using the science lasers to measure the rock and try to predict
42, 45
whether it'll fit
inside.
in one of the boxes.
Number 9 exactly. Okay, the length is
Actually, that's probably the portrait.
goes like this, maybe 40,
So you're gonna clear on top of that one? So if I was you, I would just ease it on up. And I'll start portion. This can take as long as it needs to take. We're going nice and slow.
In the background, you can hear the pilots working out their strategy. So in this instance,
When we get it close, we may actually get a better idea too.
Mhm.
If we think it's
Yeah. Take
got
a
the co-pilot
highlight.
working the manipulator, and the pilot's running the camera, bringing the porch out,
Yeah, yeah.
keeping an overall idea of what's going on here.
Out.
Mm-hmm.
I mean, they will, for sure, they will, something will break, but I mean, we have the pictures, you know.
So with the porch out, they can now open the cooler.
A bit in the back, because the other rocks would block it otherwise. Again, popped up the two camera views so you can see what the pilots are predominantly working
Do your
with.
best to get it towards the back end of it. I really think that's gonna fit.
Using the science cam for the
I thought it
head on and the situation cam for the top down.
it's not gonna fit. It's gonna be tight.
So they're able to check
Oh, yeah. It's,
where that object is in three spaces. 3 dimensions, sorry.
I think it will fit, yeah.
It's
got a little more tilt. Yep. Oh, yeah. It's definitely.
The only problem is the jaws are already all the way open.
Yeah, you're probably gonna have to wiggle it out, but
I'm over it now, please, so. Wherever it ends up. It's good.
I know it's good.
yeah, you have good. You're good.
ik denk dat het veel andere zo van die kan mee is niet live.
There you go. There you go. I think if you yaw right, it will use the back of the. Yeah, it's gonna pitch it. It's gonna twist it kind of out of your draw. Yeah, just like that. Don't shoulder right, just pitch because you're really far right now. There you go. Keep doing it.
So it was fortuitous that this sample of rock is just the right size to fit in the jaws, but also it's just the right size to fit in the jaws, so they can't open the jaws anymore.
I wish I could help you.
Oh, it's gonna come out.
So the pilots are working the angles and trying to let gravity do the work.
Yes!
Yes.
Wow. Basketball.
Slam dunk.
And a tremendous amount of excitement still here in the control room.
Okay.
Tense moments watching that rock sample
I don't need
slowly make its way over to the cooler.
Oh, the rock. Treat it one. Think about it.
Uh,
Think about it.
Uh,
How is this like the one strong one?
That's A, it's the
Oh, it's the directions.
No, it's like, I think.
Mine just crushed up on the ground on this one.
Yeah,
He was trying to crush it, but it's not.
RV bridge.
So,
Okay, I'm just bringing you around to about 120, 15 degrees to port. Understood.
There's a small little thing.
Can you cut it in?
Thanks, guys. I've got that on my screen now.
It doesn't have to go over the basically got enough. So you can just use the front of the Sharpie or
also, but you'll be able to look at it. The other side I cannot look at it.
We'll knock it off.
Now,
Now what's the important to deal with it?
let's see
Hopefully that
if
lid
it goes
doesn't
into it, or if,
Might have to push on it.
It's like that.
Oh.
yeah,
And then
no, yes, no, watch out a little bit, no? Is it possible? Can you wait a moment?
Sure.
Can you look inside there?
without the rod.
Because I think it's just like 1-2 centimeters like too high sticking up, but I think if you can, I don't know, move it a bit,
Yeah, it's
just a little,
that part in the back
Yeah.
sticks in. Open it up a little bit.
so I think that this is like a bit too high to close it like fully. So I don't know if you can move it just a little bit, that it's more like sliding in a bit. And I don't know if it's like better to go like in this or in that direction.
That's a good forward. Yeah. Get away from the wheel wells.
Yeah.
That's not no. No, that's not possible. Okay.
It's stuck, no? Is this not stuck a little bit?
Sorry?
Is that more stuck, no?
A little bit, yeah, but we get.
But
Yeah, so there's other rocks that's
Yeah,
stopping it from
yeah.
going that way, and if it goes on top of those, then it's going to stick up even more.
Yeah.
I
But
think it's the
now-
other
Back,
direction.
we're already.
Yeah, we usually do.
But now I think if you can now push it back again, then it's a bit more like that, you know. And I think we're good. But not as slow.
Just put it straight and rotate the wrist, so it's kind of-
Or should we just try once more and look with,
I've just got this back to restart.
No, it's different. Like it's
It
different
looked
now.
like. You want me to try to close it?
Yeah.
Yes, if
because
you now
I
it looks
close
good.
Because it might actually be okay.
Yeah.
It's just here you can see. So it's so difficult if freedom is a bit.
Yes, now it's good. Or is it? Yeah, now it's good.
And
Yeah.
now we put the bungee cord.
That's-
on
and we
top
don't
of
open it anymore.
Bungee cord, and we
Yeah.
leave the arm there.
Yeah.
Bungee cord and come up. This. Or, no, we put it in the voll, ich kann noch was.
Ja, sicher. Na, warum nicht? Gibt keinen Grund, warum nicht, oder?
Is that bad or is that
We come back tomorrow then with this.
I see it.
You see it? Oh. Fertig. Das ist für auch das Sammeln. Fertig. Wie viele Box haben wir noch? 4. I don't know.
You just have to think about what to do next.
Da ist ja ganz einfach, das müssen wir einboten alles. Jetzt müssen wir mal unsere Sachen einboten.
Also das müssen wir ja noch drauf geben, genau die Box. Vielleicht können wir die sogar noch mitnehmen.
Welche Box?
Nicht die Meshbox.
Achso, ja. Fertig.
Ja. Guten Morgen.
Guten Morgen. Sie ist doch vorbei. Oh.
Ja.
Ja,
Good.
gut.
Nice.
Die Meshbox wird machen wir nicht.
Ja, das meine ich.
Jetzt müssen wir alles bearbeiten.
Ja.
Jetzt machen wir das mal einboten.
Genau, zuerst die Meshbox und dann...
So the team is just cleaning the porch up a little bit, and now they're gonna secure that cooler full of samples.
Oh, let's be pushing the box.
Force in better.
There's no twist. Can you do in the middle somewhere? With this,
Can you hook it not here?
is it not like hooked more in the middle?
Not anymore. That was on the previous bow box, but the bow box is not there anymore. Now the hook is right there in front.
So in order to get the right angle
Oh, this is the
to bungee that cooler lid closed, they pull the porch in a little bit.
Comes underneath the frame very tight. When we get on the side of the corner, we
Okay. Super.
Oh, did I hit the wall?
nice. Perfect.
Get me out of here.
Thanks a lot.
That round of applause goes for our outgoing pilots. Chilling out. Got a watch change here. Swapping Chris in on pilot and Russ on a co-pilot.
Das können wir morgen auch mitnehmen.
Okay, also dann muss ich jetzt auf Platz. Oder?
Dann machen wir nicht mehr auf.
Nein, aber die Meshbox.
Die
So
Meshbox, ja.
the next thing is very good that we recover the meshbox.
To get that budget over. Just throw this up and proceed like this.
Yes, that's probably more secure. Did we put it like here, you mean?
Yeah.
Yes. If it or is it fine?
That was for the box.
I know, I was trying to think if there's anything better that you could go on with.
Done is
Yeah, that probably would be away anyway. Mesh rock.
Can we ask Stefan Stefan, do you want to take over? We have to think about what is next, and we just have to recover the mesh box and then sample some tube box and biobox spot.
Yeah, sure.
And we have to.
Where would
Power the chisel, the laser, the mesh box to recover, and then go around the corner and far away from any damages.
Yeah,
Should we have somebody on see the channel,
Going to school.
Yeah,
The masses are lifted up.
we have a bipolar so that we could all do that.
So mesh
Yeah.
box,
Mesh
small
box, chisel,
chisel,
both chisels.
No, the large one can stay
The
there.
large one
That's one
stays.
space, okay.
The smaller one you
Yes,
grab,
sure.
the frame you leave with the mesh
Okay.
box.
Just
So
the mesh
only
box and
mesh
the
box chisel, and
Yeah,
then
two bones.
2 bones into biobox
Box
board.
is
Yeah, and no
4,
muscles.
not small. Also the other one is
Thanks.
organic.
No, of course, yes, of course. It's amazing. And we have 2 more to go.
But it's all we need, you know? It's like,
Yeah, but if
it's
you
just
don't breathe-
extras. Yeah, it's
breathe,
extra, extras. But this
Yeah,
is like the.
yeah, I know.
So with this
Next Firebox. Firebox 4, the sample will be taken. Firebox 5, just more Firebox 4.
And the next sensor.
with this watch change, science party taking another second to celebrate.
That's it, right? After Riftia, we are done.
Yes.
And,
all the
Can
remaining
order,
time.
find careful, small ones.
Yeah. Okay.
back to business. So they are sorting out
We don't need
the plan for the rest of the dive.
Okay. You need that? Someone?
It sounds like maybe we'll do some sampling. And we've got Dr. Stefan Sievert on the science mic now, giving us some insights. And again, that's Chris is piloting, Russ is co-piloting, and this is still
We are just cleaning up here. So we
Tyler on the other mic, a marine tech for this expedition.
or the porch, sorry. And then pick up the small chisel and put it in the. No, yeah, we leave the big one and put it in the milk crate. And then we're gonna go on a hunt for a small riftia.
Whoops, sorry, wrong porch. Porching a little bit. There you go.
So we leave the big chisel.
Do you want to sample the riftia before you pick the box up? It's going to be obviously in front, taking up quite a lot of
Would it be easier, you think?
I think it might be easier than landing out.
Yeah,
So,
sure. I think we could always come back here, right? Yeah.
Yeah, be able to see.
So how much time in the end do we need to account for coming back and pick up
How
everything?
far do you think you want to go?
Oh, very close. We just have to make sure that there are no deployments of anyone where we pick up riftia, but I think there's plenty of riftia in the area. So, how much do we need in the end to clean up everything? Like 20 minutes?
The box might take us a little.
Half an hour.
Yeah.
Okay, so we have.
Other than this in the box, that's pretty much it, right? Oh, and the frame.
Yeah.
Frame won't take long.
So we should be back by 4 here.
Okay.
Right? Is that okay?
That works. Then.
Okay, great. Okay, just to get an idea.
All right.
Perfect. And we can really look for a small riftia this time and take our time. So in the end, we just need to pick up the mesh box then, right? Yeah. Yeah, I mean, if that makes it easier to collect the tube worms, yeah, we should go for that.
Definitely.
Watch the
See the camera.
not the camera, about the cooler.
I was wondering with the hook, would it make sense to hook it over here? And would
Yes,
that
the
be
box
more
is
stable?
built up.
Yeah, right. But yeah, okay.
And it might. It'll be really
It's
small.
Okay.
pretty secure there though. It's not going to go anywhere.
Okay, good.
Alright.
Okay,
Can you
let's.
manage to touch yourself on? All in there?
Sure. Just if you look down right there.
Hmm?
It's halfway underneath the porch. And then
What is that?
if you look right here,
Yeah.
yeah, half of that box is underneath the porch.
Oh, I see what you mean. Yeah, okay, I get it. Okay, so it's quite stable. Yeah. Okay, great. Yeah.
Alrighty.
Yeah.
And we will move over to grab the frame.
Yes. No, actually, I think we can leave the frame.
Okay.
I think we need that. Salva, can you confirm that we can leave the frame? We can leave it down here, right? The frame.
Okay. Alright, wanna go over here to this? Go over here to this and small, or you wanna go further away?
Yeah, we just need to find an area for like small riftia. Smallish riftia. I'm not sure any of these might be good size here, but
Well, that is a beautiful view. of the tube worms exploding out of the hydrothermal vents down here. It's an incredible landscape.
I think so. I think Jessica is looking for ones that are like 30 centimeters, like a foot.
Yeah.
So if you might have a better idea of how big they are. So these might be too big. I'm not sure. What do you think? Yeah.
You start taking it out and it's like, oh.
Oh yeah, exactly, yeah.
I don't know what's in the middle there. Like if you look to the right here, Russ, in the sit camera.
These?
Yeah.
Yeah, maybe that's the ones in the back there you see.
I'm saying the ones right here in the front.
Alright.
You know, laying down, there might be some smaller ones.
It does look a little smaller, that's for sure.
These, right? Yeah.
Yeah.
Yeah, maybe. It could be, and if we can, you know, if they are, maybe we can determine.
And as the sub sets down, you can watch things retract
And then if they are,
as they feel get closer.
get the whole bunch. Are the lasers on?
Yeah. But probably can't see we have to look at the camera a little bit to see.
Mm-hmm.
It's your thing.
I think it does.
Oops.
Looks good right there.
1, 2, 3, and then just one more that.
Yeah. Let's see where it is.
Yeah, if we can get that whole clump here.
Don't know if one of the giant ones.
So where's the?
Lasers are on. Where'd they go? Curious.
Yeah, it's just on the rift.
Where
If
are
you
the
go to the
lasers?
left a bit, you can see it
I don't see them.
right there.
That's it.
Oh, here's one. Where's the other one?
So. Yeah, got it.
So right now the team is using the lasers to measure these riftia.
So that whole clump is.
They're looking for riftia of a specific size. So sometimes the lasers are a little difficult to make out.
Yeah, I mean, if we can get that whole clump here, that would be good.
Okay. Well, try the one. Use it more so you get more success.
But they've identified a clump to grab, and they're going to go for it.
More leg.
Do you think like a scoop could work even? No. Better with the arm.
I don't think you'll
They're
ever come
really
up off the
hard.
ground with a scoop.
Mm.
The attack's quite hard.
Yeah, they'll
Yeah, yeah.
have to pull them out.
Then they would go into the firebox.
So if you haven't watched these collections before, when Russ is nudging the tube worm, he's not harassing it, but he's getting the red plume to retract inside the tube. And then they'll try to grab on the empty end of the tube and pull the whole tube worm out in one go if they can.
I think you get that bottom one a little bit.
Yeah, I know. Unfortunately.
So the team, the science team,
Okay.
prefers to get these samples alive, and we can get them to the surface. So they'll really probably have to do their best to get these samples for them in the way they'd like.
Yeah, that looks like a good size.
He's coming.
Okay.
It's
No.
not.
Well,
They're
then-
usually long.
Oh, okay. Hmm.
That's probably
Well, extra.
that long.
Yeah, okay, a little too long, I think. But
Should really.
so we should not go for the for the wider, maybe this one and this
Smaller
one.
diameter, yeah.
Yeah, right, exactly. Approach now some more.
So we collected Stefan?
Yes, so we have one right now.
This is sample-
Going back.
Sample as well.
Yes.
When you're ready.
I don't know if it's gonna hit that thing, so I'll come up slow.
I mean, if it actually fits in this box, it might not be too bad for Jessica. Let's see. But, yeah, that's not. I think that's a decent size.
Compared to the other ones we grabbed before, it's pretty tiny.
Yeah, maybe try these.
Yeah, that's very
This
much.
one.
Over the one on the left there.
Grab it road. Exactly.
Grab what she loses. It's pretty good.
That one's
Nice.
Oh, that's nice. Yeah, very good.
Some good sampling.
Watch out.
Nice grab. Okay, number 2. Should we try this one here, maybe?
Sure.
Is there one in? There was one in before, right? Oh, I'm not sure. It's an empty tube.
No, that was.
There was something sticking out, right? Yeah.
Yeah. It was. I don't know. It's like. Just come back here.
The crab is waiting for us to drop it. Yeah, that might be it. Maybe it's a little more firmly attached. Yeah. Hmm? Yeah, I thought about this one too, yes. Yeah. Yeah. Nice. Let's see.
This keeps coming.
No. Stop.
It's not far away now. It's a nice tail.
Ah, good. That's a good size, I think.
Yeah.
Yeah. Nice.
Concentration on not too much.
Watching out.
Actually, good.
little bit more. Hold on. There's another one just above where we were. You can see it just sticking up over the other columns.
Yeah, knock.
Oh, there's that one right there.
That might be quite difficult because those ones that are clear like that rip really easy,
Yeah. Alright.
which I noticed previously. What I'm thinking is if I can move that larger one out the road, maybe hit one of
These
these
two?
smaller ones, yeah.
There's three.
See
small
this
ones
one?
there.
You think you can get that? You can also go to another place.
Well, but I'll just nudge him.
Okay.
Out the road, so
Sure.
we're not
I mean, this one might also be quite small. This one.
Yeah.
Yeah.
just nudge him over the road there.
There you go.
Got a crab that is undeterred by the
It's competing with
giant
us.
titanium claw descending from the submarine at it.
Yeah, I'll go with that. He doesn't want to go. Do you want to try the short one? Yeah.
Yeah. Oh, this one. That might be the. Yeah. Okay. We'll see.
So if you're just joining us, this is live footage from the bottom of the Pacific Ocean. You're watching the camera feed from ROV Sebastian, Schmidt Ocean Institute's remotely operated vehicle. And we are on the Underworld of Hydrothermal Vents expedition. And earlier in the dive, we indeed saw the Underworld of Hydrothermal Vents when we were able to pry up a rock sample and get a glimpse inside one of these vents. And
It's really difficult to get to.
that actually confirmed the team's
I'm going to try it.
questions of what they're coming out here to do, whether two worm larvae travel through the hydrothermal vents to repopulate an area rather than, or maybe in addition to, moving through the water column. So the team revisited a previous experiment site they laid down at the beginning of the cruise, found that amazing discovery. We've got samples from that site, packed them up in the blue cooler on the sub,
I just find that the
and now they're doing some additional sampling because we have some more time down here on the seafloor. And currently we have Dr. Stefan Sievert on the science mic. Chris is our pilot, Russ is our co-pilot. I'm Tyler, Marine Tech, and I'm about to duck out here for a little bit, and we'll put Tomar in my place. And he is raring to go. Alright, so thanks for joining us here. And we've got some more time left on this dive.
Right. See what you know, can't reach it.
Alright, I'm gonna do it a little bit. I don't know, it's over on the edge.
Yeah, okay, let's move on.
Alright,
Come on, just a little bit. Alright.
I think that one to the left of it, and going out the way might be easier. It's whiter white.
So there, yeah.
No. See that? Yeah, the one that's going that direction.
This one?
Okay.
Or
No, not to the left. The one that's.
this one?
Yeah, that guy.
Yeah.
I'm just saying it's whiter. You know what I mean? Might not be as.
Soft.
Yeah.
Yeah, these other ones might be younger ones where the tube hasn't been fully developed yet.
Yeah, it's
Okay.
easier. I might not have anything in it though.
I don't know. Yeah, we know. Yeah. It's definitely small enough. This is like the, I think the third or the fourth?
This is the fourth.
Fourth, yeah.
Okay.
Okay. Maybe let's go and find some other place.
Okay.
I think these smaller tube worm aggregations might be a better target than the really big ones.
Alright.
Maybe I think maybe going this on this side.
Alright.
I think these are all really big ones. I mean, I'm not sure. We might find some smaller ones in there, but I think maybe on this side, in this direction, there might be some other.
Some small ones right there. Very big.
And as we fly over here, you might actually notice this hole in the ground with the chisel next to it. So that was actually the site of experiment 1 that was today recovered with spectacular results. So we definitely have proof that the rift jar might colonize the vents from below, from the underworld. So that's really exciting, and I'm sure you will hear more about that in the coming days.
Pun intended, but that's groundbreaking discoveries.
Yes, that's a good one. Low-hanging fruit. But it truly is. Okay, let's find some smaller ones.
I don't see anything too small.
Unless you can make the other side, see that what comes here, where the slab is?
Like, see, maybe, I don't know, these are empty maybe.
There's that one small right there.
Like over here.
These look smallish.
Alright.
This one.
Okay, maybe you look smaller, that's true.
Like this one here.
Yeah, we can probably
Maybe a few
put a
of
few
that.
options there.
Maybe, maybe some here, these.
Yeah.
Might be. Yeah, there might be a good number of smaller ones, right? Like these here. Unless they go down really deep.
Project this one?
Yeah, we can try. I'm not sure. It might be deep in there, but. Wow, you moved the whole rock. Look at that.
Quick.
Oh no. Oh shoot.
Well, it's still there though, it's just a bit shorter.
Yeah, the one was fine.
It's like, where's my room?
Maybe this.
Yeah, I see.
This one here.
Not these ones.
Why?
You have to walk around the corner in the big clump. No, not these ones. They mostly they're dead because you don't get them out.
Okay, we got some already. Okay.
Yeah, but this is also not here.
Okay.
Go up, go around. You see big lava flow.
Okay.
Big
Yeah.
applications where there are small ones in it.
Okay, we'll try that. Okay.
They
Yeah.
would be better.
Okay.
Yeah. Because here the most of them you kill.
Yeah, we got a few now.
Three
We got 4.
3 undamaged, 1 damaged.
Yeah.
Yeah. But the other one place is much better for this.
It's more size.
Because you just have to lift the muscles from below with the scoop.
Yeah.
A little bit, get it up, and maybe then you get the whole scoop. Russell, you were collecting such a clump, no? In like the first dive or second or so.
Okay.
It is easier.
Just have to be careful not to disturb any experiments.
These are all massive.
Yeah, this is where we were on the other side. I'm just making sure that. Yeah. I've been over this way a couple times. Probably I can get down there, but I can get around to the other side, hopefully.
But we're also not supposed to get any mussels. So.
Yeah, what I'll do is I'll just strip
Yeah.
them out the road.
Okay, yeah.
That's the easiest way to do it.
Okay. Yeah, but they all look really big. But maybe we get lucky, get some small ones.
Anywhere between 3 meters and 2 meters.
Yeah, it's really big. Let's continue to the right.
Have a Manta visiting the camera.
Oh, top side?
There's small tubes in the edge there.
In here. Or
Yeah, there's a couple. Just this landing's a problem.
Figure that out. You want to check it to the left or?
I mean, check. Maybe what about this side here?
There's a bunch of
Yeah, we can work that out.
there, you know.
Yeah. Or we'd rather handle left than anything really. I don't want to back that around. It's too nice, too pretty.
Yeah, maybe some of these. But
I mean, we don't want to come in from the port sides. There is. Oh, they're just drop weights. Alright.
down there.
Okay, so down some more.
Well, maybe look legit.
I don't know, it's kind of far. We can give it a shot.
There's a lot of ones over that side too, seeing that.
I mean, we could easily,
We can try
no,
this as well. There's a bunch here, so I can rake that and see if we get some muscle blow.
I
Start from there.
can come down into it if you want to go over there.
I mean, they look all rather big, but I'm not sure.
Second over there. The little downward ones.
Oh. Yeah.
That's
We got 4 that seemed like a correct size. Trying to get more. We have time.
She said, I have like an hour.
Yeah.
She was Monica was suggesting we pick them up with the scooper.
Hmm?
There's a spot.
I don't know what's going on, dude.
These look pretty good.
That was crazy. That
On
was
the side over here.
Fishy 17. Alright.
relocation.
So there should be a laminated page that has all of our
I don't think we got it.
has,
What? You need a paper?
it has my,
It's okay,
it has
you
my
want-
power supplies on it. It was just meters in here somewhere.
Here's,
Yeah, she's 17.
oh, 7, right?
17.
That's what I see last time. Back to up to so that's what I see on my side,
Okay.
which is seems like well, take a note.
Alright.
Right, try to read. It'll be okay because I'm.
Alright, where do you want to go? Which one do you want to try from around? Move over there.
I think that we should drop down and try and start with that.
Some-
Up in there? Alright.
Like in this area here, or
Yeah. Could we scoop it with the
I think-
scooper thing, or is that?
I'm just going to rake the muscles out of the road.
What a beautiful sight. Where is this?
It's
It's
just on
it.
the corner from where.
flower flow.
Fava Jean flow?
No, flower flow. I think it's
Flava?
called.
Well, it's just
Who's that?
J.
I have no idea. I think Lauren named that. But the ones that we have right now were taken near the experimental site. So
Okay. This is that white structure next to the two rooms.
that's Bishop's Spire.
Is that all just that
Alvin
map?
Miller.
Okay.
Right, you can just pin it with that, I'll do it.
Right here?
I'll do it. We'll work it out.
That's going to be really close because there's nothing to do here. It just keeps going. Alright, we're solid.
Thing is, we don't know how long we are. So we might pull them out and they're longer than expected.
pull that up a little bit.
I saw the footage. First of all, it's great to have a shot.
Yeah, in the middle.
Now, the super, that first collection that you guys got with Manta that first day.
The big, huge thing is barely fit with the.
Yeah.
Yeah.
That you got with the super, that had a lot of small ones on it.
Alright.
Is that?
We have it.
Yeah, we're not. Yeah, we shouldn't get too many mussels. I think so.
No, give it a try.
But
Alright.
I mean, that's probably too long, right?
That one's
Just get it.
not.
This is in the road. I'm trying to
Remember, do the ones right by the manipulator on the bottom.
Like some here.
Can you pull them out? You try that first.
already got 40 in this method, so yeah, it works.
It's quite long.
Yeah.
It's probably too long, Justin.
Need a little.
So,
Yeah, quite well. Come on.
I think that's
These tiny ones here.
yeah.
You zoom in at it?
we go forward. That one I had.
Yeah.
Oh, this one here. Yeah. But yeah, I was.
Grab all three. Is that four?
Yeah. Cool, that's it. 3 ones.
Well, so much for not collecting mussels. Are they all?
So downward doesn't mean you're short then.
Yeah, I think these are all too long, right?
Alright, maybe we need to fly a little bit around to see if there's.
I mean, that was the advantage of being at the experimental site, because
Yeah.
we could. But Monica wanted us to come here.
Well, she said that we could take our time.
Yeah, no, we have time, but
Yeah.
it's not unlimited.
Okay, Chris, you go.
I'm wondering if there's like some on the periphery that could be smaller.
I mean, these seem to be all. I mean, they just stick out barely, but then they go down deep. Maybe fly around a little bit more to find another site. I think it's best when we see them, like, in total. I mean, this could be a good, maybe.
Yeah, on the bottom of that pump.
Yeah, yeah.
Alright.
At least we see all, them where they actually. I think that could be good. They're not hidden.
So you're thinking like this one,
Yeah, yeah.
this one.
Yeah, yeah. I think.
Yeah.
Yeah, I think this is. Yeah, I think
Like these kind of size.
this could be a good spot. Yeah.
There's the loose, tiny.
Perfect.
That's wasting bubbles there.
Yeah, I think that's a good spot here. Like some of this here.
Push you out a little bit more space, but so there's that one,
Yeah.
that one,
Yeah. I mean, yeah.
this.
There's not a plume sticking out right now, but maybe. Yeah. It's best to go for some that have the plume sticking out.
Now, if this scooper could scoop underneath that little.
Yeah, that's a good one. Nice. Okay, number 5. Yeah, I think this is probably a good spot here.
Yeah, these are fine. Even just crack those a little bit. If you look at the box, you'll see the ones we've got, they're not particularly large, they're reasonably good size. Just leave them dark, look at the rest. It's going to count out.
Like this.
Yeah.
This one. Yeah, this one here. And this one maybe, yeah.
Although I have to like grab one of the bigger ones just to get the. So.
Yes.
Oh yeah.
Nice. Might be 2. Yeah, I think this is a good spot. Fill it up.
Guys, empty. The price to pay.
Right. Okay, let's see what I'll open it out.
Yeah. I don't see anything in that little book that way.
This one.
Yeah, that would be in there.
Yeah, there you go. No,
Don't see any blood.
no, some of the others look good too. Yeah, just put it in. I think that's great.
I can feel it.
Yeah, that's what. How many? 3?
I think so. Yeah, I've had it real good.
Nice.
I can try to put you out a little bit more, make the angle easier.
What's in the blue box?
Rock.
Big rocks.
The Underworld.
Yeah, that looks really cool.
Yeah.
Yeah,
So that really shows that the rift gears are colonizing from below.
that is so
Not only rift gear, Ossetia was there too.
on the bottom of that rock.
Yeah. On the underside.
How heavy do you think that rock's going to be?
7 pounds.
That might be a team effort.
40.
We'll get a fitter Alex out here.
40 kilos? Something like that?
Yeah, it's impressive.
Yeah.
And it's solid. It's not like the super crumbly stuff.
Yeah, no, it's a solid plate, basalt plate, yeah.
Oh, man.
But it broke. I mean, it's beautiful, right? It's like right where the experiment was.
Yes.
It's
and
like
there was
amazing.
stuff living underneath.
Yeah, totally, yeah. Couldn't have been better.
Yeah.
What a successful story.
I mean, really, yeah. Like, it's like, oh, well, we can figure out if things are coming from underneath there by looking on top, or we could just flip the rock over. But, like, I don't think anyone anticipated that cork of a rock popping out. And then there was a void space underneath it with stuff.
Yeah.
Yeah. I saw you all reaching down with the manipulator and kind of peering around in there. So.
But I mean, the whole series of experiments, it's so well planned and executed.
It's
super
insane.
impressed.
You're going to get
I'm
so,
super
like, the
impressed.
amount of bacteria that was inside the boxes
Yeah.
is insane. But yeah, this rock flipping is really just a cherry on top. Definitely some decent data and evidence towards that sort of Vernian process of life living under the crust.
Might be difficult, but this one right here, the one, yeah, the curly one in between, right there, yeah. That one's a good size, and like, I feel like if you keep calling.
But it's also like a testament to the productivity, of this,
Oh, 100%.
like what happens below, like all the amount. But it's interesting. This mesh box, basically, what it did was extending the subsea floor environment into the,
I heard you talk about that, and I mean that's really it, right? You're basically making the subsurface
more
like taller, like
more, exactly.
Yeah, you built an extension on,
yes.
and that whole environment then can shift into there, and then you just take that away.
Yeah.
No, it's a amazing experiment setup, and seeing it
no,
today.
I'm super impressed, like how Monica and Sabine planned this and executed it.
Yeah.
I mean, they tested everything in shallow waters, and it's really great that it worked to the deep ocean on the first try, and it's like a charm, right? I mean, it was like amazing. 1, 2, 3.
Just it's absolutely amazing.
Other small ones, like grab.
Yeah, and it's really great that with this cruise we can shine a light, so to say, on the darker part. I mean, deep sea vents are dark in general, but the subsea floor.
Yeah.
You could see as like the dark side of the vents, but it's really great that we can highlight that a little bit more and how important it actually is for the overall productivity and connectivity also of the system.
Yeah, and I was thinking, like, obviously, we have push cores for soft sediments,
Right.
and you do have like drill ships like Joyde's Resolution that can get rock, but it wouldn't have captured that gap. You would not have seen that without being able to open it like that.
Yeah, no, and I mean, Joyce's Resolution, I mean, they go all, they can drill into ocean crust for sure, but doing this on axis is super challenging and not really possible. They try it sometimes, but it's like the rocks are so brittle and porous that it's not really, you don't get good recovery. So.
So having the ability to take a crowbar down to the bottom of the sea.
Yeah.
Just
I
incredible.
mean, like a smaller rock drill might be really interesting, to sort of localize.
But
deeper.
all you would know maybe is that you punched into an open layer.
Yeah.
But to actually be able to lay eyes on the underside of that rock was incredible.
Right, that's crazy.
But it's super cool also, like, we are kind of, I think we are nearing like the halfway point of at least of the dive program, and so it's so exciting to have that kind of highlight at this point in the dive program.
Yeah. I didn't catch it on the screens, but did you all stick the heat probe, the temp probe underneath there?
No, I think that's gonna happen tomorrow. Yeah. Yeah. Because they took the water sample early. I mean, they could have measured temperature, but I think we're gonna take a water sample.
We take the water.
Yeah.
That'll be fascinating. Fascinating to see.
When the rock was still there after you removed it.
I remember that. Yeah.
So there's that one little one in the background there.
No, that's incredible. Absolutely.
Can we Fauna, please?
And as we're looking at this, you might notice also that we see different types of tube. I mean, most of it is Riftia, of course, but then we see all this other type of tube worm here with a different tube. That's Ocysia, I believe.
That's the one that kind of looks like a horsetail or bamboo or something.
Yeah, exactly. Yeah.
Okay, so that's obviously
Ooh, look at that. And then,
Nice.
and then there's like a few off of it. Off of that one too.
Has anyone counted? Do we have like 10, 12?
I'm counting.
Hmm?
I'm counting.
How many do we have now?
The tubes, are the tubes sealed at the bottom or are they open?
They're usually sealed, but a lot of them have false bottoms, so.
This is the 13th. Only no.
3 0?
No.
1 3.
Yeah.
13.
Yeah, we
Okay.
can't stop now because that's not looking.
That is.
No, we have time. Yeah.
Yay. Let's fill up my vessels.
Yeah, that's great. Another exciting day for you, on this dive. Get some nice, decent-sized tube worms.
That chunk of basalt that we have on board then, so would that be part of the event from 2005, 2006? That's only like 18-year-old basalt?
Yeah, yeah.
Wow, okay.
Yeah, definitely. Definitely.
Incredible.
Yeah,
Yeah, it's so exciting if you think about plate tectonics and to be at a place where new ocean crust is born.
Yeah, you're like twice as old as the rock. Yeah, it's incredible. Yeah.
And then you just move a little off axis and you see slowly the sediment accumulating as a dust first, and then it's
Yeah.
definitely not.
It's big though.
It's big.
It's way down the bottom. Those little ones are way down the bottom. So I can grab, I can hold on to
It's
it.
going to do a nice little, just a tiny little. See where we go.
So at the very bottom of that, you have two small ones. Might be able to pick them off.
How do the worms attach themselves to the substrate, typically?
Well, like a straight.
They're glued down. Something similar to like almost like a byssus on a
Yeah, but it's not as strong.
mollusk or something. Yeah. No, they seem to pluck off kind of
Yeah.
easily. Yeah. Because I'm more familiar with like almost like the tube worms we've seen elsewhere, the little doodly ones where they're like glued down to the rock.
Yeah, the one, and then there's the ones that you see in the sediment that have like.
Oh yes, yeah, for sure. Yeah. Or like Christmas tree worms in corals and stuff where they're like boring into it. Yeah.
And then the sedimented worms actually like they can get their sulfide from the sediment.
Mm.
In addition to their gills.
Do you think these riptia they excrete something at the bottom that to make it like
Yeah.
mussels do, like to attach?
Yeah.
Yeah.
And the histosome.
It's fascinating.
What is the basalt beds made out of? Yeah.
Oh, of the mussels?
Yeah.
Good question.
You see?
Protein? Yeah.
Right.
14.
Nice.
15 is like right there next to it. So.
It's nice to do this in a relaxed, no rush manner.
This is a great site though.
Yeah. And it's good that we don't, here we don't take up any mussels unnecessarily, so it's good. Only what we need.
It's one of the benefits of having an ROV with these manips and stuff and the cameras right there is you can be pretty selective. And having the scientists like point at the screen, be like, that one, get that one there. It's nice, versus more blind technologies that can get you a lot of stuff, but maybe more than you need or in a condition you don't want.
Yeah. I mean, working at vents submarine. I mean, either remotely operated or actually human occupied.
Yeah,
because like it's so spotty, and there's no way you can get the samples you need from above without having that.
No, and it's rough and everything. I wouldn't think like a dredge would work well or
right. I mean, if you work in the abyssal plains, right? I mean,
Yeah.
taking a sediment core, it doesn't really matter if it's like 1 meter there or there.
That's it. Yeah.
But here, it really matters.
Yeah, I'm sure, like, we've talked about, right, we see the ballast weights, and many of those ballast weights are from subs. I'm sure some of them are from sensor platforms that are left behind with releases.
Yeah.
But it's not like they're dropping those blind from the surface. Those are probably getting placed by the submersible and left in place, you would think? Or?
Yeah, actually, that's a really good point. So it's both in a way. So when we have deployed instruments, for example, with the weight, so we guide them in to some extent.
Yeah.
Trying to get them relatively close to the site from the ship, like with a cable.
Yes,
so we get them like to 50 meters above the seafloor, and like in a good distance away from the vents, where it's safe to drop them.
Okay.
And then the next day, the submarine or would come and
reposition it,
and then reposition and bring it directly to the vent where we need it, for example.
No, that's a great question.
I mean, with our visa, it doesn't matter too much because we have long dive time, especially if you stay down longer for for several days.
Yeah.
But for example, with Elven, dive time or bottom time really matters. So you want to be as close as possible, to minimize the
that makes
transit time.
sense. Yeah. But yeah, like you were saying, I mean, it's kind of like the abyssal plane thing again. You can just huck a lander, and it'll probably land on sediment, right? Like,
exactly. Yeah.
Be scared.
That's a little one, eh?
An interesting part of bringing the deployments back is so you release the weight and then they have buoyancy, flotation to bring it back to the surface.
Yeah.
And here in this where we are, you really have to think about the thermocline, which is super strong. So you have to have enough rotation to actually penetrate that. So it could easily happen if you don't calculate that correctly, that it gets stuck at the thermocline.
Oh, yeah.
And it might just be like 40 meters below the seafloor, but below the surface.
Yeah.
But you have no way of knowing where it is and recovering it.
Yeah, that not something I would have thought of. So that's why I'm not out here putting them in. But yeah. Fascinating.
Yeah, have to put on some additional flotation.
All right, for any folks maybe just joining us, we're in probably about the last hour or so of bottom time planned for today. But if you're just joining us, this dive has been fantastically productive and just fantastical in all sorts of ways. If you scroll on back through, today we recovered experiment number 1, so that's what was early on. Now we are collecting worms, and we've been collecting pretty successfully here. Nice variety of worms, but the experiment number 1, if you've tuned in for earlier dives, we have had these mesh boxes, these mesh box experiments on the seafloor, and the idea was that we would come back and pick those up and see if anything had come up from the vent fluid beneath, and then we were going to collect some rock samples from that area as well. But Sebastian, with the help of a large pry bar, successfully just basically popped the cork on the seafloor and opened up to a subsurface space that was there. And indeed, there looked to be life in that space. And of course, one of the questions of this expedition is there some sort of, I don't know, I always think Vernian, like a Vernian process by which life is traveling through the Earth itself, being carried by hydrothermal fluids. And so a huge step towards answering that question today. But we do still have a little bit more bottom time here. and we will be streaming the Ascent over on our YouTube. So if you're already on YouTube, you can just sit tight for that. If you're on our Facebook and we finish up bottom time here, and you want to hang out with us some more, you want to see what we might find on the way back up 2.5 kilometers through open water, that'll be over on our YouTube. But like I said, it's still got some time on the bottom for now.
No, you'll have some good ones now,
gonna
huh?
keep me busy.
Yeah. No, but there should be time in other days actually where we can make more collections.
I know. I don't even know how they are like.
See one, yeah. Just get into it, that's all.
Yeah, so you just knock all that stuff out of the way to see what's that's
I just want to see what this one seemed like 20 markers then, yeah. It's really good.
I don't know, not seeing it anymore.
Yeah, I think this one might be.
Yeah, I can see it. It's just right in that mist.
Oh, I mean this one here. So.
There's a couple there now.
Alex, have you actually been at deep sea vents before, or is this your first visit to a deep sea vent?
I've been to Endeavor Vent Field off of the Wanda Fuca.
Oh yeah.
Yeah, I was on Nautilus for a fellowship, and so I've seen those vents. Right, those are more chimneys, and that was working with Ocean Networks Canada. So more equipment installation and checking up on instruments and stuff. A lot less biosampling. Yeah.
Yeah. Yeah, very interesting. I mean, they have this network, right?
Yes.
And,
yeah. Ocean Networks Canada has
the cabled observatory, right?
The cabled observatory network.
Yeah.
It's incredible. Yeah, and you know, I wish it's something we could do everywhere, right? Like imagine a cabled observatory that came all the way out here, right?
Yeah.
Incredible. So
quite challenging, yeah. But
How those tiny ones have that? Yep, we're gonna have to use the other arm to peel those off.
But that's also interesting. Like, you know, each vent field is, you know, there's some similarities, but there are also distinct differences. In,
100%.
In the animal communities that you see, and
yes, this is
geology,
a very different environment to like Endeavor. Looks like some Pompeii worms down at the base there.
You can pick that up a little bit, get it easier for you.
were you on the cruise earlier this year to the Mid-Atlantic Ridge? Or?
No, I was on the ship as we crossed the Atlantic from where it had been being worked on in Spain in Vigo. Came across with it to Puerto Rico, actually testing a lot of these systems now that we're using for the live stream. But no, I got to watch those from home, and just looked incredible. Yeah, we have a lot of good dives this year. After this, of course, we're headed down further south. We're going to take a look at some vents down that way, and yeah, no, it's going to be very, very interesting.
Yeah, that's really exciting.
What's the cruise between?
I'm not doing that.
this cruise in the Galapagos?
I'm off after this one.
It's-
I have to
it's
look.
a sediment
Chris, can you
thing.
pick this up because I can't reach?
Yep, I got it.
Yeah, so over on our website, you can pull up the cruises page. We're going to be looking at microplastics on our way down to the Galapagos. Then we're looking at hydrothermal vents in Galapagos. After that, we're going to do a second cruise in the Galapagos looking at vertical reefs on some cliffs there. Looking at just how fine can you actually map the sea floor.
Can you spin it?
Yeah, good.
And that'll bring us towards the end of the year.
Everybody chill.
Yeah, and then if you look at our 10-year plan, you can kind of see where we'll be scooting around for the next few years.
I got a little bit more. Hold on. So we freeze it. Alright, it's frozen for you.
Nice.
Like a cheese stick, just kind of peeling it off there.
I was just wondering if it makes sense to put in the whole thing, but it might be too large. You know, I have a good collection.
I count 17 so far. I think that's the same rule we got. Russ said he wanted 20, so yeah.
I mean, it just makes sense, you know.
It's a good number.
Yeah. Obviously, so. All right.
Hi.
Probably 19.
A full bouquet almost.
Yeah. I'd hope the bio box is tightly closed. I will make sure it's tightly closed.
Okay. Yeah. Actually, look, this one here is a good one.
How much exchange actually is happening between, I mean, there's so many different submarines or research submersibles available. I mean, is there like exchange between different groups using different submersibles? Or?
Yeah, so we have a thing called SOVA. What's it called, right?
Yeah, which means island check.
No, fair enough. And that's a collaboration started by Jason Williams, our mechanical engineer, who, along with Russ, and we'll design Sebastian. But we have interactions, we have meetings, we have shared technologies, ideas, thoughts, visions, whatever.
Mm-hmm.
That's been going on for a year or two now?
Yeah, almost.
Yeah, that's great.
And I know that on the upper levels, all our management, between us and all the other research companies and buses, they're in communications all the time.
Mm-hmm.
Yes. Okay, that was your last one.
Okay, I think that's perfect
I'm
time.
not the cards for you, tell you what.
Nice work.
Thank you so much.
I'll keep you busy in the next couple of days.
Yep.
Yeah, I think that's good timing because we can head back now and then wrap up. When we get the mesh box.
Good job.
Good timing. Yeah, excellent. Nice work.
You guys.
Look at this box. It's
Oh, can't look at that.
I'm gonna go take a quick nap because it's gonna be a long night.
sorry.
It's like a disco dress. I'm not sure it's going.
Thank you.
It's gotta be like
Can we make sure that none of the tubes are kind of at the edge of the box, preventing it from closing, completely closing? I think I'm looking. Oops. I'm looking at here. That's. Oh no, I think it's fine.
Yeah, it's
Right?
Yeah, it's fine.
I can sort of move.
Yeah.
Cool. Alright, I've got another arm and I'll store that as well.
And do we do a bungee on this one?
There is no bungee on this one.
Oh.
We can.
Is it possible to put the arm on it? To.
No, because it'll be underneath the.
Okay.
It's that one's actually good. It's
Yeah.
it's the good one.
Okay.
So,
That's nice.
it's got-
Okay.
it's got new seals on it. So
Okay.
it's, decent.
And we changed the orientation of the
Okay,
ramps
that's
so that
the
it'll.
arm secure.
Ah. Okay. Okay. Cool.
Alright. Pushing it.
Got our camera menu up there. Kick you over to another camera real quick while we get that fixed.
Nope. There it is.
Perfect. Every once in a while we got our menu that pops up there.
Disco party.
In celebration of today's dive.
Is that a soft bottom yet or not yet?
No, we
Okay.
have to go collect that.
Just making sure.
Hey buddy, sorry about that. You swim away. Saw a picture of it. Yeah, we only saw a couple octopus and that's about it. Some crabs. That's all I've seen when we're sitting down.
There we are at the experimental site.
This is driving me crazy. It's good thing I don't have like epilepsy. But this was the first time that we saw an alarm, I think, associated with this issue.
Yeah.
So,
Could be an overload.
we saw power supply 7, I think it was 17, but I don't think it was 17, so yeah, it might have been just.
I put it both ends of the check. I definitely see 7. I'm not just being 17 maybe in case. No. Double check it.
Where did they put that box? Just right where it was?
So the bar should be.
Not the bar, I don't pick up the bar.
The boxes in this area. Is it like they're all in the similar area?
Yeah.
Go back up.
Yeah, I mean, it should be very close. I mean, I think the side, the experimental side one was here somewhere.
It's not far away from that.
Yeah, that's the frames.
The boxes really.
Yeah. There's the bar. There's the hole.
Yes.
There's the box. There it is. Alright. Do you have a preference on how to set up the rest?
Not really. Whatever you think. I mean, I'm going to be grabbing that.
Yeah. It's right here. Yeah.
So now here you have a really good view of our experimental sites. There are 2 remaining experiments. Upper left you see experiment 3 and then next to it experiment 2, and what we recovered today was experiment 1. So the mesh box was removed, and that's what we are picking up now, so it's empty before we go up to the surface. And in the back of that mesh box you actually see a hole in the ground, and that's where the mesh box was situated, and we were able to break off a piece of rock, and that rock actually had proof of the idea that rift deer and other tube worms and potentially other animals actually use the subsea floor as a avenue to recruit from below. So it actually had smaller and already grown tube worms on it on the underside of the rock. So that was a very exciting day that we had.
Get it.
Porter's
I'm going to use the port.
Okay.
Yeah, you just got there. Yeah.
And you will probably hear more about this, or not, yeah, very likely hear more about this in the coming days.
I think what's exciting, I mean, it's all exciting, but like this was box 1 of 3.
Yes, exactly, yeah.
Still got more to look at.
And definitely, if you didn't see it, go back and then check it out. There was like there's some really nice footage when we removed the mesh box, and a lot of microbial biomass also being produced down there.
Did you, so obviously like we were expecting some because there was stuff growing on the box.
Yeah.
Did you expect it to be that full?
Yeah.
Oh,
actually,
wow.
I mean, based on previous experience.
Yeah. I guess that's true. Yeah. If you've done stuff with it. I was quite surprised by the amount that was in there.
Yeah. Yeah. And I mean, that's just a testament also how much energy is available in this system, right? So
sure, yeah.
Where the hydrothermal fluid mixes with the seawater, these organisms have an abundance of energy to grow and to fix CO2.
Yeah.
And yeah.
I mean, if you're able to chemosynthesize, or you have a relationship with someone who is, this place is amazing for you. So.
Right. Exactly. Yeah. That's the place to be.
Yeah.
And it's also interesting, that when we removed the mesh box, I mean, it was like we were in a snowstorm, basically.
Oh yeah.
And when this site was discovered in '91, they had dives with Elven, and at that time they realized there had just been an eruption a few months before that.
Mm-hmm.
And there was like this type of event happening naturally from a lot of different vent sites where this material was released from the subsea floor environment.
Well, and that's it. Even when we're just cruising around, you'll occasionally find these little clouds of like flocculated bacteria. You know, it's definitely a thing. It just kind of rains down.
So now we're making preparations to put the mesh box on the porch and secure it before we actually end the dive and come back to the surface.
At 100 meters before we touch the surface, you need to make this.
Yeah. Thank you, Moira.
I just saw it.
So that's we need to fire the Nistons at 100 meters.
100 meters? Alright. I wonder if
Can I leave this for you here? You remember?
just stick it right there.
Excellent. Okay, cool.
Can I get to see the arm?
Yep. Which arm do you want to see? That one.
Okay. Tell you what.
Zach, what do you think about taking four destinations, pushing down everywhere? I get it. Splash out. Just
have to keep an eye that it doesn't leak or something, because it's not normally under those conditions, but other than that, you should be able to.
Alright, that way, because there's like 21 small rift worms in there for our chest, and that would make for them not to.
Get you some tape?
Yeah.
Successful collection.
I get a lot more on your shoulder. That'd be the best way to. Yeah, so that'd be the best way to get down there with everything else extended. The back side of it. Yep, that's good.
Okay, hi, it's Juliana. Our final task is to put this on our porch and stow it and head up.
You're gonna do it with the strobe light action. No.
Party.
Alright, I'm gonna put the camera out a little bit.
I'm gonna let it go there. Imagine this space.
Oh, nice.
Oh,
Perfect.
nicely done.
Like Tetris. Score.
Sometimes gravity is useful.
Yeah, right.
Alright, I'm gonna porch in. Should have pulled the
That's how you do it.
pull first. Better idea.
Magic hands, you see.
Alright. Get that
bungee up out of there before I porch it. So it get caught.
Yeah, and on this matchbox actually you can see still some of the remaining silicon that was at the bottom to seal off the vent, and also this caulking material that we used to patch up the cracks as well.
More
So-
you look at that.
I think it might be okay, you know.
You can barely see it, but yeah, we'll brighten that up and then zoom in.
So these mesh boxes have worked really well. Great design by Monica and Sabine.
There you go.
Nice.
Box is on.
Alright, I'll porch in some more.
Box is on. You're hitting. Oh, good. No, you're good. Mic drop.
Alright, that's it. Anything else over here? Before we go?
No, I think.
Oh, yeah,
Yeah, at 100 meters.
Oh, 100 meters.
Yes.
Open 100
so
meters.
There you go.
remember that.
No worries.
Yeah, we leave. Not here, but we will.
Oh, 100 meters, like above the sea.
From the surface.
From the sea, so,
Oh, that's
yeah,
cool.
yeah.
Yeah, no worries.
In the yeah.
Excellent, 100 meters.
Alright, one last look.
Nice work.
Yeah.
Great job.
21.
Got
Excellent.
Logbook. 21.
An amazing day at the seafloor. And here, just before we leave, you see the hole in the seafloor.
Yeah. That explains why we're flying so weird. I'm like, I forgot we have a huge rock in there.
The previous place where the mesh box was, that we just took on the basket or the porch.
You want to set the.
And now we are all excited up here to get the rock back, and Monica and her team will go to work, and everyone else to work up the samples and to really prove the hypothesis that we had going into this cruise.
No way of telling what could come from that hole tonight while we are not watching.
Yeah, tonight will be exciting, like a lot of work. Also with the rift jars that we collected, a very good collection. So there will be a lot of experiments that being started or being prepared tonight. And so sure you will hear more about that the next coming days.
Cool. Okay, thank you. If you are watching on Facebook, we are at our limit and we will be ending the Facebook stream shortly. If you want to watch our Ascent and Midwater, you can hop over to our YouTube at Schmidt Ocean YouTube channel, and we will be showing the Ascent as ROV Sebastian gets recovered. We've had a manta ray just doing laps along the starboard side of the ship all morning, so hopefully we'll see a lot of cool stuff on this Ascent.
Yeah, and in general, we see a lot of really cool stuff when we go up. I mean, we sit in the conference room sometimes and then have meetings, and we always see some interesting things and then stop our meeting to watch as well.
Yeah.
It's really exciting.
Yeah. It's like our own personal dinner show.
That's right, yeah.
Okay, see you guys over on YouTube or tomorrow for our next dive.
Bye.
That probably cut off.
No, I don't think it works like that.
Mic's still on. Thanks.
Okay, YouTube, thanks for sticking with us. We are making our ascent. It'll probably be about 2 hours till the surface. So point out if you know what animals we're seeing, and we'll see you at the surface. Just so you know, we're testing some lights right now, so if things keep going out, Sebastian is fine. We're just testing some lights. Okay, while we're playing with our lights, I put on the aft camera, or actually it's starboard mid camera, and we have a manta ray. It's just out of screen. It's right under the door, but I will come back online when we can see it again. So in general, we have a lot of critters just attracted to the ship when we sit on station. For this entire cruise, we've just been in one area. And so when we recover, we'll generally just drift overnight and then come back on station for the next dive for the next morning. So that just attracts a lot of animals because we have a lot of just lights on all the time. And so we, if you go out at night, if you're on the Falcor 2 on this cruise, you can see we almost constantly have sharks around the ship. There's been a lot of mahi and some small tuna as well. Someone also saw a whale breach earlier today, so it's kind of like a special treat when we're sitting on station for multiple days because sea life will congregate around us. Okay. We are doing our final operation, which is firing some Niskins at roughly 100 meters. We have the floats on the surface, so those will be coming off shortly. And yeah, that egg yolk jelly was amazing. So thank you again for joining us for the Ascent. This was a cool one. We'll be back at the same time tomorrow, and if the manta ray is still around, we'll give you as much as we possibly can, but we haven't seen it in a while, so fingers crossed. Send good manta ray vibes to the control room. See you guys tomorrow. Okay, in our back view you can see the manta. Hopefully it'll get curious and follow us.
It has to be beside on our side of the boat.
Some of the IRV pilots were saying how, in their experience, manta rays are very, very, very curious to these vehicles, and they'll get up really close to them.
There's one level where it's like
He's coming on the left. He's coming. He's
Yeah.
going to come around our starboard side if he leaves that canter.
Yeah.
There he is.
Oh.
He's still in the back.
Yeah, he's
Oh,
coming.
look, he's coming for you, dude.
Oh, that's so cool. Look at him.
He's right behind you. He's about to be right overhead.
He's coming over.
With the 4K.
Yeah, can you look up with the 4?
It's already is.
Okay.
Can I move it?
Yeah. Oh, these are some of my favorite animals.
Wow.
Can we get more lighting on him?
You got the manta in front of this guy.
Lighting?
He's in a really bad
Ah,
spot.
I see, okay.
have to do the boomers that caused that one.
Yeah, no
Just
worries.
letting you guys know that the mana's giving us a show here, so.
Want to stop on the wedge?
No, just keep doing what you're doing
If
right
he
now.
keeps going shallow
Be
with us,
prepared,
he'll
we may
get
stop here.
Yeah. We're freaking out in here. This guy has been with us for since the morning, I think.
least.
All day, I think.
Yeah.
6 foot off. Got it.
He's in the rear again.
Yeah.
Is it half camera again?
It's counting limits.
There you go.
Right there, if you come up a little bit,
Yeah, but
next to, especially
Oh.
the person in front of us.
7 foot up.
yeah, they're super curious, man. They love flying around the RV. Like, I'm not gonna even have to do any work. That thing's just gonna check this out.
Yeah, that's cool.
So I'm gonna throw some more.
8 foot up.
He's coming under you right now,
Is he?
I think. I thought I just saw him shoot under you.
Drone in position.
Copy. We got 2 more floats on the first set, and then we got 1 more set of 3. Alright, they got a couple of more floats to do, but I think you got, what, 30, 40 minutes of battery on that thing, so
Yeah, good. I got like 25 minutes.
yeah, we'll definitely be up by then.
Please.
You just want us to come out with lights on again?
Yeah.
Alright. Can you help me remember to turn the
10
back
foot
on?
up.
It's
Slip it
time
on.
like crap for this matter of video.
Can I help you up?
The focus is up. This is just so cool to see. He's been following us. We were really far from the ship when we first saw him. And he's just hanging out with us.
Oh, good. Yeah. So you brought the soda to the water before? Yeah.
That's amazing.
Yeah. So
Oh.
You got plenty of room for them back up.
Yeah, back up all the time.
the box open.
Currents kind of.
Gotta get it, position everything out.
Look at all the remoras.
Let it sit on the surface for a minute.
Cool.
Bravo to these pilots. Not only are they following this manta ray so we can get the shots, but they're also in the middle of recovering the ROV. So, utmost focus to do all of the things.
As an interest in knocking it, yeah, so likely it'll come up and you'll stop the swing and the rotation on the damper, and the FC might have you come out and see where it spins. We might have to spin it for you, and then when we're ready, we'll.
Are you coming up or
Yeah,
do you
you'll
stay at depth to film?
release the locks.
All right, guys, start painting.
Yeah, we're recovering all the time.
We're covering all the time. They gotta do one last set of flips, so we're doing both.
And you can bring the docking head slowly overhead. Yeah, that's good. That should be good.
Sweet. Yeah, that was cool.
Tomorrow at Highway.
This is also a highlight of my life.
Slow down a little on the winch. On that. See if it comes in a little closer.
Okay. We're 12 meters down.
Winch for a second. Let it come in. Stop. Let it come
Close
back.
to the ship. There he is again. Such beautiful creatures.
Back a little more for us. Girl.
Yep, backing up all the time.
Good. There we go.
Amazing.
Okay, we're gonna end the stream shortly. That was such a great show. Thank you guys for tuning in. Hopefully we'll keep seeing cool things tomorrow on the Ascent. And we'll be diving at the same time. So join us again tomorrow. See ya.